Welcome to day 2 of the BDC Summit. This is Alex Kozalewski, our communications manager.
Hello, everyone. I'm Alex. Thank you all for joining us today, early this morning, if you're on the East Coast. Before we jump right in, I want to give everyone a couple of logistical details that will explain a little bit more about this year's summit. So since we can't be together in person, we have asked all of our student teams to create a video that we will be sharing today to explain their project. And after we've played those videos for everyone on the broadcast, we will ask all of our judges who have joined us to come on and ask some clarification questions and explore the project with the students as well. So we've also created a couple of community resources for everyone. So if you're watching on our summit page on our BDC website, you'll find those just by scrolling down a little bit on the page. And those resources are a Miro board, which is a place where you can go and share resources. And there's a What Biodesign Means to Me page that you can write, which is a thing that we've been asking our community for the past couple of months. We also encourage everyone to vote in our Community Choice Prize, which is a new prize we're offering this year. And that's open to the public. Hopefully, if you're voting, you've been watching, and we ask everyone to vote by tomorrow, Thursday the 18th at noon Eastern time. And lastly, if you're joining us on social, we will be tweeting and Instagramming. Find us on our handles and also the hashtag BDC 2020. You can follow that if you want to see what we've been posting. That's it. Over to you, Dan.
Thank you. So yesterday we saw a lot of fantastic projects. If you didn't get to see them, you can find them on our YouTube page. You can also go to the individual school pages. We posted the student videos overnight, so feel free to check that out. This morning we're starting with something a little different. We'll be airing 2 finalist projects from the science that are eligible for the Science Sandbox Prize for public engagement. The prize was created to encourage students to create projects that amplify public discussion among audiences and encourage discussions that challenge cultural norms around human identity, social relationships, and ultimately humanity's place in the living world. So we're going to get started with our finalist teams. They are actually 2 teams from NYU, the IDM program, so that's very exciting. In the same classroom, competing with each other. Let's see what, let's make this very exciting. Yeah, so we'll be airing that, and then we'll be joined by John Tracy from Science Sandbox, and we'll be asking the students some questions. So let's air the first one.
Close your eyes. Take a deep breath. Now, picture the first summer that comes into your mind. When was it? What does it smell like? What does it taste like? Open your eyes and imagine a world where it's possible to capture those smells and flavors of your memory and use them in the kitchen. What if we told you that with the aid of microbial cultures, we might just be able to make that future a reality? Introducing Culture, microbial kits that bring your memories to life. Using cutting-edge genomics research, we are finding new ways to bring life to your kitchen. Our custom kits capture the smells and flavors of a particular time and place. And our recipes help you make your kitchen creations even more unique and delicious with a touch of nostalgia. We have work to do to make our vision a reality, but as we like to remind ourselves, the future always begins as science fiction before it becomes science fact. Hi, I'm Nikhil.
Hi, I'm Sarah, and we are the Microbial Memories team. You just saw a speculative vision that Nikhil and I imagined, one where we could capture moments in time with the aid of microbiomes and bring those flavors into our kitchens. Today, we want to share with you a little bit about our inspiration and the science that we're building off of.
So first, let's talk about the science. Our vision involves creating a sense of time and place through flavor, and not just through fermentations of food, like what has been done already at the NOMA Fermentation Lab, among others, but through a novel approach that builds on some emerging genomics research. We started with a question. How is a food's taste affected by its place? For example, if you tasted enough wine, you know that the same grape made into wine in different regions can taste completely different. Why is that? To begin answering this question, we read the work of Nicholas Bukalic, Susan Ebeler, and other researchers who have investigated the role of microorganisms in wine terroir. Terroir is the complex flavor that encapsulates so much information about that wine in the glass. Where it's from, what climate was like, when it was made, the soil conditions, and more. All of this information comes together in the subtle nuances of flavor that could allow you to tell the difference between two bottles made from the same grape. Could it be that the specific microorganisms present in fermentation are a factor in the development of terroir? Bukalic and team conducted some experiments to explore that question in particular. First, in their experiment, they took samples of pre-fermentation grape must, basically the grape juice, before it becomes wine, and they extracted the DNA of microorganisms present and then sequenced their genomes to get a list of all of the species present in that grape must and their relative abundance. They did this across California for multiple types of wine in different regions, and after analysis, they found strong correlations between the microbiomes in these samples and their corresponding geolocation, climate, and time of production. So in other words, if you were only given this list of species present and their abundances, you might be able to pinpoint exactly where that wine was made, from which grape, and at what time of year. Kind of like how a trained wine taster can do exactly the same just from the taste.
So we had two questions in response to this research. First, could we identify microbial terroir in other types of foods? And two, could we understand how microorganisms contributed to specific flavors? To answer these questions, we designed an experiment to explore whether we could identify a terroir in homemade yogurts that we made in our own apartments. But before we could conduct our research, COVID hit. So instead of investigating the connection between the microorganisms in our own yogurts and their unique flavors, we reviewed literature to learn more about the microbial basis of flavor. We studied research about metabolic pathways, the chain of processes in which microorganisms interact with their environments to produce molecules that we ultimately perceive as flavor. For example, we see tasting notes for wine that say Meyer lemon, wet pebble, and unsweetened tangerine drop candy. We know that these flavors were not added to the wine. Instead, the molecules produced in these metabolic pathways might have evoked those scents and memories for the taster. Given the opportunity to carry out our own research, we would cross-reference the microorganisms in our own yogurt with known metabolic pathways to specify why my yogurt might have tasted totally different from Nikhil's. But we're pocketing that for future projects when the lab is open again.
If we had been able to do those experiments, we would have shown more evidence for a microbial basis of terroir. And we would have shown how specific configurations of microorganisms in our yogurts could lead to subtle different flavors, depending on where they were made. So we started working from this assumption, and we began imagining what this might have looked like in the kitchen, assuming our research had worked.
Yeah, and that's where our idea for microbial flavoring or a microbial spice rack came in. We imagined taking the microbial terroir genome sequences that we compiled from samples from different times and places and creating flavors from them. We could imagine having a bottle of liquid with a dropper, kind of like bitters, that could be used to kickstart a fermentation or even be dropped on a finished product for an extra touch. Really, we would expect cooks to use their creativity to find new uses for these flavorings, like any other ingredient. Nikhil, you used to be a chef. What time and place would you make into a flavor if you could?
It would be really interesting to add, this whole new set of options into the kitchen. But right now, the main thing that's on my mind is the racial injustices that we're continuing to see in our country. And as I think about the violence inflicted upon the Black American community here, I think about its roots in European colonialization. And it might not feel this way sometimes, but we're only a generation removed from the colonial era around the world. My grandmother, who's alive, was a child during the British occupation of India not that long ago. And India wasn't even the last country to gain independence. So when I think about what time and place I would want to capture and use in the kitchen, I think about a time before colonial rule. I think about finding ways to capture the flavor of that period. I think about sampling DNA inside of an old fermentation vessel used in West Africa prior to colonization and creating a sort of like flavor of freedom. I don't know what it would taste like, right? We don't know what bacteria would be in that sample, but knowing that those non-colonized species that we found in the sample would produce the flavors in our bottle, that would be enough for me. That surprise would be interesting. Every time we eat food, we're eating our history, and I think it would be so empowering to create an ingredient that allowed us to imagine recipes for an alternate world without the legacy of colonial rule.
I think that's super interesting, and I really would want to taste that and see what that alternate reality would taste like. And I think it's interesting that you mentioned colonization for another reason too. As you know, my research has focused on cultivating microorganisms for space exploration and settlement. And the language we use to describe space exploration often uses the word colonization, like space colonization or space colonies, etc. And those words are so loaded, especially given the sovereign nations and highly capitalized private companies that are the two main forces backing missions. And these are the same two forces that backed colonization in the last millennium. But those words are also used to describe microbiomes, right? Microbial colonies can be diverse, with many species of bacteria, fungi, protozoa living in the same environment. There could be power struggles just like ours, but there can be harmony as well. I don't want to be reductive and overly romantic here, but my broader thought is that we can travel space. As we travel space, we can take inspiration from microbial colonies to study how we can create a manifesto based on diversity and symbiosis from the outset without repeating the same mistakes we've made on Earth. Maybe we could create a diverse library of microbial cultures made with samples taken from people, foods, places all over the world. In other words, creating a microbial archive of human cultures and history on Earth that we can take with us as we explore the cosmos. Other artists have created similar works related to microbes and memories. Cecil Toles, for example, collects smells from interesting places in a city and is able to recreate a city's essence through smellscapes. Also, Juan Wu's project of collecting hand biomes to use in fermentation could be another inspiration. Maybe we could collect these microbial samples and create a microbial broth, to borrow a term from Alison Cutland, that could remind astronauts of home.
Yeah, I think all of these would be really interesting, right? Creating new flavors, creating a way of archiving memories and creating a new future as we settle space. And I guess as we reach into the past and project into the future, one thing seems pretty clear. We need a diverse set of voices to help us imagine a better society, build on curiosity, empathy, and respect for life in all forms, macro and micro, right?
Yep.
Neither of us are scientists by training, but we're committed to pushing the frontiers of biodesign, and we want to hear from many more voices that are often left out of these conversations. So before we sign off, we want to highlight a couple of organizations that are doing work to expand opportunities in STEM. We're going to put these up on our slide, so please go to these links and consider supporting these organizations. Thank you so much for listening, and we're excited to answer some questions.
Thank you, everyone.
Well, welcome back, everyone. We're here with Sarah and Nikhil. We have John here from Sandbox. And let's kick off the questions. I'm going to give it a start. So once you can get back to the lab, what would be the first experiment that you would do?
We might have different answers. I'll just say I want to test out the yogurt experiment that we alluded to be able to really like add to the body of research that's establishing the microbial basis of terroir, and do the work to really understand how microbial terroir actually influences flavor.
Yeah, I agree with Nikhil. I think performing that research would be essential to our project. So that would be the first step.
John, do you want to follow up?
Yeah, totally. First of all, hi, everyone. I'm John.
Hi.
Hi. First, thank you for playing Animal Collective at the top of your video. It took me back to my own college days, for sure. That song was a jam. I'm interested in the existing field of research around terroir. And are there scientists with whom you've collaborated with, or whom you aspire to collaborate with in this field, which is something that I've always viewed as sort of something you hear about at restaurants, Michelin star restaurants, and less in a lab setting. So could you talk a little bit more about that?
Yeah, sure. The figure Nicholas Bukalich, who we mentioned, is in many research papers that we surveyed. The paper that we drew from the most established strong correlations between basically the microbial presence in pre-fermentation grape must, or like basically grape juice, and all of these different determinants of terroir that you could talk to a winemaker and they'd say are what lead to terroir. So things like soil makeup and location, etc. We haven't collaborated with him, but it would be awesome to. Sarah just graduated. I still have another year left, and I'm going to be doing a thesis next year, and I if I had to make a bet, it's going to be related to this project. So hopefully I'll have a chance to reach out to him and his team. On a less, I guess, academic note, there's the NOMA fermentation lab has done a lot of really great work establishing both, some research that I think is both scientifically rigorous, but also useful for home cooks. And David Zilber was the head of the fermentation lab until last week. He's a fascinating person. I don't know him, but I've messaged a little bit on social media, and hopefully, he's somebody I would love to be able to work with as well.
Have you thought about authenticity of these flavors? So, you're trying to recapture flavors that are long gone. How do you know that you've actually captured something that's real, or you've pulled a note from something that's new?
Sorry, do you want to talk about Cecil Tolas's way of approaching this?
Yeah, I was going to mention that. So when Cecil Tolas recreates smells, especially smells from the past, or smells from a species that no longer exists, for example, she acknowledges the fact that these smells are abstract, and as much information as we can gather from the species genome and the molecules that could produce certain smells, we can never be 100% certain that this smell is 100% this replica of the original. So we have the same methodology, and we know that, especially when we're trying to capture the memories of the past from years ago, there's no way that we can recreate that with 100% certainty, but it's going to be more abstract and a sentimental gesture. So do you want to add to that, Nikhil?
Yeah, I agree. I think, if done done tastefully, exposing the methodology or making sure there's some transparency about the process will give people confidence that, this is a valid approach to designing a flavor that none of us really know.
And I also wanted to mention that, so when we use these microbial flavorings, there's no specific flavor to the culture itself, because as you add the culture to different substrates, different foods, it's going to interact differently depending on the content of the substrate. And it's going to produce different flavors depending on so many different factors, like the substrates, the temperature, the duration that you let the food ferment for. So I think that's an interesting part of the experiment, to see how many different flavor profiles we can actually create with a certain culture. And what we provide would be more of a symbolic flavoring, I would say. Like we have an example for let's say Moon 1969, like moon landing. And again, there's no like certain flavor for that, but as you add that flavoring to different substrate, it would be interesting to find out what kind of flavors could be resulted from that specific culture.
I'm sure this is a big question, and I know we don't have too much time, but can you talk a little bit about ways that you've imagined the public will interface with this product? Yeah, maybe I'll leave it there.
So
Absolutely.
I think there are two ideas that we were exploring. One is this microbial flavoring product, right? And we mocked up a website that I believe you all have, where we imagine having kits that capture certain times and places, and then eventually the ability for people to send in samples and design their own bespoke flavorings from the samples they send in. And then the second idea is something that would allow any people anywhere in the world to send in a sample of fermented products. So basically, we were imagining yogurt as this kind of cohesive thread. We'd send a mother yogurt that's standard, allow people to make their own and send in a sample, and then we would sequence the genome, capture this idea of a microbial terroir in their yogurt, and then create an archive of these microbial terroirs around the world, and then start to do some interesting analyses of what you can glean from that information and what you can learn about trends that occur in different parts of the world or unique to very specific regions, and hopefully in the process build empathy and see the sort of commonalities across the world, and also our unique identities reflected through microbiomes.
Great. Thank you, Nikhil. Thank you, Sarah. That's all we have time for. Fantastic presentation. We're going to move on now to the next NYU IDM team. This is It's Biolab. Let's roll it. Thank you.
In Mayan cultures, soil is treated like gold. Traditional farmers today of Mayan region believe that soil conveys eats, a holy substance that gives life to plants. Soil biota is a matrix that all life depends on, yet oftentimes viewed as nothing more than dirt. 1 billion acres of farmland have been abandoned due to land degradation. But why have we praised built environments? How can we reconnect with our soils through building practice? How can we communicate eats in our soils, like the Mayans, to illuminate its importance in intensifying our sensory, cultural, and social experiences? Rammed earth, a widely considered natural construction technique, utilizes compressed soil, gravel, and clay as building material. This technique has been passed down for centuries, but is not considered a durable building technique because it is vulnerable to earthquakes. How could we naturally build strong rammed earth structures that could rival cement? Introducing Eats Lab, an at-home biobuilding kit to stimulate soil importance. The kit comes with a special bacteria called S. uriae that once grown can solidify the natural aggregates, soil, clay, and sand through a process called microbially induced calcite precipitation. This bacteria deposits calcite crystals in between the cracks and voids and morphs into stronger and durable sustainable building material. This kit comes with everything you need, from nutrient broth, agar plates, solution, and an instruction manual to start growing your bacteria and applying it into rammed earth building practice. The only extra thing you'll need is a cooler and a lizard heater, which will act as your incubator, a plastic bin with 2 holes cut into it, which will act as your sterile environment, and locally sourced sand, soil, and gravel. Once you grow your bacteria, you can apply it to sand, dirt, and see microbially induced calcic precipitation take place. Building out of readily available materials like soil, clay, and gravel transforms soil into a meaningful cultural soilscape that can allow for a re-engagement with our heritage and deepen our socio-ecological connection. So how does EAT's Biolab work? How can you start to become a microbially induced rammed earth builder and soil steward from the comfort of your home? EAT's Lab is a simple step-by-step process that can get anyone excited about the possibilities of science. First, you're going to want to locate your bacteria, Escherichia, and nutrient agar plates within the kit. Next, you're going to want to go to your plastic bin sterile space and work inside of it. You're going to want to take a bacterial swab and gently place it on the agar plate. Wherever you swab is where the bacteria is going to grow. Tightly close bacteria and store it at room temperature for later use. Flip the agar plate backwards to avoid condensation and any potential contamination. Once you're finished, you're going to go to your DIY incubator and place the bacteria at 23 to 25 degrees Celsius to accelerate growth. Leave it in there for 2 to 3 days. Now that your bacteria is incubating, you're going to want to prepare your cementation solution. You're going to want to take the baggies of pre-measured calcium chloride, ammonium chloride, urea, and baking soda and combine them thoroughly. The urea and calcium chloride in particular will allow for the bacteria to bind between the aggregates of sand and soil. After 2 to 3 days, your bacteria should be ready to be taken out of the incubator, and then you can identify growth of the bacteria. You're going to want to gently swab the bacterial colonies present on the agar plate and place them in the nutrient broth provided. Make sure you mix it into the broth vigorously to make sure all the bacteria gets off the swabber. After, you're going to want to incubate the nutrient broth with the cementation solution together for 1 day at 23 to 25 degrees Celsius. Now, your bacteria is ready to be introduced into the sand and the soil. You're going to want to make your clay and get your sand. Now you can apply your cementation solution and bacteria to the clay-sand mixture. Now you can start to have fun with it. Unlike most biocements and biobrick-like materials, there is absolutely no cement additives or limestone screenings. The kit comes with a manual to start experimenting by making microbially induced rammed earth clay pots, which are sustainable alternatives to kiln-fired pots, where you can begin to see how MICP materials work. The kit also comes with a manual and demonstrates how MICP can improve granular soils that have been damaged due to wind erosion by strengthening the foundations. EAT's lab has all the tools to run experiments on granular soils and strengthen damaged soils. Microbially induced rammed earth can do multiple things and has multiple applications, which is what makes it so powerful. MICP can increase the strength of foundations in sands and silts. It's cost-effective, non-disruptive alternative to chemical grouting, and environmentally friendly. EAT's lab wants you to take matters into your own hands and start biobuilding. Citizen science is for everyone, and EAT's lab fosters a new kind of soil steward who wants to find biological solutions to environmental problems.
Welcome back, everybody. We're going to get started. Thank you, Anik, for that great presentation. John, why don't you kick us off?
Yeah, so this is super interesting. I wonder if you could talk a bit more about why this is a desirable alternative to existing building materials used, in major construction projects.
Yeah, hi, thank you for having me. So basically this is a different kind of bacteria that's normally used in a lot of the biocements. They use S. pasteur, and I was looking into this bacteria that is already in use, and it was quite expensive. And I wanted to see what was the bacterial strand that had the same properties, but that was a lot more accessible and a lot more affordable for the general public, and that was safe. And so that's why I kind of chose to go with S. uriae. Also, I felt like doing it in a kit format would allow people to kind of get excited about science and microbiology and the different enzymatic functions of these bacteria. Because a lot of the biocements that exist today are already they're fabricated, they're made for you, and that's it. And so there's that detachment where people don't really see what's actually happening. So seeing kind of the opportunities and how can biocement and biobricks be kind of a citizen science tool, similar to the Amino Labs synthetic biology kit that they are now selling, or Ecovative's mycelium building bricks. So kind of that similar format and kit type.
Can you talk a little bit how you came upon soil as your subject?
Yeah, so I've been super interested in soil. I'm very curious about it because it's something that's deeply concerning with the kind of agricultural industry and how we, as populations increase, that there's going to be more demand for food, and our soil kind of microbiomes are being degraded with use of pesticides. And so how do we start to foster soil stewardship through science, and how do we promote regenerative futures through citizen science? And so kind of using education and biobuilding as a way for people to see, similar to Legos, kind of like a physical product where they can build it and understand the importance of, microbiology and within soil microbiomes. So that was kind of the inspiration. Yeah.
Yeah, I'm super curious about this from a citizen science perspective, and I wonder what you imagine is the hook of this product. Considering that citizen science is sort of an increasingly crowded field. What are some if you were to tell someone what to do once they got this kit, maybe you can expand upon that.
Yeah, I think that kind of architecture and biobuilding, that's kind of the one area of citizen science it's lacking. I feel like a lot of people are focused on CRISPR and synthetic biology and bioluminescence, and I find that fascinating as well. But kind of seeing how it's so multifaceted and there's so many different opportunities within microbiology, and because they perform so many different functions, and just kind of expanding upon that is what's exciting. And, this is something that's also super cheap. It's everything I used for the kit was locally sourced. And, it was using plastic bins and, a cooler, and very DIY. So it's something that can be used throughout the world for a lot of different communities, which is also very important.
What was your experimentation like? Were you working at home? Were you back at the lab? What did the process look like for you?
Yeah, so originally I was going to do this at the NYU lab, and unfortunately that didn't happen because of COVID, but kind of presented a new opportunity. I was determined to see how I could build a biolab out of my house. So I just started researching on YouTube how to build these DIY centrifuge and a cooler, and seeing how I can turn that into an incubator, and looking at flow hoods and kind of these at-home DIY biolab kind of ways to build things. And with the help of my professor, she was really helpful in kind of guiding me in how I can build these out of my home. And then it was a lot of experimentation. There were things that did not work and that, blew up. But that was kind of the fun of it, was experimenting and seeing doing multiple trials, what was the best experiment to go by. because it was not like a one experiment that I followed that worked. It was like I had to go through a series of experiments that were easiest to do out of the home, and then trying to kind of facilitate my own recipe pretty much for how to biobuild using SCRA.
Thank you so much, Anik. That's all we have time for. Thank you for joining us. Fantastic project. John, thank you so much for joining us on stage, if you can call this a stage.
Some form.
and we'll be talking soon. Now we're going to move on to University of Aalto. Let's see the video.
Around 70% of Finland is covered by forests, making it one of the world's most forested countries. The forest industry here has thrived for centuries, and the proximity of nature to everyday life has created a deep cultural bond. Forests have helped people survive and evolve throughout time, providing countless resources at both the macro and micro scale. Finland's forests are not only cherished economically and recreationally, but also for research and education. Recent studies have shown biotechnological applications within the forest sector are a high priority in academia and industry alike, especially the utilization of side streams for bioenergy, biochemicals, and other food or pharmaceutical applications. Pine, spruce, and birch are the three major tree species found in Finnish forests, and the spruce tree has some particularly compelling properties. Similar to any tree, the spruce tree has layers of protection in the form of bark. Bark shields the inner systems of the tree against disease, animals, and insects with various chemical compounds. The type and amount of compounds are unique to each species. What makes spruce bark exceptional is its concentration of phenolic compounds within the inner bark portion of the tree. These compounds can be easily extracted and provide UV protective and antibacterial properties. The extract contains natural stilbens such as resveratrol. These compounds are strong blockers of UV radiation in the range of 200 to 350 nanometers, absorbing a large amount of the wavelengths most harmful to humans. Studies have shown these compounds to be comparable to commercial sunscreens in their UV absorptive capacity. The antibacterial properties result from a combination of several compounds, including stilbens and various resin acids. While no single compound explains the effect, the extract shows strong antimicrobial activity against a broad spectrum of bacteria. Currently, industrial wood processing involves removing bark and burning it for energy. With such valuable compounds going to waste, our team explored how to utilize this side stream in a more effective way. We asked ourselves, what if we could take the protective coating for trees and turn it into a protective coating for people? Spark Fabric utilizes microcapsule technology to embed spruce inner bark extract into man-made cellulose fibers, producing a protective fabric that is versatile, organic, and renewable. This embedding of spruce inner bark extract enables the creation of lightweight and breathable UV protective clothing for warm climates, shielding its user from harmful rays as well as preserving vibrant colors and fiber integrity. The fabric's antibacterial properties minimize body odor and allow for longer periods of use between wash cycles. These fabrics help resist pathogens, providing a more hygienic garment and responding to a growing desire for personal protection in public environments. Because bark is removed and burned as part of current industrial processing, the sourcing of spruce inner bark fits nicely into the existing system. The bark is collected from trees prior to storage in order to preserve the active compounds. Ethanol is added to the bark, mixing with its existing water content. The mixture is then heated and pressed to remove the liquid, leaving the entirety of the bark solids to be used for energy production. The ethanol is then recovered and recycled. The extract is then microencapsulated using the simple coacervation technique, with starch as the wall polymer. These microcapsules are attached to the fabric through a bath containing a binding agent and a catalyst. The walls of the microcapsules break down based on acidity, heat, moisture, and other factors, enabling a slow release of the extract over time. In this rapidly changing, highly populated world, consideration of a product's life cycle from beginning to end is vitally important. Spark fabrics are designed using renewable resources and industrial side streams. The extract is 100% natural, and attaching the microcapsules to man-made cellulose fibers creates a biodegradable fabric that is made entirely from trees. The forest is precious. In our product, it provides inspiration, chemical compounds, and composition of the fabric itself. Our mission is to gather and utilize these resources in a way that protects the user as well as the environment it is sourced from, ensuring the health and longevity of these forests. For some extra protection, Spark, Shield of Spruce.
And welcome back. We're very lucky to have Aalto University with us. Thank you, students. Great presentation, great video. We have a number of questions from our judges already, so let's get started. Elaine, why don't you kick us off? Don't forget to turn on your audio.
Am I here? I can't tell if I'm here. Okay, great. Hi, everyone. That was such a beautiful video. I really was entranced by your dancer and like the light, and thanks for transporting me to Finland. I wanted to ask if the fabric that we're looking at in the video, is this something that you've already experimented with? And I'm curious about like the washes and the life cycle, because you did talk about that in your video. So like how much does it actually bond, and in terms of how many washes? I know that for myself in production, whenever I'm trying to buy a fabric, whether or not something lasts like 5 to 10 washes, or if you can put basically those properties back into the fabric.
Yeah, I can answer that. So the fabric that you saw in the video is not yet something that you can buy, or it's not that far. So what we've been doing is a lot of scientific paper and article research, and we use that as a background for the whole concept. So we consider this more like as a speculative design project, although we do have the evidence how we could possibly make this fabric.
Great, thank you.
And to add to that, the wash cycles we used for the microencapsulation, a paper of aloe vera added, microcapsules of aloe vera added to stockings, and that lasted for a few wash cycles. But our hope is that it would last a similar amount to other UV protective fabrics. So maybe like 10 to 20 wash cycles, but we would have to do testing for that, and we haven't been able to do that testing in the lab.
We did start the project though, so we did source the extract, and then we did we started with the extraction process, but then the labs were closed down, so we couldn't continue with our facilities.
Got it. Allison, do you want to follow up?
I had a similar question. Great presentation. Thank you so much. And also about the durability of the treatment. And I was wondering, since it's available for a limited amount of wash cycles, if you had envisioned this potentially as a treatment that people could do at home to treat their own clothing, and then have the properties of the bark if they're using a cellulose-based fiber on an existing garment.
Yeah, we did talk several times about the possibility of making a spray that could be administered at home, or even coming up with something that people could extract the compounds themselves, because it is not actually that difficult of a process to gather the bark. But in the end, we wanted to make it more of a cohesive project or product. And the microencapsulation was a really interesting way of trying to make the compounds last longer, because we're not sure how long they would actually stay functional as just a spray. They would need to possibly be protected for a slower release.
Fantastic. Christine, do you want to follow up with a question?
Yes, hi everybody. Thanks for the great presentation. So from what I gather so far, you guys have done some research on this, some sort of desk research, and you weren't able to go into the lab. But I'm wondering, did you talk with anyone in the industry, like who's producing cellulosics or suppliers, to bounce this idea off of them and how they would apply it, or what their thoughts would be about the process?
Yeah, actually, we have had some interviews together with the professionals from the industry or people who have been researching this. For example, Maria Rissanen. So she was also helping us as a mentor, but of course our instructors, Pirjo Kääriäinen and Tapani Vuorinen and Iina Solola, they were helping us through the whole process. So they have the from the chemistry side, they have better understanding.
And we'll end with one last question from Harris.
Yeah, that was a really exciting concept. I guess I had two short questions. One is, what is the scalability of this? How many trees is necessary to make a garment, for example, or how many garments can be made from one tree? And also, what is the smell like? What are the smells and kind of the aromas that are coming from this? Does the fabric smell like spruce, I guess?
I can answer to half of that at least. So from inner bark of a spruce, 10% of that can be steel pens. So steel pens do have those properties what we are looking for. And when there's one thesis done around the topic that was looking into UV properties, for example, of the steel pens. And there it was said that less than 5% of the extract is actually needed to produce a similar protection than 30 to 50 SPF of a regular existing sun lotion. So actually you need to mild down the extraction quite a lot. We didn't do that precise calculation necessarily, but there is actually relatively much of steel pens in spruce inner bark.
And in terms of the scalability, our thought is this would be done entirely with side streams from the forest industry. So no trees would be cut down for this specific purpose to make the extract. I'm not sure about the man-made cellulose fibers, but in terms of the extract itself, as long as there's bark to be utilized as a side stream, that's what can be made.
Great, thank you, Aalto University. Next up, we have NYU ITP. Thanks, everyone.
My name is Julia Lizardo, and I'm a graduate student at New York University at the ITP program. And today I'm going to talk to you about amber. Amber is a plant-based waterproof biomaterial made out of rice husks and pine resin. We convert food waste into usable product. Where does the idea of amber come from? Well, in a comprehensive study published in 2018, the United States had the highest amount of food waste worldwide in 2017, reporting a staggering 200,108.3 million pounds of food waste. If we break this number down, this means each person in the United States produced 950 pounds of food waste in 2017. And looking at the stages of the food supply chain, the stage that produces the highest amount of food waste is at the consumer stage at 414 pounds. So if we take a closer look at the consumer population within the United States, the two most commonly wasted food types in the US is fruits and vegetables and dairy. Analyzing this with the lens of ethnicity, we see beige represents Black or African American, dark green represents Hispanic American and Latino, and orange represents white. In both food types, Hispanic and Latino communities had the highest percentages of food waste. Why? Well, there's many different factors at play here, such as accessibility to compost initiatives, space limitations, and financial resources. So at Amber, our objective, our mission, is to create all natural products with rice husks and pine resin to repurpose agricultural waste and give agency to Hispanic and Latino community members to help eliminate food waste in their daily practices. Amber is made of two main ingredients, rice husks and pine resin. Rice husks are the individual shells or protective covering around each grain of rice that needs to be removed during the milling process. We use rice because it's cheap and abundant, it's an agricultural waste, can be grinded into a fine powder, and is currently used as fertilizer, building material, and even fuel. It's also culturally significant in Hispanic and Latino cultures. I am Dominican and my family cooks rice up to 3 or 4 times a week, and we usually pair it with beans to make a dish called moro, or with soup in a dish called sancocho, which is our version of chicken/beef stew. 100% recommend, definitely try it out if you can. Our second ingredient that we use in Amber is pine resin. So pine resin is solidified pine sap used by trees to heal their wounds. As a result, it's antiseptic and anti-inflammatory. It's durable, water insoluble, oil insoluble, and its consistency at high temperatures is that of molasses, while at low temperatures it resembles more like glass. And due to its sensitivity to heat, the surface of resin remains sticky. While Amber consists of two main ingredients, we're not the first brands to use rice husks as a building material. There are two main brands similar to Amber. EcoSoul Life, which provides reusable dinnerware made out of rice husks and natural wax, and it's biodegradable after 2 to 3 years, as well as dishwasher and microwave safe. EcoForms is the second brand, which offers reusable flower pots made out of rice husks and natural binding agents, and it has a lifespan of 5 years outdoors and 10 years indoors. While both of these brands aim to create products out of material that resembles plastic as the feel and aesthetic, what makes Amber different is that we're a borrowed material aiming to resemble glass and create a personal connection between the products we offer and the people who use them. In order to make Amber, we follow 4 simple steps. We first melt pine resin using a double boiler until it liquefies. We blend rice husks with color dye until it forms into a paste. Amber comes in 9 plant-derived colors. To make our colors, we use spices, fruits, and vegetables such as achiote, saffron, carrots, and berries to create natural pigments. Next, we mix the colored rice paste and melted pine resin together until the mixture becomes uniform. We then use silicone molds or hand-shape the mixture into the desired shape to create beautiful, ready-to-use products. Amber has the potential for many different use cases, such as storage, dinnerware, jewelry, and art. So far, we've been able to successfully make containers, dinnerware, pendants, paperweights, and earrings. Our next steps include testing additional parameters. We want to explore additional natural flame retardants and sealants in order to minimize pine resin's sensitivity to heat. We want to use industrial power tools to aid in moldability, since pine resin hardens very quickly once exposed to cold air. We want to test how food safe the material is. Although all the materials used here are natural and plant derived, pine resin is a natural source of turpentine, so we want to double check the sources that we are purchasing our raw materials from. We also want to test the biodegradability time period of our products, as well as expand to using not only rice, but plantains, another ingredient prominent in Hispanic and Latino cultures, to make additional products. What this means is that amber, in its short-term results, can reduce the consumption of plastic by providing consumers with a sustainable alternative, as well as convert food waste into an impactful, ready-to-use product. In the long term, amber can lower the plastic resin production in the United States from the 2018 total of 119,559 million pounds a year, as well as national prevention of food waste can prevent up to 3.66 metric tons of CO2 emissions per short ton of food to be released into the air annually. As you can see, amber has potential for many different applications. With continuous research and iteration of the manufacturing process of this biomaterial, amber can succeed in its mission to reduce food waste. Thank you so much for your time. Have a wonderful day.
Welcome back, everyone. We have Julie with us from NYU ITP. Thanks for joining us. And the judges have some questions. Why don't we start with Angela?
Hi, thanks for your presentation. I was just wondering, is pine resin food safe if you're making dinnerware with this?
Yes, it is food safe. I'm right now, because of the quarantine, I'm just buying bulk pine resin from Amazon that sells food grade pine resin. But again, as with any company, you want to make sure all your sources are reliable and double check that to make sure there's no liability with that. Because again, it does have natural sources of turpentine.
Questions from our other judges? I have a question. Can you talk a little bit about how you came to this project in the first place? What was the origin of it?
I am Dominican, so this is a topic very personal to me. And I come from a biology background, and I've always been interested in sustainability. And one thing I've noticed even within my own community is that composting really isn't a thing. It's seen with a lot of skepticism, a lot of hesitation. And again, there's many factors to that, preconceived ideas. And my amber came about from this notion of what can I do to help destigmatize composting or reducing food waste. And so I looked around my house and what are ingredients that I saw some ingredients that could potentially form into a building material. And I immediately thought of rice, which I have seen being used in many different forms, including to disposable plates and other bowls. So I immediately was drawn to rice. And I started looking at natural adhesives and came across pine resin and its cool properties of looking like glass. So,
Great. Thank you. Denise, please.
In your test cases, I was a little confused. First of all, I'm interested in this idea of the transition from composting and making it desirable. But I couldn't tell if the rice you were using was cooked or actually uncooked rice for your products.
So it's not actually the rice grain itself, it's the protective covering around the rice. So it's usually removed in the milling process when you harvest rice.
So it's not actually part of a domestic compost project. It's actually an industrial compost project.
Right. Yes. When I say compost, I mean that this product is meant to start conversation and get our users, which are the Hispanic and Latino communities, to be more curious about food waste. Right. And we're using industrial waste, but again, the bigger picture is to inspire and promote different initiatives, which include composting at home. But this is just a starting point to bring this curiosity, like, wow, these products can be made out of something that is usually thrown out. Yes.
Elaine.
Hi. Okay, I think I'm on. I wanted to ask about about all your tests that you did. I think it was really great that you showed us such like a wide range of experiments that you had done. And in particular, though, I was wondering about how you were talking about you had like bowls and spoons and that, and I was wondering about the flavor. Like, is there any sort of flavor that comes out, or a flavor or like aroma? I'm imagining like having a tea and then being able to have like this additional taste. And then I was also wondering, like, maybe some conversations that you would had with people in your community or your family and friends of like how effective they thought your choices were.
Mm-hmm. Yeah. So in terms of the aroma, it does smell like pine. So pine resin, you can really smell the pine tree in it. But in terms of taste, it doesn't really taste like anything. And in terms of texture, it's just very smooth. Again, feeling that texture of like glass. And in terms of my conversations with people who have seen the product, well, that is a little bit limited to my classmates who saw it before quarantine and my family. But even within my own family, it has brought up conversations on how we use rice and how we can potentially bring more initiatives into our home to help minimize our food waste, which could mean different ways of recycling or purchasing less food. But it's definitely brought conversations about the possibilities of food that are not just limited to being ingested.
Have you gotten any like additional ideas in the time that you've been focused on your project? Like, do you have like a version 2 already in mind?
I definitely want to go bigger in the terms of the products that I'm making, because right now I'm hand molding them. So they're limited to be quite small, so they fit on the palm of my hand. But I definitely want to be able to make like a dinner plate, which can be quite bigger, or a vase, or like bigger products with more materials and more time.
Nice. Really great.
Thank you so much, Elaine.
Thanks.
Now we'll follow up with Jason, and then Angela with the last question.
Hey there. Great job. I was just wondering if you had thought about the scale of how to scale this and availability of product. So I imagine that rice, sorry, what's the rice covering called?
Rice husks? Yeah.
Excuse me. Good morning. It's really early in the West.
It's okay.
Is much more available than pine resin. So I was kind of wondering if you thought about, like, if this was to really go to scale to try to replace plastic, how would, did you look into the amount of pine resin that might be needed or is actually available out in the world? So
yeah, that's a good question. Pine resin is definitely the limiting agent. I haven't quite looked at what exactly that means in terms of how many trees per amount of pine resin that is, but that's something I definitely will explore moving forward. But in terms of reducing the amount of pine resin that it's needed. Pine resin tends to stick to everything in the manufacturing process. So really perfecting and iterating on this process can help minimize the excess that is wasted in the manufacturing. Because for example, if I melt it in a pan, the pan gets coated with it. And if I change colors, then I need to use a different pan, and that pine resin is potentially wasted. So finding ways to really perfect that manufacturing process can help in scalability.
Great, and thanks. And Angela.
Great. So my question is actually kind of addressing scalability as well. I was wondering, it seems like you're hand building all of these objects. So I was wondering if you have experimented or thought about a way to like maybe pour it into a mold or something just to make it easier to produce it on a larger scale.
Yes, so I have used silicone molds, but one of the issues that I've experienced is that pine resin starts to solidify very quickly once it hits cold air. So as I'm pouring it, it already starts looking like glue as it's going down. And so one thing that I'm planning to explore is using different pans and pots, maybe having a heated ladle that I can pour into the silicone mold so that it retains that temperature and viscosity for a longer time period.
Cool. Thank you.
Yeah. Thank
you, Julie. Thanks for a great presentation. Next up, we have HBBE. Well, let's roll the tape.
Welcome to Culina, the world's first gastrolab. We've fused biotechnology, architecture, and design to create a new kitchen concept. Experience it for yourself. Culina goes back to traditional processes of cooking, re-establishing our connection to the food we eat. Here, we air dry food in low humidity and low heat, using natural ventilation through air bricks. In this indoor farm, we grow organic fruit and vegetables. We use hydroponics, a method that uses nutrient solutions instead of soil to drastically reduce water usage and pest problems. These vertical panels are filled with grow stones, which we repurpose from glass waste for superior air and water retention. Irrigation is constant and controlled, ensuring a bountiful harvest all year round. Culina's architecture emphasises how everything is connected. Everything that is generated is conserved and utilised fully, from heat to refrigeration. So you have our oven room to thank, not only for your food, but also for this dining room's cosy temperature. Speaking of which, please take your seat and make yourself comfortable. At Culina, you don't choose a predetermined dish. Instead, you gather your ingredients, starting with your main. The algorithm suggests ideal combinations which suit your developing flavour profile. Here, you have a selection of pickles which complement your choices so far. After you've made your selections, we surprise you with your unique meal. Your dish is Mesosaurus gravlax. Excellent choice. Mesosaurus has been extinct for centuries, but you can enjoy it today thanks to scientific leaps in protein production. No animal will be harmed in the creation of your dish. Would you like to learn about the technologies that makes this possible? Wonderful! This is where all our protein is made. From beloved favourites like chicken, beef and pork, to more exotic options like Mesosaurus, dinosaur meat and dodo poultry. Our chefs are even experimented with yet unnamed meats. It's all possible through Culina Pro Yeast, patented perfusion bioreactor technology that harvests protein from genetically engineered yeast. Select from our range of cell pods, each a customised yeast cell line for production of your meat of choice. You can even design meats thanks to advances in synthetic DNA technology. Here, our yeast cell line is optimised to efficiently secrete recombinant protein via its secretory pathway. Proteins are transported into the surrounding culture medium and selectively filtered into a protein pipeline where they travel to a protein reservoir under high rates of perfusion, Proteins arrive at a second chamber, where larger target proteins are separated by size exclusion chromatography. With the help of gravity and additional vacuum enhancements, the protein is forced down the column, while smaller nutrient molecules are recycled back into the reactor vessel for continuous fermentation. Finally, protein molecules diffuse into a scaffold chamber, where they bind to a macroporous 3D scaffold. The 3D matrix provides a structure alike that of meat tissue, where immobilized proteins begin to collect. And there we have it, clean meat with unrivaled taste and texture. As your chef finishes cooking your dish, let's see where the rest of your ingredients come from. Here in Culina, the chef is not the only one creating your meal. We also make use of tiny energy, working with natural processes that transform, distill, and flavour your food. These processes happen throughout the building, where the requirements of these microbial processes informed the architecture, making it a truly symbiotic environment for both microorganisms and humans. The tropical room obtains its humid environment from a sous vide bath for slow cooking. The humidity provides the perfect conditions for microorganisms like Aspergillus oryzae to multiply to produce a key ingredient, koji. Fermentation mostly takes place in the cellar. Bacteria and yeasts enable us to lacto-ferment fruit and vegetables, create interesting vinegars, and make the many flavours of kombucha we pair with our dishes. The refrigeration room is kept at a steady low temperature by evaporation cooling and natural insulation, so it doesn't use up a lot of energy. Once we've reached the flavour profile we are seeking through our microbial processes, we place the jars and containers in the refrigeration room to halt activity by reducing the microorganisms' energy levels, thereby maintaining the perfect taste until required. We end back in the dining room, which is as simple and natural as the food we serve. Everything comes together on your plate. Your dish is not just a meal, but a medley of tiny energy, biotechnology, and human ingenuity. We hope that touring Culina made the invisible visible, adding richness to your experience and a sense of connection to your food. Speaking of which, your food has arrived. Enjoy your meal.
You may be asking, how did this all begin? Culina evolved when 4 students from completely different backgrounds came together with a shared love for food. I'm Emma, a molecular biologist. I'm fascinated by the vibrant potential of microorganisms for sustainable development. My current focus is on microbial cell technology for clean meat production.
I'm Dawn from South Korea. My background is in user-centered spatial design. I helped Emma design a user-friendly bioreactor as part of a bigger immersive experience.
I'm Pippa, the aspiring architect of the group. I'm working on creating a common language to make the invisible world of microorganisms an accessible design tool. Using tiny energy principles, ecological design can be encouraged to harness the abundant energy embodied in microorganisms.
I'm Adrian, a medical doctor from the Philippines, now studying innovation design. I'm investigating ways to introduce radical ideas through incremental changes by involving people in the iterative process. That got me interested in helping Pippa translate her ideas into an early prototype.
When the unprecedented pandemic, COVID-19, brought the whole world to a standstill, flaws in our food economy began to show, and with it, a newfound urgency for sustainable innovation. These 4 friends saw this as an opportunity. The lockdown didn't stop them from doing what they did best, cooking up innovations. Combining their interests, they came together to create the very first GastroLab picnic. And with that, Culina was born. An innovation using a mixture of ancient techniques and tangible biotechnologies, imagined into one cohesive system. The aim of Culina was to provide a solution with immediate implementation to prevent humans heading into a dystopian future, but an ecological one with strong connections to nature. What set it apart was a deliberate effort to work with nature, not to replace it, and to balance a complex science concept with accessible design. When Culina first opened, there was praise and there was controversy.
GMO or clean meat?
Genetically modified organisms, or GMOs, have long caused controversy, despite evidence suggesting they do not pose any human health risk, but provide nutrient enhancement and economic benefits.
We're not sure about the nutritional benefit. Farmers are losing jobs.
I see it as an opportunity. I earn much more than when I was working the fields.
Vegan is no longer niche. It's a megatrend.
The Culina prototype became a momentous open-source experiment. Lawmakers started considering the GastroLab concept as an answer to food security. Scientists and chefs collaborated in the design of new meats. Culina's website became an open-source cornucopia of recipes, resources, and ingredients, inspired by the founder's cookbook that made sustainable cooking techniques available to everyone. Architects are now exploring how to incorporate the concept into a residential tower block, replacing individual kitchens with a modified GastroLab running through the entire building. The vision is to ultimately integrate food security with modern life in urban cities. From a humble picnic to a GastroLab and finally to the home. Culina is a testament to how biotechnology and design can come together to create ecological solutions for a sustainable future.
Thank you, Culina. What a great video. We have the team here. Judges, do you have some questions? I'll start. I'm curious, what would be your imaginary favorite meal that one of you would make?
It's not a difficult question to answer because we've actually made 11 courses using this principle and techniques that we've established. So the favourite one of mine actually is it's shiitake mushrooms, and you cook them with a lacto-fermented honey glaze, and you also pair it with koji croutons, which is essentially kind of using bacterial spores to ferment pearl barley. And actually it comes together in a really nice dish, and I was very pleasantly surprised.
What were some of the hurdles that you encountered as you started to explore the science behind this technology?
So with the hurdles, I think one of the biggest issues with food, like synthetic protein production, is the idea of it being like a GMO. And a big sort of factor was trying to bridge the sort of social perceptions of a GMO product and what we kind of want to envision, and sort of backing it up. So we went through lots of stages of the science where we added extra layers of sort of purification. So with the first sort of chromatography step and then the second scaffold, and it was like important was to establish like a biosafety concept throughout. Thank you.
Angela, do you want to follow up?
Sure. I was just wondering, what do you feed the cells in the bioreactor with, and is the whole process vegan?
Yeah, so the second question, the whole process would be vegan as compared to sort of, say, like a cellular agriculture method, which uses cells extracted from the animal source. We're completely animal-free, which is important to us. For the first one, so it would be a nutrient media inside the bioreactor vessel, which keeps the yeast cells growing and thriving. And we're going to use a continuous fermentation process so that nutrients is recycled, which doesn't end up in the final protein product. And then sort of like a future outlook is that we want to somehow incorporate other rooms of Culina into like a cyclic process into the bioreactor. So whether that's sort of processing biomass that the yeast can feed upon, or like vice versa, that some of the waste from the bioreactor cycle is transferred to sort of feed plants, etc.
Cool. So the nutrient media doesn't have any kind of animal products in it at all?
No, it's just based on like sugars.
Okay.
Yeah.
Thank you.
You're welcome.
Christine, do you want to follow up?
Yeah, great presentation. Well done with the video and very well thought through. I'm wondering, it seems like the Kalina concept is one where it's very symbiotic. It's a really holistic concept. Is there one piece that you think would be harder to scale or harder to make a reality than others that could be a blocking point as you try to develop out this concept?
We may have different opinions. I'll just start with mine. It's an interesting question. Thank you. So I think for me, the bioreactor is something which is kind of hard to sort of like scale up and sort of be truly symbiotic, say if it was in a residential situation. But sort of one thought is that whether it could be like downscaled, sort of like a kitchen top bioreactor, so like for the individual household, or in sort of an upscaling process in the future, that kind of creates a specialist job within the community, that someone actually comes and engages through the Kalina prototype years ago and understands the whole process, and then they understand and they become a specialist themselves, and it creates new jobs and a sense of connection with this kind of alien process, which may have been considered alien to others. So yeah, there may be other responses.
For me, scaling is an issue from the architectural perspective in terms of engaging people with the biotechnology and looking at things from a microbial perspective. So I think that the issue in terms of scaling it in an architectural sense is engaging designers to think from a different perspective to what they're used to. And so that's part of the reason why we developed this concept of tiny energy, to try and create a microbial tool set that allows people to engage with it and understand the processes, for them to then adapt those to their own design needs. So I think that would be something that would need a lot of work and conversation.
Yeah, and to add to Pippa's, scaling up technologically is one aspect, but the other side is getting widespread adoption, like a more perception type of scale up. And that was why our whole design approach was with incremental changes first. Kalina is like a prototype, a place to engage with people, to see what their reactions might be, what needs tweaking, so that they themselves will try and adopt this, because invention without adoption is not innovation.
Denise.
First of all, I just want to say that I thought your sort of collaborative effort across all of the disciplines was really fascinating. And your opening image of feeding us this defunct species also sort of captured my imagination. But I'm wondering, how you thought about the implications of tempting us with extinct species meat and in a vegan context. And does that in any way incentivize people to go out and actually, hunt more exotic species? Like, how do you see the project relating to, wilderness and other, non-human biotic species in the grand scheme of things?
That's like a really complex question, which I have been thinking about. So I guess it kind of, if I'm interpreting your question right, is that we kind of want to give a sort of consumer more sort of ownership over kind of what they're designing. So we give them the tools and like a toolkit, then they can kind of design their meal and sort of be with the whole cultivation process. And then sort of this idea of kind of going out into sort of away from sort of like a plant-based product to something exotic and something that you've never heard of before can create something more exciting. And in terms of like a vegan society, I guess vegan wouldn't be the target anymore. It would be just sort of ethical consumerism and something that, it's just it kind of a broader target market away from sort of the vegan that may not even want to eat meat still. I don't know if that answers your question properly, however.
Yeah, but to add to that, we had a very interesting conversation with the group about how something like this could open up an entirely different space, so that it's not polarized where you're either vegetarian, vegan, or a meat eater. But like Emma said, we're all ethical consumers, because this space is where everybody can come together. I think a big challenge to get meat eaters to adopt a more vegan mindset is they feel limited by the choices. And this is our way of exploding the choices, exploding the market with new options, get excited about what you can do with this kind of technology.
Thank you. That's an interesting response. I like the idea of opening this new uncertain territory. Cool.
Next up.
So I live in Arizona. It's where the Biosphere 2 is, which you might have heard of, which is an experiment, a closed biological experiment in the 1990s. I was wondering, it had a lot of troubles with its controls. This is more of a question for the architect on the part, especially bacterial controls and food controls when it was also run by a cult, but that was another whole other issue. So I was just wondering if you had looked into that case study at all, and if you had thought about architecturally what kind of controls you'd be putting in place for purifying. I mean, this is going to really need to be something that, especially with visitors coming in from all over the world, that you really think through the kind of controls, the bacterial controls. So I don't know. I don't live that far from that crazy project, so I was sort of wondering if you had thought about that at all.
Yeah, so whilst I haven't looked into that particular case study, it is something that has been brought up and that we have discussed and looked into. So essentially, in terms of kind of microbial containment in regards to the bioreactor, that's the thing that cultivates the kind of the clean meat. The idea of that is that it's a contained unit, so there's not the risk of kind of leakage and contamination through those. And then what the whole point of this project and what makes it, I guess, quite a different perspective in terms of approaching biotechnology is that we're using old traditional techniques for a lot of these processes. So things like fermentation, smoking, curing. And so these are using microorganisms that are just abundant around us. They're not harmful to human beings. And so whilst there would have to be an exploration into, say, the material surfaces of the building, which enable those microbes to still thrive in that setting, but also to allow them to not interfere with each other while they're trying to do their work. It's not so much an issue of, is it safe for humans to come into this space? So that's kind of what we were going for, is actually trying to use traditional techniques that we know they work and they don't pose a risk to humans, and bring those into the 21st century again.
Yeah, I was more asking about cross-contamination of the meat-growing facility, so thank you for answering that.
Elaine, last question.
Cool. Hi, everyone. Super interesting. I love the fact that you really caught my attention and you made me laugh when you put unicorn on the menu. And to go off of, I think it was like the last person who asked, or the one before, asking questions of this idea of like, this is a vegan process and you're talking about designing meats. And I was, this is more, I guess, sort of a question related to perception and your use of language and your storytelling. You used dodo and mammoth, and in your video, you guys are really excited about like designing new meats. And I was wondering, in a question of language, so for example, in India, they say non-veg and veg. And you're talking about meats and like vegetables. Had you guys considered maybe like a new name for something that we're going to consume, instead of like all the baggage and the history, for better or for worse, that vegetables and meat come with?
I think that's a really good point. And it's something that Adrienne spoke about earlier slightly, in that we are wanting to create this new culture where there's an equal platform for consumers. But this is kind of a new topic that we've only just kind of started to dig into. So we've had our own issues with what we call things, and we've had those conversations ourselves, like, oh, by calling it meat, that doesn't quite sound right because it's not meat, but it's not vegetable. And so it's actually just a conversation that we've only just begun. And so we haven't really come to any conclusions with that, but it's definitely something that's like on the cards as we develop this project.
Thank you, Kulina. Great question, Elaine. Thank you, everyone. We're going to follow up next with Parsons School of Design.
To the biodesign community, come with us and imagine an old growth oak forest, one that is complex, biodiverse, and undisturbed by colonial impact. Once upon a time, New York City looked just like this, and was a forest within the Lenape-Hoking region. The forest was lush with life, human and non-human. This is where we are now. And here is what is being dreamt up for our urban environments in the future. Can we honor our past as we build the future? Could we create a forest city? How would such a drastic change start to occur? As a team, we're interested in what happens right after the now. These types of questions fall within the field of transition design, which recognizes the interconnectedness of social, economic, and natural systems to create solutions, transitioning our world into the one we want to see. These are also questions that call for transdisciplinary design, which is to say it involves all of us in each of our fields to imagine alternative futures through design-led research tools and methods to address the most urgent issues and challenges of our time. We are Serial Space. We come from diverse backgrounds in design and policy within the New School here in New York City, and we've come together through Biodesign to create a set of clear guiding principles that allow for more transparent, ethical, and just biodesign practices. We will exemplify these principles with a case study that reimagines urban spaces, starting a social movement to rewild our city. Our name, Serial Space, comes from the idea of a series, which when paired with space speaks to a concept of a space that engages in the evolving story of a community, where space is a medium for a series of ideas, episodes, or events. In these next 10 minutes, we want to show you that we do not have to look to scientific and technological innovations to design the future first. What we need is to restore and reclaim what has been devalued, destroyed, and nearly lost to quote-unquote civilization and progress. And in this process of restoration and reclamation, we must ask ourselves, whose is this to claim? Who needs to be involved? And who should be leading? At this time, systemic change is upon us. Our communities and cities, our common spaces, are in a time of episodic crises, and there are outcries all around us to reimagine and rebuild the world anew. We believe the only way to do this is to ensure our future is designed for the people and by the people. And we believe that the only way to move forward is to understand where we have come from. Through our project, we are calling on all of you to step outside of the lab and academia and onto the streets. We especially challenge practitioners of biodesign to question assumptions about who has authority and who gets to be involved in designing with living matter. We are proposing the following biodesign community and environment principles. When designing, we commit to 1. Restore and preserve communities and their environments through our processes as well as our outcomes. 2. Invest and directly involve the interests and expertise of local communities and their environments through knowledge exchange, co-creation, and ongoing relationship. And 3. Respect the inherent rights of Mother Earth, all life forms, and the indigenous way of life on the land we practice on.
So how would these values and commitments be represented within a project? To illustrate, we present a case study, Rewilding. Rewilding is a large-scale conservation movement. In our urban context, in one of the world's most densely populated cities, rewilding is about bringing life-supporting native plants with all their genetic diversity back into public spaces. Native plants include all the species that were around long before 1609 when Henry Hudson first discovered Lenape territory. These species are actually in decline in New York City, and only 62% of plants currently found in the city are native. A decrease in biodiversity means a decrease in ecosystem resiliency. We must plant native to deliver ecological services such as carbon sequestration, food and shelter for wildlife, and retention and cleaning of stormwater. According to the New York City Parks Department, increasing native biodiversity cannot be accomplished by cramming them into our parks. So we must get creative on where we plant. Could the next phase for a city for the people and by the people look more like this? The first step in a series of interventions involves first personally inviting friends and community residents to come together and grow native plants all around the city. We want to invite you and your community to rewild your neighborhood. This card is an invitation to come to a meetup to grow native plants. The first event could start with planting along a sidewalk or organizing around landscaping in a vacant lot. It can be anywhere and everywhere. The card is also embedded with the plantable seeds of a local native plant that you can plant yourself at home or bring to the meetup. By using this seed paper, we address our first principle through responsible and restorative practices within our process. We designed the postcards so you can also send one half to a friend. We are building a committed social network of mutual friends while also reviving the practice of sending handwritten cards via mail for a digital generation. We also look to existing street interventions as a way to invite anyone from the public and expand the social network, ensuring an investment in the local community, our second principle. This is a street pull tab flyer where you can either scan the QR code that directs to our site or text a number to get in touch for information about the planting meetup. The invitation system works not only by directing people to a website with meetups to take this movement to the streets, but also provides resources on native plants, information on related community organizations, and suggests tangible, everyday actions for people to rewild their homes and communities. The education around native plants upholds our third principle by prioritizing Mother Earth in her indigenous state. Rewilding as a way to design the future requires a vision for communities and by communities. We, as Serial Space, are merely sending the invitation, but it is up to all of us, including you, your friends, and your neighbors, to come together, decide the terms, and write the story. Rewilding, in this way, not only upholds and seeks to restore native life forms, but values the knowledge and experiences of people from all walks of life. We believe that biodesign is often too limited to the realms of academic discipline, and that we need to do better by expanding the invitation list of who gets to participate in shaping the future. We argue that without meeting people where they are at, biodesign will continue to reproduce the exploitative and extractive practices of the current capitalist regime. Without instilling clear ethical principles into our practices, we as biodesigners will rely too heavily on the exact mechanisms that have led us to the Anthropocene, an epoch in which the Earth's systems are literally being altered by human activity. When we make new, we have a responsibility to first recognize and learn from our past, and more fully understand the present. Biodesign can better withstand the challenges of our time under a set of timeless principles, designing with life on the basis of respect, relationship, and restoration. And within each process, cultivation, cooperation, and care. These ideals are embedded deep in our heritage and collective experience within the natural world. Every time we feed another being, learn from another species, plant a seed, or tend to the soil so that life can thrive and reproduce. For centuries and millennia, designing with life has been less about inventing the new through synthetic biology, and more about connections on the ground at eye level, witnessing life and death and life reborn again and again. Thank you, and sincerely signed, Serial Space.
Thank you, Serial Space. That was a gorgeous video. Denise, why don't you start with the questions?
Okay, so I guess I'm going to start by asking if you could sort of clarify. You talk about the importance of rewilding, and yet you're proposing a project that is ultimately cultivation, which is, you know, the underpinning of urbanism, which in most contexts we understand is the opposite of wild. So I'd like to hear how you sort of rationalize that you're creating a higher intensity of cultivation in the city, and yet you're framing it as wild. And connected to that is just the observation that by sustaining humans as the controllers of what gets planted, and you're mandating that it be native plants, which runs into concerns about nationalism, I'm wondering, you know, how you imagine that you're somehow making humans components of nature and not sustaining them as controlling nature. Is the question complicated?
A little bit. Just to clarify maybe our understanding of the question, you're asking how are we mandating this when it requires human cultivation?
How do you understand the act of human cultivation as creating a wild condition?
Yeah, so in our project specifically, we're framing it as native being anything that was existing before 1609, so before colonization in New York City, and before invasive plants took over as becoming more predominant in cultivation. So rewilding under these terms has to do with bringing it back to, or restoring the land back to its original state, being before the land was colonized.
So how do you factor in the essential nutrient inputs, irrigation, watering, everything that would have to go into somehow recapturing an abundance of those species in our current urban condition? Because that would be not natural.
For what we understand, oh, go ahead, Hadley.
I was just going to jump in there, and I know in our research we looked at the New York City Parks and Rec guidelines for native plants, and they do recommend quite a few. I think it's over 40 native plants that do really well in successional old fields and urban lots. And so they don't need too much tending to, because they are so hardy, and they have been directly pointed out by the New York City Parks and Rec Department.
So I would just say, to pursue this project further, I would encourage you to interpret your idea of what's native with more looseness, partly because Parks has been criticized for recommending such a small cohort of plant material, because you then risk having a monoculture. It's not a resilient system. But also because with climate change, what is currently, well, what was native in 1609 is no longer native because our climate's changed since 1609. But certainly for the future, if we were to adhere to only planting things that are, you know, adapted to our current city, we would run the risk of unplanting ourselves for the future. Okay, just a thought to take forward.
Thank you, Denise. Next up, we have Christina.
Hi, thanks so much. I really appreciated what you were saying sort of about biodesign not perpetuating like old extractive practices. And I kind of wanted to jump off from some of the questions Denise was asking and maybe ask you to speculate or share with us a little bit more. Like, it seems like your project actually like kind of opens up a little bit an idea about like what it means for something to be wild, what is natural, how these concepts themselves have come to be colonized. So I don't know, maybe yeah, I didn't hear an answer to Denise's question about sort of like, what does it mean? Like, how is it wild if it is being cultivated? Like, is that like the right kind of dichotomy, or is there actually something else that we could be, that you're pointing to or could be pointed to? Anyway, that's sort of what I'm curious about, which is not quite formulated fully as a question.
I can start with this one. I think there's something really wild in connecting people through, so we're proposing these tactics, and this is our case study. I think we're happy to talk about this case study, but more largely, I think we're pushing the principles and kind of want to drive the conversation that way. But there is something wild in having these tactics of the pull tab flyers and the mail seed paper and connecting people, and we're just starting the conversation and creating social networks. And then it's up to the people in that community to decide, okay, I've learned a little bit about native plants. I've learned a little bit about what used to be here before colonizers. I wonder what we can do as a community. And so there's a wilding aspect in like how we're setting this up as a network or as a system. I don't know if anybody else has something to add there.
Yeah, I think we wanted to, like, part of our research that we did this whole semester was understanding there's already a lot of amazing community and systems out there, and we want to support those people who are doing those things. And a lot of it's really, how do we give agency back to the people, right? And re-educating people about what are native plants actually, are what we're seeing around us surrounding native plants. I don't know. And so really just, I think, prompting people that question so then they can start really reconnecting with the urban surrounding they're in, and then potentially join different organizations or ask more questions and take that agency back to the people.
That's interesting. So I appreciate that it's not, right, like you're not offering a designed like product to solve the problem like of capitalism, right? That and so like I think there's a lot more that you can do to kind of continue to push that idea. Like how do you totally like what do you do to like break that down, right? So this is like one intervention, but it sort of speaks to an opening up of like what biodesign could be in the first place, which is interesting. So thank you.
Thank you, everyone. We're out of time. We're going to take a 15-minute break. Thank you, students. Everyone in the audience, feel free to watch these videos we've compiled for you. They're a mix of advertisements from our sponsors, previous year's videos, as well as professional videos. We'll see you soon. Thanks so much.
Mother Nature gave us something that's richer than our imagination.
We saw a very peculiar pattern.
Never seen anything like this before.
I remember him saying, remember this word, CRISPR.
We've never had the ability to change the
Mother Nature gave us something that's richer than our imagination.
We saw a very peculiar pattern.
Never seen anything like this before.
I remember him saying, remember this word, CRISPR.
We've never had the ability to change the fundamental chemical nature of who we are, and now we do. And what do we do with that?
This doctor told me, just hold on, there's something coming.
You can actually use CRISPR to change DNA. You can actually do it.
We could engineer a single gene that could potentially make us all more muscular. But should we make that universally available?
Что человек может создавать человека, человек, который может воевать без страха и без боли. И вот то, о чем я сейчас сказал, может быть страшнее ядерной бомбы.
Should we really be manipulating the heredity of future generations, given our lack of knowledge about so many things?
I don't know where you draw the line between not having albinism and deciding your kid needs to be an extra foot taller so they can be a good oarsman and go to Yale. Where is that line? Who's going to draw that?
Anything that will stop my child from suffering, I'm for.
You know, draw this ethical line wherever you want, but don't draw it in front of my disease.
What does that mean for this science, where we have the capacity to edit in some things that we think are important? Are we playing God?
You don't realize it's disruptive until you look backward. Often you don't realize that you're in the middle of a revolution until after the revolution has occurred.
Hi, I'm Rowan Willow. I'm the lead scientist here at BioShield. For a while now, we've been studying your tears. It turns out that tears of people with ovulatory cycles contain chemosignals that reduce testosterone and make it harder for those around you to tell that you're ovulating. Testosterone is linked to heightened libido and, in some cases, aggression. We've been hard at work in the lab, crying and collecting data. We've taken this data and we're proud to introduce our new product, the BioShield Tear Collector.
Collected tears, collected voices, collective action.
It's an all-in-one covert collector that allows you to harvest your own chemosignals. When you sense a threat, you can just pop open the ring and let the chemosignals do the talking. This product can be used to lower occurrences of microaggressions and work as a biochemical shield against unwanted sexual attention. This ring is not meant to prevent any threat not associated explicitly with testosterone. While the ring may help diffuse a situation or reduce microaggressions, we do not recommend this ring in more serious cases of sexual aggression. When using the ring, make sure to open it fully and within smelling distance of your intended target. We cannot ensure any outcome. All bodies are chemically different and we cannot guarantee your mixture of chemosignals are the same as others. Hormonal medication may affect your production of tears and chemosignals. Side effects of crying include stress and mood, dehydration, and nausea. Ask a therapist today and see if crying is right for you. BioShield. Because while we're waiting for social and legislative change, sometimes all we have is our tears.
Morning, Director. Your 9 o'clocks are here? Okay, go right ahead.
So, you're going to be the best diaper.
Yep.
Yes.
Hey, your baby.
Yes.
Yup.
What's your story?
Pretty typical, you know, I'm the classic diaper made of polyethylene and polypropylene plastic, sodium polyacrylate, and wood pulp. I grew up deriving my parts from fossil fuels, and not to brag, but my family is known as the national leading brand and have historically been named the best diaper, so I pretty much thought I'd get this job, no question.
Interesting.
Gee Willikers. Well, I was born on an orange grove in Cali, but my most formative and fermentive years were spent in a lab where my absorbent core was grown as bacterial cellulose with the help of sugar and a symbiotic colony of bacteria and yeast. That BC was freeze-dried into an aerogel, which is 99% air and makes me light and absorbent. I'm new to personal hygiene products and really excited to serve your baby.
What is your greatest strength?
My strengths are pretty standard. I can absorb solid and liquid waste and keep it out of the way for your baby, but like, everyone likes me already, you know, so it doesn't really matter.
Actually, we did a little bit of a competition out there in the lobby, and I was able to absorb 32% more water. In addition, of course, to being sourced from agricultural waste, which is a big strength.
Last question. Where do you see yourself in 6 months?
Yeah, pretty much the same as I am now.
How about in 1 year? 100 years?
Yeah. People from my family don't really degrade, you know? So after I collect what I'm supposed to, I just kind of lounge around in landfill for at least 500 years.
Well, I'm made of all bio-based materials, so I really see myself blossoming here with your baby. Biodegrading in a year or so with the help of mushroom spores that I have integrated in me, and hopefully being promoted to compost and really fostering the growth of more plants that can make the world better for your baby.
Thanks for coming. We'll tell you the oldest seasons.
Thank you.
Thanks.
Well, what do you think? Director. Which one do you like?
Orange.
especially Timothy. It smells like sunshine, mild and non-glare. And I can see it holding a bright, clean jewel every morning. My name is Hua and I was born in Beijing. Recently, I turned 40. When I was 20, the trade war between the US and China ended. People came to a consensus deal with food and energy crisis. At the time, they conducted a population census over the globe and cleared unlivable areas. Due to the extremely harmful air quality, Beijing is one of them, so we were driven away from the city. Before we left, we received a genetic checkup. According to the report of their examination, I was assigned to New York. A couple years ago, the new law announced each of us should insert a chip into our wrists in order to make sure we're resistant to extremely frequent fluctuations of the supply of food. They said, the chip is a new type of external mRNA which doesn't trigger transplant rejection. It transmits a steady and continuous signal to the human brain and somehow helps us digest more efficiently. After that, I gradually began to eat more and more hay. Somehow, I feel like I'm a rabbit. The recent food conditions are relatively stable. It is a suitable day for traveling. Tickets are not difficult to buy. Instead, food preparation is more difficult. I redeem my citizen points for traveling package of food. It took me a lot of effort to get the point. I love my parents, but I don't love what they eat now. So I always prepare my own food when I go visit them. We used to eat together and share dishes. It was a common custom here. After we changed the food that we eat, we still eat together and even exchange nutrition from each other. I ate 7 kilograms of hay today, but I still need more protein. I asked my parents extracting and exchanging the usable resource with me. After digesting, the food converts into another format. The nutrition is discovered in bubble, which is protected and then transferred via optical tweezers to another person directly. Nature cannot accommodate the population we have now. So we eat the food from the bottom of the ecological pyramid and try to operate on every use. It's easier to feed more people on food at lower trophic levels rather than higher ones. No one is an outsider. This is how we live together in 2070. Beyond 100%.
Genuine.
We are from the Greenside Design Center, College of Design. My name is William Rawlings, and me and my partner...
The Biodesign Challenge Summit, Day 2. I'm excited to start off this next session with Greenside Design Center.
We are from the Greenside Design Center, College of Design. My name is William Rawlings, and me and my partner, Candace Holland, will be explaining Golden Leaf, modular water filtration system.
We would first like to introduce the context and relevance of our project and why we decided to make a water modular system, the needs and insights we found, and the existing biology and science that we researched and used to develop this project. Golden Leaf is a project that aims to bioremediate polluted water sources while simultaneously monetizing the waste the system generates to create wealth for impoverished rural communities. The name for Golden Leaf symbolizes rebirth and has connotations of fixing things and returning them to their natural state. Therefore, it's a fitting name for this project because it's a system that uses plant-based filtration to restore polluted water catchment areas to their natural unpolluted states. Golden Leaf comprises of 3 components that work together in a unit or independently of one another depending on the pollution that it is being applied to solve. Our project is framed within the Sustainable Development Goals. The primary one is number 6, which is clean water and sanitation, and interlinks to secondary other ones, which is life below water and decent work and economic growth. But why? Why we decided to focus on water is that our South African Minister of Water and Sanitation has revealed that water pollution poses the single biggest threat to the country's water security and quality, and is considered as one of the top 5 threats to our economy. Through this, we identified the 4 largest water polluters in South Africa. The first one is household sewage waste. We have very brittle infrastructure, therefore a lot of broken pipes, and our sewage doesn't get where it needs to go, and thus seeps into the ground and leaks into our catchment areas. The second one is industry waste, such as businesses and factories are illegally dumping their chemicals and waste into our water sources. The third one is the mining industry. South Africa is a mining country, and thus we have different types of mines with different types of minerals, and therefore different chemicals to mine those types of minerals. However, with that, our mines, with a permit or with not a permit, are found dumping in our water sources and contaminating them. The fourth one is our agriculture. We also have a massive agricultural sector, and the pesticides and herbicides used in treating these crops create runoff onto our water catchment areas. How our water is used in South Africa: 70% is used for agriculture, 20% for industry, and 10% for personal and household consumption. 7% of this water is also used for mining. However, even if it's a little bit of use into it, the mining industry still produces almost 3 times the amount of toxic water back into our system. Further context would be that for South Africa's water, 70% of it is surface water, 9% is groundwater, and 14% is renewable water. And that less than 50% of South Africans have access to pipe water in their homes, thus making them travel long distances to water sources. Sometimes it is a tap, sometimes it is water catchment areas like dams and ponds to get their water. We looked at current solutions that have been implemented, some in South Africa and some in other places. The first one is the water hyacinth plant. This has been currently used in the Hartebeespoort Dam to clean and purify polluted water. However, they are a very invasive aquatic weed, and they use a lot of oxygen in the water and also suffocate the living organisms underneath when they reproduce quickly. Coca-Cola teamed up with a local fertilizer business to harvest and compost the water hyacinth and turn them into fertilizers for farmers. However, they were not quick enough and have been overwhelmed by their reproductive systems. Thus, water hyacinths are now a big problem in the Hartebeespoort Dam. The second solution we looked at was aquatic wetlands. They have been implemented into different waterways in different parts of the world and have been used for a very long time. They are a very successful solution which use different types of plants to absorb the toxins in the waters, then filter and release and clean the water back. And they use phytoremediation to purify the water. The third current solution we looked at was the super sponge. The super sponge is a synthetic sponge, just like a kitchen sponge, which was infused with selenium bacteria and nanotechnology. It was used to absorb mercury from polluted water within seconds. Once you have captured your sponge and you squeeze the water out, it'll become clean because the nanotech and the selenium bacteria hold on to the mercury within the sponge.
Now I'd just quickly like to tell you a bit more about what exactly our system is and how it works. Golden Leaf is a scalable, modular water filtration system that consists of 3 components that can be used either together or independently, depending on the kinds of water pollution found in a particular water source, as each component deals with a different kind of water pollution. First component is bioengineered water hyacinth, second charcoal-infused mycelium sponge, and the third woven basket, which holds everything together. Golden Leaf is built upon a couple of different initial ideas. These ideas are as follows. The first one is a drinking filter system, which comprised of creating a biologically engineered sponge that can be placed in a bucket, filtering water as the user carries it home. Second one is a living sponge spliced with water hyacinth, which can be used to quickly absorb spilled mine waste. These would be dehydrated and sealed in water-soluble capsules, making them small, portable, and easy to use. And third is floating islands. Basically, this would comprise of creating floating islands that combine the water filtration abilities of oyster mushroom mycelium and water hyacinth to remove harmful substances from water catchment areas. The components used in the final design are as follows. The first one is the water hyacinth. The water hyacinth is the world's most invasive waterborne weed, and despite this, it is great at absorbing minerals and metals in water and can survive in pH levels as low as 4, making it ideal for remediating water contaminated by mine waste. As seen in the Hardebeespoort Dam, though, there are some problems with using this plant for bioremediation, which were addressed with 2 of our major tweaks to this plant. The first major change that we made is that we spliced it with the hydrangea, allowing the flowers to change color based on the pH level of the water. This allows the plant to effectively function as a visual indicator of the quality of the water. The second major change, probably the most important one, is the introduction of the terminator gene that eradicates the plant's reproductive system. This effectively renders the second generation of the plant infertile and stops the problem of it spreading out of control. The second major component is the mycelium sponge. The mycelium sponge specializes in trapping organic agricultural pollutants such as herbicides and pesticides, as well as helping with processing sewage. This sponge is, in and of itself, made up of 3 major components. The first component is the mycelium sponge base. Mycelium is used here as it is a strong, buoyant, malleable, and completely renewable biomaterial. This sponge is then infused with charcoal. Charcoal is a natural water filter, which excels at trapping contaminants due to its porous surface. Finally, enzymes are used to break down any pollutants that the rest of the system cannot deal with.
The third component is the basket, which acts as our vessel and container to keep the entire system in place and be able to float on the water. Our inspiration was African basket making, as it is an age-old craft that was historically used for agricultural produce. We looked at Zulu basket making, which is native to South Africa, and we also looked at the African East side, which had fishing trap baskets. The fishing trap baskets were more cylindrical shaped, and the Zulu basket was more plump and round. We decided to combine these 2 aesthetics together to create our final form. The weaving technique that we decided to employ was the twining technique, which is used in creating fishing baskets because it is breathable and that it lets the system breathe and actually float in the water, as well as to suck in the water for the sponge to absorb. The shape we used was a circle, as it represents community and unity. The basket exploration that we did, we looked at a very modern, more organic version for this one. However, when we tiled it together, there wasn't enough sunlight being let through, and this is a major problem in our system because there needs to be sunlight coming through to the other organisms underneath the water surface.
The final design for these baskets gets around this problem, as it forces large gaps to be left between baskets, which allows sunlight to get through, which in turn sustains algae that lives underneath these islands. Keeping this algae alive means that oxygen levels in the water stay high, allowing aquatic ecosystems to survive.
What sets our system apart is the scalability of our system. It can go from an individual all the way up to a commercial aspect, such as NGOs, a government, or large corporations. The system can be implemented as one component in a pond, all the way up to maybe 10 or 3 different types of components in the dams. Even one person can make a difference with this system. The implementation and how our project will be used, how it will be funded, and how it will be distributed to these different types of individuals in the community. We decided to create a startup kit, which can be purchased from garden centers by individuals, small communities, or can be provided in bulk for larger corporations. The starter kit is comprised of a year's worth of water hyacinth seeds and an instruction manual on how to care for the system as well as assemble it. The manual was created not only to provide instruction, but it is also a way that people in rural areas who do not have access to internet or anything like a mobile phone to access a website can be able to implement the system and assemble it and also have the information that they need.
Okay. And the following is an example of how the system could potentially be integrated or implemented in a rural community. Baskets could potentially be produced by the women in rural communities. The weaving of these baskets will generate income and job opportunities. The mycelium sponge and water hyacinth seeds will then be delivered to the rural community separately and then assembled inside these baskets. Baskets will then be implemented into local water catchment areas. The modular system can be customized depending on the kinds of pollution found in a specific water source, with different quantities of each component being used in different situations. The system will then filter the water and output clean water to the people living in rural areas near the catchment area. This water will still need to be boiled before it is consumed, as the system does not eliminate harmful bacteria. Then the life cycle. Here I'd like to discuss the system lifespan, how the components can be recycled, and how the byproducts of the system can be used to add value to rural communities. Every 60 days, the system needs to be harvested and replaced. This generates large amounts of biomass, which is actually quite a valuable material. The biomass generated from the water hyacinth can be burned and phytomined to extract heavy metals. It can be composted into a mineral-rich fertilizer, or if it does not have enough minerals in it to make it worth phytomining or to be used as a fertilizer, it can also be burned to produce electricity. The sponge can also be composted into fertilizer if it is exposed to a water source containing little sewage. If the sponge is too heavily contaminated with sewage, it can be burned to produce electricity. And then the basket. If the basket is still in a good condition after its 60-day implementation, it can then be reused either again in the system or as a normal household basket. It could also potentially be composted into fertilizer or burned for energy. Then to conclude, we'd like to discuss the risks of our systems and run a comparison of the strengths and weaknesses. The strengths of the system is that it is scalable, versatile, and good for the environment. It also helps people living in impoverished rural areas by giving them access to cleaner water and creating employment opportunities, generating wealth in these communities. With the trade-based skills residents of these communities acquire through building and maintaining the golden leaf system will also help them when looking for work in the future. Then risks. People could potentially exploit the system for financial gain, completely taking over communal water sources to grow biomass and limiting other people's access to drinking water. The islands could become breeding grounds for mosquitoes and other disease-spreading vectors. If harvesting is left too late, the water hyacinths used in the system will die, and in the process suck up lots of the oxygen in the water that sustains underwater ecosystems. The enzymes used to remediate some kinds of area-specific pollution may be harmful if consumed by humans. And then finally, our explainer video is packaged along with this presentation. If you're interested, please check it out. And then thank you for listening to our presentation.
All right, welcome back. We are going to have the students join us for this call. Judges, why don't you come on and start with your questions? Any questions from the judges? Great. Why don't we start with Jason, and then we'll follow up with Simone, and then Allison.
It's a really interesting project. I did wonder if you had thought about a couple questions. One is like how culturally it would be implemented into different cultures in South Africa. And then secondly, I worry about anything that covers the full surface of a water surface, because there's a lot of life that depends on that sunlight underneath it. And I was kind of wondering if you had thought about that too. So sorry, 2 questions.
Cool. I'll talk about the sunlight first. Sunlight is a major concern. Turning the baskets into circles did allow more sunlight to go through. We weren't entirely sure if this would be enough, but it is an improvement. And I think that you would want to probably cover segments of a dam. You would never want to cover an entire catchment area with the system. You still do need areas to be left alive. We haven't done any actual testing on like ratios of water that would need to be left uncovered. So I think it is a pretty good concern.
Go ahead, Simone.
Right. Great presentation, thank you. Just a couple of things. I grew up in Bangkok, so anytime I see water hyacinths, I sort of worry about entire canal systems being overtaken because, as you mentioned, it is incredibly invasive. and pretty hard to control. So I've seen these plants kill entire canals. But my question is actually more about the flower. You use the flower as the indicator. And I'm just curious how often and how quickly sort of water hyacinths actually do flower, because my recollection is not all of the plants do, and it's not that often. So if that's your indicator as to what's in the water, I'm just wondering how you thought about that.
So we did do a lot of research in that, and we found that it did come from America first, and that when it flowers in a bunch. It doesn't flower when it's on it by itself, and that depending on the weather, if it's in a tropical sphere, it can flower for a very long time. It flowers for, I think, mostly in summer and spring a bit. But we have very hot weather here, so they don't die out at all. We don't have frost to kill them. And we also clump them together in our basket so that they can flower together. Otherwise, if you leave them individually, they don't flower. And they can bloom for a long period of time in a hot climate, which we also have here. I hope that answers the question.
Yeah, we may have some issues in winter, but it hopefully shouldn't be anything too serious if we understand how the plant works correctly.
Thank you.
Allison?
In a related question to Simone, I was wondering, also great presentation, by the way, how will users be able to know that water is clean? And I mean, I'm guessing that the cleaning is going to depend on the volume of water, the concentration of whatever the pollutants are, the presence of other plants, potentially other non-terminated water hyacinths that are also cleaning the water. Is there some kind of calculation that, let's just say, a user can use to see, like, when is the water clean? When is it been appropriate? When has it been cleaned? I know that the indicator you showed was for pH indicator, but of course there are many other, as you've said, there are a lot of other pollutants. So, yeah, how will a user know the water's clean, and how many of these things they need to plant in their water body?
Yeah, like the water hyacinth with the pH level, it's a very, very basic indicator. Yeah, that's a good point. We hadn't considered that thoroughly enough, I don't think, because the nice thing about the pH indicator is that it indicates the presence of mine waste very quickly, and also the presence of soap, which is a major problem with in areas surrounded, well, in rural areas, people often wash their clothes in rivers. So the presence of soap is a major issue. It can only really tell between those two. So like look for the presence of mine waste and look for the presence of soap. It isn't a whole lot more specific than that right now. We'd have to do some further developments on that, I think.
Great, thank you, Candace. Thank you, William. Sorry for the video trouble here. Some technical difficulties, but I think we've got it taken care of. Unfortunately, we have to move on to the next project to show. That is Jacksonville State University. Let's see the video.
30 years ago, the rising costs of healthcare crippled the public. With incomplete insurance plans, gross medical overbilling, red tape referrals, and a plethora of legal hoops that contributed to a broken system with a population that was unhealthy and overburdened, until VitalSkin made healthcare personal by combining continuous vital monitoring, medical-grade lab analysis, and a real-time alert system. VitalSkin was the beginning of a healthcare revolution. VitalSkin is a 3-part system designed to work best for you and your loved ones. The first component is the continuous monitoring implant. This device is placed in your upper arm in a simple outpatient procedure done under local anesthesia. The easy access port allows for you to take care of all of your medical needs, from taking medication or receiving vaccines. With the ease of inserting a simple cartridge, it can measure vitals such as glucose levels, oxygen saturation, heart rate, and more. For more complex treatments and in-depth health analysis, the implant connects directly to the home processor that functions as an on-demand medical lab. Personal units can be purchased from your home, or public use units are available at most local pharmacies. For routine health monitoring, once a week you will plug into this machine, and in 2 minutes you and your healthcare team will know all they need to know about your health. The machine will be connected to an app on your phone, so you can look at your own information on demand, while also sending the data to your healthcare team to keep them up to date and informed. They can even write you prescriptions for cartridges, which will then be mailed to your house upon any signs of trouble. The final component is the namesake, VitalSkin. This is a multifunctional sleeve that protects you and your implant, while also providing you with instant feedback on your health. Using the in-depth health analysis from the machine, the VitalSkin sleeve will utilize electrodermal screening, also known as skin resistance, allowing the perception of the smallest changes in an individual's body chemistry. Studies have shown that skin resistance varies with health and disease, and that these variations are measurable. Using the body's own electrical system to measure resistance and capacitance, healthcare providers can assign normal ranges to each individual. If any monitored measurement falls outside of an individual's normal range, the VitalSkin system will notify the wearer with a visual cue. While the implanted monitor is unable to diagnose, it serves as a crucial preliminary step of a real-time notification to a potential health problem. VitalSkin. Wellness within reach.
Thank you, Jacksonville. Great video. Judges, if you have questions, come on in. Well, I'll start. How did this project come about?
We as a group were really interested in just the culture behind a lot of this biodesign stuff, and just the fact that we're really seeing that we're gearing towards home health systems. You already have Apple Watches, Fitbits, everything is already monitoring your health at home, and we're seeing that it's hard to get appointments, it's expensive to get appointments, and so we're really anticipating the fact that we're going to go to telehealth systems, especially after this pandemic that we're seeing. We're already starting to utilize some of that with these drive-through services and everything. We're really long story short, this is where we're going. We wanted to jump in on that and make sure that we could kind of control it a little. And by we control it, we mean we the people and not a corporation. And that's really the point of our implant, is to put your health in your hands while we're seeing that it's, I don't know.
And two, we kind of looked at the current marketing that is geared in the medical fields for even prescription drugs. You can see commercials on your TV for prescription drugs that you can't even get yourself. You have to go to a doctor. So the marketing is already there, geared towards individuals trying to make those decisions, but they're not able to. So this system just brings it closer to home and really ties it all in to where you have control, whereas now you get all this marketing where you can see all this stuff, but you really can't take part in it. So we just wanted to kind of explore that idea more and see where we could go with it.
Great, thank you. Harris, why don't you follow up?
Great, yeah, that was really interesting concept. So I guess I wanted to ask from maybe two-part question. One is about kind of equity of access. How do you envision that everyone may have access to this, and how does the kind of the healthcare disparities between kind of populations in terms of access to healthcare contribute to this, or how this project could help to drive things that are maybe more equitable? And then the second is really about kind of safety or cyber safety, right? Like, how do you ensure that these things are safe from intrusion or kind of unwanted kind of access to health information by the users?
Okay, to address the first question, equity of access, that's something we've really talked about a lot. It's important for us to understand that healthcare is expensive. And while you can look at this at the long term, like, oh, this is gonna, long term, it's gonna save referral. You're not gonna have a referral appointment for your health insurance. You're not gonna have to, go through, you're sick. Okay, now you have to go through 9 doctors, multiple appointments, miss all that work. Like, you can look at the long term that way, but short term, how are people going to have access to this device? And what we're really gonna try to do is utilize a lot of these first world the abundance of resources that our first world nations have. We're going to use a lot of investing, a lot of crowdsourcing, a lot of people for we're going to utilize that dispensable income that a lot of the population has and make it so that we can then donate a lot of this to some of our lesser fortunate brethren. And that is idealistic, I understand, but there are already a lot of companies that are doing that. If you've heard of LightAid, they were a group of 2 women from MIT who they went through and they had this safety light, and they utilized a lot of that investing and selling it to America and England and Canada, our first world nations, so that they could use that profit, and instead of pocketing it, they can donate this system to other people. And then second, we have different levels of access that you can have. So yes, you have to have this implant, but that at-home health lab, if you can't afford one, you don't want one in your house, there are always going to be Rite Aids and pharmacies and doctor's appointments that you can go and you can utilize that system there. Just like we have blood pressure reading cups in pretty much every mall everywhere, this will be the same thing. As for the second part of the question, that is more of a computer question for Will. I cannot answer that.
So as far as security goes, we were trying to keep the system more closed loop than having it versus Bluetooth and kind of these things that are more easily accessible to people who might want to abuse the system. And also we were going to keep, as far as the implant goes, have it as minimal as possible and really have the home health system that's in your home be the meat of the program that actually does a lot of the processing and keeps your data. So you won't really have your data on you at all times. It'll be in your home, which you won't be out walking around having that issue. So we try to keep it in the home versus out on the streets in that regard with the home health system and having a little pod at home where you plug in and it kind of gives you that manageable package data that is easier to read than maybe you don't have to have a medical degree to see kind of what's going on with your body and what you might can tweak or change to really be healthier. So
great. We're gonna just we'll finish with Elaine.
That was basically my question there about the privacy and hacking accessibility, because your tagline is vital skin, wellness within reach. And so my automatic question was for who? And in that regard, in terms of costing and let's say you have a chronic disease, right? Is the costing different? How long does this device last? What if your body rejects? All the things that can go wrong with it. Had you thought about the actual pipeline of what this would be? Could you speak to that and things that can go wrong? I sort of wanted to hear more about the hacking and privacy issue.
As for the long-term costs, we honestly really think it's going to help those with chronic diseases. It's going to be cheaper, because right now, if you have diabetes and you either have to pay for an insulin pump, so you have to constantly pay for insulin, you have to pay for the needles constantly, you have to pay for the lancets constantly, you have to pay for everything constantly because everything needs to be sterile. Where this system gives you an easy access port where it will always be sterile, so you can pretty much buy 1 or 2 things, and you would just have to buy, again for diabetes, just buy the insulin. If you have an autoimmune disease and you have to take a lot of medication, you would just buy that little cartridge, that medication to insert. And so overall, we really think it's going to reduce everyone's costs, personally, hospitals, everything.
Great. Thank you so much. We're out of time. Next up, thank you, students. Next up, we have projects that are participating in the Mana Prize for the Future of Beauty. These are our 2 finalists. The idea behind the prize was to ask students to consider all aspects of beauty, including beauty routines, concepts of beauty, ingredients, packaging. Our 2 finalist teams this year come from University of California, Davis and Universidad de los Andes. We'll start with UC Davis, Bioshine.
Hi everyone, this is Team Bioshine from UC Davis. Due to the negative impacts of the mica industry, we have created a mica replacement by using cellulose nanocrystals.
This Statista research paper outlines the most frequently used brands of all beauty products. 100% of the top 15 brands that produce highlighters use mica in their product. We also surveyed 40 women and found that 78% of our consumers were either somewhat or not aware of the use of mica in beauty products. Due to this large gap between awareness of mica and frequency of highlighter use, we decided to target both consumers of highlighters as well as manufacturers like Mana as the audience for our product. We also learned more about the numerous concerns of mica's production and use.
There is a significant child labor issue with respect to mica mining. Children are exploited and labor laws are frequently violated. Additionally, many respiratory health concerns arise for the workers due to frequent long-term inhalation of mica during the mining process. Environmentally, mica is a non-renewable resource. Mining for mica leads to significant deforestation and displaces wildlife, which poses a major threat to the well-being of local ecosystems. Many products containing mica, as well as synthetic mica replacements, are not biodegradable. In terms of cultural concerns, many consumers of the beauty industry do not care or are not aware of the unsustainability behind their cosmetic products. Bioshine wants to change all of this by promoting ethical values and offering a sustainable, bio-based alternative to mica in the beauty industry.
What inspired Bioshine? We looked to nature for our solution. Mica shines, but so do many organisms. Take butterfly wings, beetle exoskeletons, squid skin, or even bacteria that produce iridescence. It turns out that cellulose, one of the most abundant organic molecules on the planet, can be coaxed into forming iridescent particles known as cellulose nanocrystals. Research articles, such as this one by George and Sabapati, helped us understand the optical and mechanical properties of cellulose nanocrystals, their high crystallinity, and thermal stability. Since a highlighter needs both shine and a skin-compatible base, we chose glycerol for our gel solution. The materials that we will use are bacterial cellulose, known as BC, cellulose nanocrystals, known as CNCs, and glycerol. We start with bacterial cellulose, BC, because unlike plant-derived cellulose, it has no lignin, hemicellulose, pectin, or wax, and therefore this reduces chemical waste. Also, nanocrystals of BC have the highest crystallinity and thermal stability. Bacterial cellulose is used in various applications, such as packaging, food, and facial scrubs. BC is generally recognized as safe by the FDA. Cost-effective BC is grown in rotary bioreactors in medical industries. To achieve high output of the biofilm, Commagotibacter sp strains will be used. This bacteria is non-pathogenic, so it is harmless and safe for humans. Compared to others, this strain creates the highest yield of cellulose. Therefore, we will work with this bacteria in our project. To sustain it, we use the standard media for pharmaceutical quality bacterial cellulose. This media contains carbon, nitrogen, and other macro and micronutrients required for bacterial growth. Glucose is commonly used as a carbon source. Because glucose is found in fruits, we are also considering alternatives, such as fruit waste from farmers. After harvesting our BC, we sterilize it with food-grade sodium hydroxide. We then wash it with water until neutral pH is achieved and dry it at room temperature. The cell-free BC is then hydrolyzed and functionalized by treatment with sulfuric acid for an hour. The mixture is centrifuged to separate the cellulose nanocrystals from the acid. An ultrasonicator helps the nanocrystals stabilize into an iridescent film, which occurs through evaporation-induced self-assembly. We then grind up the film into tiny shimmering particles. Unlike the carbon chains in the bacterial cellulose, the final composition of nanocrystals is a helical structure, which allows different wavelengths of light to pass through it and reflect back. This allows us to see a range of iridescent colors. Different amounts of ultrasonication can alter the pitch of the helical structure, changing the wavelengths of the light that is reflected. We therefore hope to be able to produce red, orange, blue, and violet colors once we are able to return to the laboratory. We then add our iridescent nanocrystals into glycerol, a biodegradable and non-toxic gel solution that is commonly used in eye drops, lotions, food, and beverages. Glycerol can be made from vegetable oil subjected to hydrolysis, which then forms glycerol and fatty acids. Although cellulose nanocrystals have been used in gels, plastics, glazes, and food packaging, Bioshine's application of them for shimmer is absolutely new.
The risks of Bioshine first include our product may irritate very sensitive skin. Tests will need to be done. Second, our product is not waterproof, and humidity might alter the integrity of the cellulose nanocrystal structure. Once we are allowed to experiment in the lab again, we will explore this. Lastly, the acid waste from synthesizing cellulose nanocrystals could be misappropriated. With more research and development, we hope to minimize acid use and acid recycling in our production process, as well as research other methods of producing stable, highly crystalline cellulose nanocrystals.
Bioshine strives for a circular product life cycle. We hope to partner with a variety of vendors to create our sustainable product. Bacterial cellulose is grown with glucose inputs and sterilized with food-grade sodium hydroxide. Glycerol comes from the vegetable oil industry. To get the nanocrystals, we hydrolyze the BC using sulfuric acid. This is not an ideal solution. With more research, we hope to discover a less toxic process. We then combine the nanocrystals and the glycerol to create Bioshine. Our containers are made from cellulose acetate butyrate, or CAB, which has been used for pens and handles. It is biodegradable, resistant to UV rays and outdoor weather conditions, which will preserve the shelf life of our product. Companies like Omicron Plastics create CAB containers. Our product is then transported to retailers where consumers can purchase Bioshine. We hope to offset the carbon dioxide produced through transportation by fuel-efficient vehicles. After consumers use Bioshine, the cellulose and glycerol will wash off and biodegrade. The containers can be reused or recycled. The waste sulfuric acid in our current process is neutralized and then recovered by a company like Veolia, which claims to provide a cost-effective, environmentally sound alternative to traditional sulfur management systems. This allows us to then reuse the sulfuric acid in our process. Finally, the leftover product can be mixed with fertilizer to help find vital nitrogen, phosphorus, and potassium molecules in soil, prompting the growth of new sources of glucose.
These are the cited sources. And these are the research articles that we worked with. Thank you for listening to us, and we hope that you enjoyed our presentation.
Thank you, Team Bioshine. Great presentation. We're very lucky today to have Maria and Yelena with us from MANA. I'll let Maria and Yelena kick off the questions.
I have a question. It's actually a great presentation, and I'm very excited and look forward to see already your unique pearls on market. But question is, did you conduct any research to realize market, like cosmetic industry needs for mica? Because volume what we use of mica is huge, and how fast you can deliver your mica to cosmetic industry. Not mica, your Bioshine to replace mica to cosmetic industry. How fast you think it can be done in 5 years, in 10 years, for us to predict, because it could be a new trend in cosmetics.
I think that it will be really hard to kind of like mass produce it, so it will take a couple of years to like have enough money to have the machines, the equipment. However, I think it's really easy to make cellulose nanocrystals just because we're using bacteria, and we know that bacteria reproduces every 20 minutes. We have research on how to take care of the bacteria, and they transform the glucose into cellulose, and then we just add sulfuric acid, and then other processes are made, and then we get cellulose nanocrystals. So the process itself, I think it's really easy to do, and I think we can produce it on a mass scale. It's just a matter of having the right equipment and how long it takes to have that.
Yes. Thank you. And another consideration, like cost, because I know this biotechnology is not like cheap technology, and it's like just the beginning. That's why until you will get full scale and reduce cost, any possibilities you think in your technology can help, because, cosmetic industry, it's not only prestige market, it's also mass market, and a lot of companies, they make actually a lot of money on mass brands. This way, they like to kind of scale down your technology and be available for mass market also. Any possibilities of that?
Yeah, so from what I've researched, the bioreactors would be around $3,000 to $30,000 depending on the size of them. The bacterial strains are around $300. The grinder is from $10 to $50. And then for the media, depends what media we choose, but the most important things are glucose and yeast extract. So for glucose, it would be $147 per 10 kilograms, and yeast extract $327 per 2.5 kilograms.
Okay, thank you.
I just have a quick question. Hold on for a sec. How is this different than the other synthetic mica that's currently in use in the cosmetic industry?
This is biodegradable because the other synthetic mica, they coat it with titanium dioxide, which is proven to be carcinogenic to humans and also bad for the ocean, for corals and so on. So our product is biodegradable.
So, I mean, that's huge. Huge and fantastic. Thank you for that.
Oh, and just another thing about toxicity, because if it's going to be drained in oceans or lakes or whatever, the particles that are made for dye, so like, I guess like positively charged dye materials that people use in the textile industry, or like metal oxides or metal small pieces adhere to the CNCs because they're negatively charged. So then this creates kind of like a flocculation, and so it would make the water less turbid and more clear because like all of those particles can
like on the ground basically, because the reason why they float is because they're very lightweight, so like it increases the weight and then it can make the water clear. I don't know if that made sense, but I can explain more if you want to.
No, it made sense, and this is a great discovery. Thank you.
Can you talk a little bit more about the strains of bacteria that you plan on using? Are you planning on genetically engineering E. coli? Because you did mention the 20-minute turnaround time for the cells to divide.
No, we are not going to engineer E. coli. We can use this bacterial strain. It's called Commagotibacter, and it produces the highest yield of cellulose.
Great. We are out of time. Thank you, Bioshine.
Thank you.
Fantastic presentation. Okay, we're going to move on to Universidad de los Andes, Linneo.
माइंस की खदानों में जब घुसते हैं तो वहाँ पे बहुत अंधेरा भी दिखाई देता है और हम लोग को बहुत डर भी लगता है।
Do you know what are the key ingredients in most cosmetics? The Journal of Applied Toxicology in 2009 stated that more than 10,000 harmful substances are used in cosmetics, and yet only 200 substances have been banned by the FDA. Does our skin suffer when we apply harmful ingredients? Let's talk about the skin around our eyes. Because it is one of the most sensitive and thinnest areas of our bodies, substances and toxic ingredients are absorbed quicker, causing allergies, skin sensitivity, and sometimes even cancer. In color makeup, eyeshadows' key ingredients are pigments. These animal and vegetable origin pigments often contain insecticides, fertilizers, and heavy metals. Ingredients that give color as orange, red, yellow, and black are of mineral origin, as for example iron oxides. The shimmer you find in eyeshadows, highlighters, nail polish, and others is given by mica, a valued mineral within the industry for its ability to reflect and refract light. Also used are excipients and binders, where its most commonly used ingredient is talc, because of its lack of color and its strong humidity absorption. But what are the impacts behind the use of these ingredients? Iron oxides, mica, and talc are sourced from open pit mines, altering its ecosystem and causing overexploitation of minerals, not only harming the environment but also the human body in the process. But what's not commonly known is a quarter of the world's mica comes from illegal mining in India, where over 22,000 children are submitted to forced labor in dangerous conditions, placing their health and life at risk. Therefore, we present Linneo, a bio eyeshadow palette made completely from fungi.
Our palette contains 6 colors, each one corresponding to a specific fungi species. Fungi pigments act like a fungal armor, protecting the host from harmful environmental conditions such as solar radiation. The biopigments also act as bioagents. Bioagents boost the human organism when consumed. Just imagine what it can do to our skin when applying nourishing biopigments, as it activates the skin and regulates exposomes repercussions. Why should you choose Linneo over the infinite possibilities in makeup brands? Because in addition to coverage, Linneo also provides nourishment to your skin. Our formula provides hydration, UV protection, antioxidants, and anti-inflammatory properties, decreasing the aging process around our eyes. All of this happens thanks to the bioagents fungi offer. Linneo addresses the situation by replacing harmful ingredients, therefore creating a socially, economically, and environmentally sustainable cosmetic. But why fungi exactly? Colombia's biodiversity has always been a great source of inspiration and research. We can find around 1,500 species of macrofungi alone. We went out, explored, fell down, got up, and got dirty, initially finding more questions than answers. But we landed on a wooden mushroom from the forest near our university, and that's when Linneo's journey started. Our first attempts were unsuccessful due to the macrofungi fragility. However, it inspired us to explore further a kingdom full of color, leading us to discover filamentous fungi, setting the way. How is all of this possible? An eyeshadow requires excipients, pigments, colorants, corrective, active, and preservative substances, but Linneo would only require 2 substances, pigments and binders. Pigments not only provide color to our formula, but also act like active substances, providing hydration, UV protection, anti-aging, and antimicrobial properties. The binder acts as an excipient and corrective substance. It thickens and smooths the formula while nourishing the skin. Using only 2 substances guarantees an ingredient reduction and lowering Linneo's carbon footprint. The process starts by growing and incubating fungi in the lab. When the media is full of color, the culture is extracted, sterilized, and filtered. Liquid pigments are obtained and then dried and ground into a fine powder called microma, a sterilized biopigment. Linneo can be escalated by using a bioreactor containing different compartments controlling airflow and temperature, ensuring fungi's optimal growth. The environment's light and pH can be regulated as well. The reactor's horizontal containers optimize culture space and pigment production, as fungi grow faster when cultured in larger areas with less depth. By controlling culture conditions as temperature, humidity, light, and media, we can obtain different pigmentation shades from the same fungal species. Pigments require binders, so eyeshadows can be applied smoothly and pressed back in a steel pan. Our binder is obtained by drying Ganoderma lucidum fungus. It has low rates of humidity, meaning that when dried down, the powder's volume doesn't diminish drastically. The resulting powder is colorless and rich in chitin, which is perfect for mixing color and nurturing the skin. Finally, pigments and binders are mixed together and the eyeshadow is ready to be pressed packed in steel pans and placed in the palette. We use different fungi to obtain both pigments and binders for our formula. As of now, we're working on 6 initial colors and obtaining approximately 3,000 eyeshadows in a 150-liter bioreactor. However, we're working on our biopigments optimal development. Now, to obtain a sustainable palette, we suggest using agroindustrial waste from Colombia's sugarcane industry, creating a biomaterial used for Linneo's eyeshadow containers and packaging. We propose this be made according to the agroindustrial waste available in each country to reduce costs and carbon footprints. Also, Linneo works with interchangeable refill pans, which will come in biodegradable packaging. Linneo is committed to respect the planet's biodiversity and our diverse communities. A refined cosmetic must be born from noble intentions. We present to you, once again, Linneo, Nourishing Beauty.
Welcome back, everyone. We have the Linneo team with us, and again, we have Maria and Yelena from MANA. MANA, feel free to ask your questions. Great presentation.
Thank you.
Maria, you go ahead, or I will start.
You go ahead, Yelena.
Okay, I have a question. How your pigments are pH sensitive? And then if you notice, if you change pH, color will change, because for some pigments, we even play on the special effect of pigments. For example, people buy green lipstick and it's turned to red lipstick, and people have a lot of fun with this color changing effect. Did you think about that? Do you have experience with that?
Yes, we did. We are conscious about this. However, we control the environment by controlling the environment and the characteristics as temperature and light. We can standardize the color that we produce with each fungi. However, it is a possibility in the future that we can experiment with these characteristics and specifications in order to obtain different colors.
I'm wondering with the pH thing that plays. Yelena, help me if I'm incorrect here, but that would become very customized, personalized beauty, right? Because as it's applied to each individual skin, they're going to get a unique payoff. Am I correct with that, Yelena?
Yes, that's what I meant. And plus, sometimes, as I said, it was a trend for color changing effects.
I mean, it's pretty brilliant when you think about it that way. Everybody's looking for personalized beauty, customized beauty, things like that. The fact that it is pH sensitive actually works to their advantage, right?
It's
yeah, it's very, very interesting. Very interesting. Is the fungi specific to Colombia, or is it something that can be grown everywhere?
The fungi we use are from Colombia. They are native. However, as long as we can ship this fungi to other places, they could be grown in a controlled environment in the lab, in order to not introduce another type of species that are not native from the place we're shipping to.
I'm curious about what kind of lab work you were able to do. Did you experiment with growing the fungi? Did you experiment with these bioreactors? Can you just talk a little bit about some of the hands-on work that you did?
Yes, we can. We did some experimentation in the lab. We managed to grow the fungi ourselves, and we managed to extract yellow and orange pigments already. But we are still working in the process of mauve, brown, and black colors, and still working on the process of the binder. But our time in the lab got cut short, so we will continue with that when we go back.
So yeah, I want to add something, because in the lab we could change some medium or pH or light that could affect the pigment that some fungi produces.
Thank you.
All of your materials speak to the eye area. I'm assuming this is something you would look to use across the face, perhaps even on lips. Is that sort of next steps for you, or was it specifically just for eyes in your viewpoint?
We started first with the palette. We analyzed the cosmetic market. We found that people tend to use more matte eyeshadows, but in the long run, sure, when we perfect this product, we could work on obtaining a wider catalog of products.
And have you seen any dulling of the color with the binding agent at all?
We have done some tests with the first prototypes of the pigments we obtained. And Karen actually has some samples in her home. And since we got those samples, the color of the pigment has not changed, but we will need to look into that later on, as soon as, again, we can go back to the lab.
Yeah, okay. Thank you very much.
Black pigments, let us know, because now it's big trend for mascara and eyebrow.
Yeah.
I know, and even blackberry extract, it's always very, very deep, like violet, not really black. But I think with your technology, you can find something that will give us natural black pigments, very like stable. And it's kind of for future. And I hope Mana will be first who will use it. And then I have some question. You mentioned that your pigments, they provide UV protection and antimicrobial effect. You have study supporting those claims, or those claims just came from literature?
Well, it's supported by the Journal of Agriculture and Food Science that biopigments have several functions, as you just mentioned. It's also proven that fungi have all those benefits, not only to the skin, but in the body. But yes, we are just based in literature yet.
Okay, thank you.
Thank you, team. We're out of time. Thank you, Maria. Thank you, Elena. We're going to now go to our lunch break. We will be airing some videos until about 1:15. Everyone, feel free to come back then. Thank you, everyone. It was a great morning.
Lava fields on the southern slopes of Mount Haleakala, the island of Maui, Hawaii. The turn of the 20th century. The forests are being lost to colonial cattle farming. One tree will be lost forever. The Hibiscadelphus walderianus. The Hawaiians called it Maui Haukuahihi, the mountain hibiscus. By 1912, it will be extinct. The habitat lost, the plant lost, the relationship between the two lost. Could we ever regain a glimpse of what was lost? A few years ago, a small group of synthetic biologists at Ginkgo Bioworks, a bioengineering company in Boston, set out to resurrect the smell of extinct flowers. Christina Agapakis, Ginkgo's creative director, went to the Harvard University Herbarium in 2016, where over 5 million specimens are stored. Searching the collections for extinct plants with her colleague Dawn Thompson, they found 20. 2 would hold enough information to unlock the scents of their flowers, each lost due to human destruction of their habitat. A third would reveal what may be an even greater loss. The first specimen was in the Malvaceae cabinet. It was the Hibiscadelphus walderianus, from which she cut tiny tissue samples. The second was the Orbexilum stipulatum, or Falls of the Ohio scurf pea, last seen in Kentucky on Rock Island in the Ohio River in 1881, before a dam finally erased its habitat in the 1920s. The third was the Leucodendron grandiflorum, the Weinberg conebush. It was last seen in London in a collector's garden in 1806, its habitat on Weinberg Hill in the shadow of Table Mountain, Cape Town, already lost to colonial vineyards. But this flower may prove to be completely lost. The project is bringing to light that Harvard specimen, and possibly specimens in other collections, may all be incorrectly labelled. An old feud between 2 19th century botanists exposed again. Back at Ginkgo Bioworks, the scientists could begin working with the DNA once it had been extracted from the plant tissue and sequenced by paleogeneticists at the University of California at Santa Cruz. The Ginkgo team identified gene sequences that might encode fragrance-producing enzymes. These sequences of DNA were printed and then they were inserted into yeast, which were cultured to produce the smell molecules. The identity of those molecules was then verified using mass spectrometry, giving a list of the smell molecules that each flower may have produced. Those lists were sent to Berlin to the smell researcher and artist Sissel Tollas. Sissel began to reconstruct each flower's smell in her lab, using the same molecules from her collection or finding comparative ones to approximate those that aren't available. We can use technology to reach back into the past, giving us a glimpse of each flower, but we will never know their exact smell. Science can tell us which molecules they made, but the amounts of each are also lost. Building on that contingency, in London, the artist Alexandra Daisy Ginsberg designed a series of immersive experiences to reconnect the lost flowers to their habitats. These draw on the idea of the sublime, the sensation of the unknowable, and exposure to nature's immensity that makes us consider our position in it. Using genetic engineering so we can once again experience a nature that we have destroyed is both romantic and perhaps terrifying. It is sublime. Artists tried to express this aesthetic state in 19th century landscape paintings, but like these images, even the most advanced technology can only give an incomplete representation. In a natural history museum, nature's contingency is trapped in time. The clock of creation and destruction stopped for us to look at. In the installations, each landscape is similarly reduced to its geology and the flower's smell. The human connects the two, and by stepping into this abstract nature, they become the specimen on view. The smells are diffused in fragments and mix in each installation, so every inhalation is slightly different. We can never fully experience the flower in the present without its past context. This is not de-extinction, but a technological sublime, allowing us a glimpse of a lost flower blooming on a hill, on a wild river bank, or on a volcanic slope, the interplay of a species and a place that no longer exist. As the philosopher Jean-Luc Nancy says, the sublime is not so much what we're going back to, as where we're coming from.
This is Martha and George, who are soon to be mother and father. They made the choice to fulfill their pregnancy with parturience. Parturience, which provides artificial wombs for anyone for the period of 9 months. This is the parturience pod. The baby is nurtured through this object. It is similar to a pregnancy, but without the physical restrictions. Throughout this period, the family can live and work as they would normally do, whilst also being able to bond with their child. George Aylward, 29, is a journalist who is engaged with several interesting projects, due 2051. This is a communication device, which is connected with the parturience pod. The device can record desired moments of the daily life of the family. With its integrated microphone, it can record music or any other sounds desired. This is how the baby gets used to the voices of its future parents. Martha Aylward, 30, CEO of a company she started, works to ensure that the company meets their goals. It is almost time to go home. This is a portable communication device used when not at home. In between her meetings and lunch breaks, she can communicate with the baby. By simply switching it on, you can feel the baby kicking and moving. Carefully touching and rocking the device will send movements to the pod, which the baby can also feel. George is setting the table for dinner. There's Martha, home from work. George has chosen a wine to go with their dinner, and that's all right because Martha isn't carrying a child. Martha is cutting some ingredients for a special recipe that will set the preference of taste for their future child. The most important part of the growth process is the nutrition intake and the future preference of taste for the baby. The pod keeps track on what the baby has eaten and what it needs to stay healthy. After the food is prepared, it is placed in a capsule and placed in the parturient's pod for the baby. At night, the pod goes through the updates made by the hospital with the information obtained during the day. This ensures the baby is healthy and the pod's program works without errors. The day is almost there. This is the final moment in which the pod's lock is lifted and the baby finally meets the family. George, Martha, and newly born Robin, one big happy family. Look how happy they are.
いつか出会い、愛し合う二人は運命の赤い糸で結ばれている。そんな神話のような糸を紡ぐ蚕が2016年この世に誕生した。アートと科学の創造で新しい神話が生まれる時代、始まったのかもしれない。をこの卵に注射すれば恋に落ちる運命の赤い糸生み出せるはず。蚕の目が光れば成功のサインなんだけど。私ももう大人だし、オキシトシンくらいで恋が叶うなんて、恋はそんな簡単な話じゃないだなんて、そりゃ分かってるんですけど、でもそんなことやってみないと分かんないじゃないですか。失敗は成功のもとだって施術教授も言ってたじゃないですか。 だから私作っちゃったんです。ねえ、新しい神話一緒に作ろうよ。そうしそう愛ないとは言わないで高鳴る野望は止まんない。ゴールドだなんても言わないで運命なんて結果オーライ。
Live from New York, where the world's first Cas9o opened earlier today despite ongoing protests. The public backlash first started in 2051 when the Universal Genetic Screening Law was passed, requiring preventable genetic disorders to be removed from embryos in order for them to qualify for universal free healthcare. Since then, scientists and doctors have been inching closer to creating fully designed humans, what the public refers to as designer babies. The law states that parents are not allowed to handpick desired genes for their offspring, but Cas9o puts chance back into the equation. We spoke earlier to Cas9o's first family unit to hear a little bit more about how it works.
All of our genes are in our 23 pairs of chromosomes. At the Cas9o, our chromosomes will be copied and stored in the gene bank so they can later be recombined and synthesized to form the genome of a new baby. We play around with bébé roulé for each chromosome. The wheel ensures a randomized selection, but players can increase their odds by betting extra chips.
And what if you don't like a certain gene on a chromosome assigned to your baby?
CRISPR tokens, just CRISPR them right out.
This is Brianna, one of our parents playing at the Cas9o tonight.
I'm Brianna, daughter of the president and CEO of the gene bank in the Cas9o. I'm paying first to have this baby, and I feel so blessed that the government has given us access to this sort of technology. It's a really great option for people like me. Yes, it's expensive, not everyone can afford it, but what I can afford is to have a baby that's just average.
I feel lucky to be chosen to be part of this group. I grew up before humans were screened for genetic diseases, and I've had really crippling medical bills because of it, so I know I can't afford my own baby, but Brianna hired me as the primary caretaker and genetic parent of our baby because I have blue eyes, so I guess I got lucky after all.
This is truly an amazing step in the scientific community. Genetic regulations have hindered mankind to push the limits of the human race, but Cas9os are a step in the right direction. Why should we keep settling for natural selection when we can select whatever we want?
Now let's check in to see how the game is going.
Alrighty, chromosome 9. Chromosome 9 may determine your child's eyebrow thickness, forehead freckles, lip width, blood type, and vulnerability to malignant melanoma. Place your bets.
Okay, the girls are placing their bets. Looks like Brianna's got a lot of chips on the board. Aaron has next to nothing. Now they're spinning the wheel. And Molly wins!
Oh no, not those eyebrows on my baby. CRISPR those out right now.
What? Are you really going to risk editing my chromosome? What if it messes something up? It doesn't work every time, you know.
You could replace your eyebrows with mine.
Oh, please.
How dare you?
Whatever. I still won the chromosome, and I choose Aaron's chromosome number 9 to complete the pair. Next, we'll play for chromosome 17.
Looks like the karyotype is shaping up. We'll check back in with them a little later. Now let's hit the streets to hear from some of the protesters outside the Cas9. Protesters are gathering to voice their outrage towards what some are calling the next eugenics movement.
This Cas9 needs to be shut down now before we raise an entire generation of elitist superhumans. Our government should promote equality, but this government-run Cas9 is promoting rampant inequality.
Some say they fear being further stripped of their reproductive rights. Others are comparing this to the 2020 reversal of Roe v. Wade.
Cas9 are mining all of our genetic information and selling it to the highest bidder. They say they want to maintain diversity, make sure our kids are healthy, but they just want to control procreation. They want to decide who gets to be a human and who doesn't.
Cas9 are promoting eugenics. They don't want my imperfections. It's ridiculous. It's ridiculous.
A cell is a history. A cell is a home. A cell is a whole. A cell is a cage. A cell is an electric fence, enclosing your legible unknown. To break the cell is to trespass the most intimate of spaces. Dear donor T2305, I received 5 milliliters of your saliva in June. It wasn't hard to get. Using my academic email and mailing address, I simply said it was for research, genetic testing, the truth. When it arrived, I moved it quickly to the freezer to keep it fresh and alive. I sent 2 milliliters of your fluid for sequencing, and then I waited and waited and waited. 6 weeks came and went, and then another time over. The company was experiencing unusually long processing times. When the results finally trickled in 6 months later, I devoured your profile in a single sitting. You are 46 years old with dark brown eyes, a full head of brown hair, and few freckles. You toss and turn in bed, and you enjoy a savory midnight snack. You like to run, for miles, for hours, and this keeps you slim, keeps your heart beating strong, and helps tame your sometimes wild thoughts. You were my first, and I think of you frequently. I imagine your face, your voice, the way you walk. I'm curious where you grew up. I worry for your health. And I think of me poring over your genetic details. I'd like to think you would be flattered, but you might be very angry. Dear donor, on Monday afternoon, I immortalized your cells. Centrifuged and gently washed, I infected you with the simian virus SV40 to induce replication. Suspended in a solution of amino acids, vitamins, sugars, and held at the temperature of my body, you were beginning to grow. On the microscope's platform, I studied your shape. Transfected with the gene from a jellyfish, you glowed green with fluorescence, and I watched you divide. Your cells want to grow, and I passage them to feel you. Dear donor, on Thursday I uploaded you. There were 3 close matches, but one was perfect. 67 STRs, a constellation of your Y chromosome in precise alignment. Michael Daniels, you were born on April 12, 1970, in St. Louis, Missouri. Michael Daniels, you are a brother, a cousin, a distant relative, or possibly something much closer. Michael Daniels, I suspect it might be much closer. Dear donor, Michael Daniels emailed me. We're a genetic match, he wrote. Michael, what would you say if you knew? I want to write to you, but I'm so scared. Have I gone too far? T, I wrote to Michael and I told him the truth. I asked if he wanted to meet, and I asked if I could tell him everything. Dear Michael, I passaged your cells 12 times and they're still growing strong. You haven't written back to me. I think of you constantly. You like cars, you have a good sense of humor, you live in St. Louis. I shouldn't be surprised, but when I see you might live just blocks from the saliva lab, I am. My letter must have scared you, but I know in person I wouldn't. If you saw how much you mean to me, how important I think you are, I know you would understand. Michael, I'm coming to St. Louis.
Turn left onto Metro Court, then the destination is on your right.
I need to know what it is to give myself like that.
Arrived.
I want to see what you saw and sit where you sat. I want to feel it. I want to feel you. Dearest, I went to the clinic and I signed away my cells. I sat on the hard beige chair with the plastic tube strapped into my mouth. I leaned back and closed my eyes and I thought of you and I let myself drool. And 20 minutes later I'd filled the 5-milliliter jar. I took the tube out of my mouth and walked out. And now, I'm donor T3511. Sometimes I feel so close to you, but then you seem totally out of reach. Michael, your secrets are safe with me.
Hello, my name is Tom Knight. Along with my friend Al Gore, we invented the Internet. Today, I no longer work with boring computers. I work with deoxyribonucleic acid. And I'm here to welcome you to Ginkgo Bioworks.
We have only the finest DNA at Ginkgo Bioworks.
Not enough tusks. We present the very finest in synthetic biology technology available today.
At Ginkgo Bioworks, safety is our highest priority. We take containment very seriously. We're looking for the best men, women, and androids to join our team at Ginkgo Bioworks.
Since coming to work at Ginkgo, I find one of the great things is that my productivity is increased by up to 50% at any given time.
At Ginkgo Bioworks, tomorrow's kitchen is the lab. Let's see what's cooking at Ginkgo today.
Who is the bioengineer of the future? It could be you.
In a world where plastic rules all, 90% of toys are made of some sort of unrecyclable plastic.
Our brave biomaterial rebels, Fungus and Scooby, have come to save our planet. Together, they will battle the evil forces of plastic. But wait, they can't do it alone.
Join their mission of ridding the world of plastic, one BioBuddies kit at a time.
We're the BioBuddies!
Our mission is to provide you with eco-friendly toy kits to explore, play, grow, and create with biomaterials. As the world is becoming more aware of environmental sustainability, it's time to move away from plastic. This is where biomaterial design comes in to save the day. Our biomaterial growing kit incorporates science, nature, and art to make biodesign more approachable. We've designed two biomaterial growing kits, Scooby and Fungus. Scooby is a kombucha growing kit, and Fungus is a mycelium kit.
Each kit includes the necessary supplies to grow the biomaterial and explore with it.
The average toy fad lasts 8 months. Our goal is to create a sustainable kit that can be played with, explored, and developed to sustain interest over a long period of time. The entire exploration journey cycle is accessible, educational, and fun.
We tap into the waste stream by feeding our cultures with used coffee grounds, sugary drinks, and tea leaves. First, users grow their biomaterials.
Then they can play and explore through personalized projects with their materials. Of course, not all projects will succeed, so we encourage users to celebrate their fails.
In the future, we hope that we can sell our kits online or even in retail stores like Target to people of all ages. As our kits don't fit into a specific category, we think that they would best fit into the craft or miscellaneous kit aisle.
Help us save the earth by redesigning the toy industry.
BioBuddies out!
Bees are our primary pollinators and they're in trouble. But they're not the only pollinators. Flies already do about 30% of all pollination, and much more in cities. Alumbria encourages flies to pollinate much more efficiently by attracting them with natural chemical signalling. Using a system of nodes controlled by an interface, we encourage the flies to move from one node to another and another, just like a relay race, giving the bees a break and ensuring the fruit harvest for years to come. Now that's pretty fly, if you ask me.
엄마죠.
엄마들이 요리를 잘하면 딸들도 요리 잘하거든. 그래서 우리 외할머니가 요리를 진짜 잘하셨고 우리 이모도 요리를 진짜 잘하셨어요.
왼손 장갑 빨래 빨 때나 이런 설거지를 할 때는 쓰고 안 쓰고.
엄마의 그런 따뜻함을 먼저 떠올리는 것 같아요. 엄마가 이렇게 만들어줬다는 그런 느낌? 엄마가 나를 이제 신경을 써준다, 엄마가 나를 사랑한다 그런 느낌이 제일 많이 들어요.
글쎄, 아니 타고났겠죠, 손맛은. 자기가 뭐 태어날 때부터, 그죠? 자기가 좋은 손맛을 가지고 있다는 거는 좋은 재산을 가지고 있는 거랑 마찬가지잖아요.
Change happens when action replaces inertia, when the status quo is shaken up. But you already knew that. You are the risk takers, the inventors, the change makers. You don't run from change. You put it at the top of your to-do list. You say to the world, we can do better and make it happen. You exist to change the world. We exist to help you reach it. Together, we can turn vision into impact. Mission North is a communications agency for companies shaping our future.
Hi there. Welcome to the SBA BioArt Lab, where we've been designing the future of food for over 30 years. Good. This laboratory, the first of its kind, is where biologists and artists work together tackling some of the world's biggest biological concerns. Over the years, we've spawned some unique results, but nothing as successful, impactful, and delicious as this. Meet the Mayo tomato, the amazing protein-packed fruit that started it all. A biological breakthrough that changed the way we think about protein. I have something amazing to show you. Randy, get the thing.
As you wish.
Before the Mayo tomato, humans relied heavily on animals for their daily source of protein. With a growing global population and a strained agricultural system, we recognized animal protein was not an ecologically sustainable source. There had to be an alternative method. We put together some of the brightest scientific and artistic minds we could find, and the result was impressive. Take a look.
This will only hurt a little bit. JK.
In 2016, the BioArt team at the School of Visual Arts engineered the world's first fruit to produce the same protein, myoglobin, found in animals, the Mayo tomato. They found plants to be deficient in megadoses of protein necessary for a nutritional human diet. So they translated a DNA sequence found in beef and inserted it into a tomato's genome. The result? One little tomato possessed the same amount of protein found in an 8-ounce steak. This breakthrough creation caused quite the stir. It seems not everyone was a fan of this amazing scientific achievement.
GMOs are dumb and stupid and poison.
Of course we're not happy.
However, in the year 2030, mad cow disease almost completely wiped out the cattle industry, creating an urgent need for the Mayo tomato's amazing protein power. And soon, hearts, minds, and mouths were open. The Mayo tomato ushered in a global movement that ultimately ended our need for animal consumption. Not bad for a group of art students, if you ask me.
Hello, everyone. Welcome back to the afternoon session of Biodesign Challenge Summit 2020, Day 2. I'm Alex Kizilewski. I'm BDC's Communications Manager. Very excited to be emceeing this afternoon with all of our wonderful students and judges and all of our viewers at home. So a couple of reminders and logistical items to clear up just in case you didn't join us this morning. We're going to be playing all of our student presentation videos. So we've asked each team to make a video about their project. We'll be playing those. We'll have the students with us join us in the broadcast. And then we'll also have our judges pop up and ask some questions of the students. We also have a couple of community resources that I wanted to remind everybody about. Those are located on our summit page if you're watching from our BDC website. That includes a Miro, which is an online community board where you can go and share resources, write what biodesign means to you. We'll use it as you wish. We curate it for the community. I also wanted to remind everybody that this year we are sponsoring a community choice prize. So if you're watching, hopefully, you will also vote in our community choice prize if you want to. The voting ballot is also located on our summit page, and we ask that you please vote by Thursday the 18th at 12 p.m. Eastern Time. So without further ado, we are going to start off with our first student video, which is going to be the University of Michigan. Take it away.
Hi, my name's Jenna. I am currently a student at the University of Michigan, and I am pursuing a dual degree between art and biology.
Hi, my name is Roy, and I'm a senior at the University of Michigan, getting my bachelor's in fine arts.
Hi, I'm Ellie Ward.
My name is Alexandria Pompey.
And I'm Cassie Hoisington.
The death toll is already more than double the number of reported deaths of any other nation.
It just feels like you're standing on the shore looking at a tsunami, and the federal government is offering you a life preserver.
Tonight, the coronavirus devastating toll on Black and brown Americans. Preparation, public health, and political leadership. Because all experts agree this is going to get worse.
So in just a matter of months, the world has changed forever. COVID-19 has uprooted our lives in unprecedented ways as government institutions fail to take adequate action to prepare. While eventually the pandemic will pass and a sense of normalcy will return, this global crisis serves as a warning for the next global crisis looming in the near distance: climate change. While COVID-19 and climate change seem like unrelated issues, they are alarmingly similar. Just like for this novel coronavirus, our institutions are choosing not to take action and therefore setting us up for failure. But unlike COVID-19, climate change will not just come and go. Climate change will persist and affect every single aspect of the world in ways we can only begin to imagine. Our project Kabloom seeks solutions for how climate change will affect our home state Michigan while also addressing the broader underlying cause of climate change, humanity's relationship to nature. We explore and challenge the cultural norms of how humans have curated private outdoor spaces and how accessible biotechnology can be used to engage citizens to pursue change in humanity's place in the living world. Through reimagining the modern lawn and centralizing tools and resources, we aim to make it possible to work towards a speculative future of a world without lawns.
One of the major questions of our project is, what does the future that we're pursuing actually look like? What we're interested in is a future that moves past the grass lawn entirely in favor of native plant lawns for all areas. This is the kind of biodesign that keeps both parties in mind. We've seen plenty of examples of biodesign that take inspiration from the natural world and benefit humans, but an ideal form of biodesign presents benefits for the environment along with for ourselves. In a time when climate change is only increasing in impact on the world around us, we can no longer afford to think only for ourselves. Climate change is already showing heavy impacts on the world around us, but for Michigan and other states in the Midwest, one of the focal issues it presents is drastic changes in weather. While this might seem tame in comparison to things like wildfires on the West Coast or hurricanes in the tropics, weather patterns like increased rainfall present unique challenges to our state. Michigan's heaviest rainstorms are already dropping over 30% more water than they did just 50 years ago. This kind of downpour means flooding, erosion, and runoff, all of which are a great method for pollutants to reach our waterways. Michigan's natural pride and joy is our Great Lakes, and our increased rainfall is putting those directly at risk. On top of the climate's problematic patterns, we have our own. The average American family uses 50 gallons of water per day on lawn maintenance in the spring and summer. Meanwhile, we're surrounded by native environments full of plants that thrive on nothing more than rainwater. Not only would a native plant lawn eliminate all of this water waste, it would also get rid of any need for supplemental fertilizers. These fertilizers, along with other waste, can present a chemical risk to ecosystems around our homes. The increased runoff that I mentioned before will only help facilitate this risk. On top of that, the grasslands that we're used to are naturally short-rooted and have much less defense against this kind of runoff and erosion than more deep-rooted native plants. A native plant lawn not only stops contributing to climate change as an issue, but also helps combat the side effects that we've already caused. The movement from traditional grass lawns to native plants can seem like a huge transition, even a sacrifice, but the amount that we receive from nature will always outweigh anything that we're giving up. Native plant lawns will attract wildlife from the areas around us, giving us exposure to the natural world that we aren't currently used to. Additionally, many native plants will help feed beautiful insects like butterflies and pollinators in the area such as bees, encouraging the growth of not only flowering plants but also many natural fruits. This is the kind of exposure that will provide a home that does not pick and choose from the natural world around it, but embraces and appreciates that world.
In order to work towards our speculative future of a world without lawns, we employ the use of plant bombs as a way to effectively transform our spaces. Plant bombs are small balls of clay, compost, and seeds. They are an ancient Japanese technique known as suchi dango, which means earth dumpling. They were repopularized by Masanobu Fukuoka and used in guerrilla gardening activism. Fukuoka emphasized natural farming techniques, and his designs for seed balls meant that users wouldn't have to use farming tools to plant seeds effectively, and the plants that grow do so without much human interference. As he says, we are receiving that which nature decides to give us. Based off this knowledge and history of green activism, we designed plant bombs and the resulting speculative native lawns with flora that are native to Michigan. In doing so, we use the plant bomb as a simple biotechnology and consider how the plant bomb as an object and lawns as a concept fit into preconceived narratives of biodesign. Many proposed biotechnologies appear to respond to biodesign by making new sustainable technologies that can manage our current ways of life despite climate change. These solutions fall short on invoking real change because they do not challenge society's relationship with nature as the root cause of ecological issues. While biodesign presents the opportunity to find new, creative, and unique solutions to prevent climate change or deal with its consequences, it can also continue to foster the exploitative relationship we have with the Earth. The successful outcomes of biodesign depend on us using it as not just a way to find new solutions so that our way of life can be maintained despite climate change, but rather, biodesign must seek to challenge and change our relationship with nature so that it is symbiotic and cooperative rather than exploitative and abusive.
The use of simple biotechnology to push forward our speculative future of a world without lawns makes engagement with Michigan-focused climate change solutions accessible and easy. Seed bombs take 10 minutes to make, use 4 ingredients, and don't require any special tools. Many of the resources needed to make these seed bombs can be found at home or purchased cheaply at local stores. We use our website as a place to centralize resources and provide information, rather than marketing our seed bomb as a product, because it is important for anyone at any income level, ability, or age to have access. A lot of movements can exclude communities without the resources to participate in them, but we wanted this to be something that anyone could participate in. The DIY component that encourages individuals to transform private spaces, such as lawns, enables the public to take direct action to change our relationship with nature. While we acknowledge that the design and implementation of a more sophisticated storm drains and water filtration system could be a solution to collecting water runoff from rainstorms in Michigan, the government has not proved very successful in combating climate change, so we need practical solutions individuals can take part in.
Amidst the current health crisis across the globe, we have seen the extensive influence of individual action. Through self-quarantine, we have been able to slow the rate of spreading infection and temper its progress. This lack of human activity has simultaneously displayed multiple benefits for the environment around us. An unexpected and unfortunate global scenario such as this has slowly become a perfect example of how we, as individuals, hold the greatest power in solving problems on a global scale. While coronavirus is serving as an excellent lens through which we can see our combined strength, there will be a time when this pandemic is behind us. Outside of alarming situations like this, it becomes all too easy to forget what we have the power to do with our daily actions. Almost certainly, the next global crisis is climate change, and we have the power to make a difference towards it. To envision a world without lawns is to witness a world where we come together to make a permanent revision in our lifestyle that not only benefits our health, but our planet as well.
All right, thank you so much. Awesome. So, judges, if you have a question for the University of Michigan, you can turn on your video and we can get started. Heather, you're up first.
Great. Thank you so much. I really enjoyed your presentation, and especially the obvious research and thoughtfulness that you put into it. My one question, though, for you is how effective would seed bombs or plant bombs, as you're describing them, be in reshaping lawns? Like, is this the appropriate technology for the goals that you're trying to accomplish?
I can talk about that. So it might not be the best at initially just like converting like straight grass to lawn, but there are ways that you can like kill the grass first, which would be like putting cardboard over it or like tarp over it or stopping watering it, and then you can use the seed bombs. But I think for taking over it, we haven't actually seen that work. We currently have some that are planted and we're waiting for them to continue to grow more and see what happens. But those are late bloomers in the end of the summer.
Great, thank you.
Yes, I think I saw Anne first, and then we'll move to Jeannie.
Yeah. So, hi, everyone. Yes, and thanks for that great presentation. I guess my question to you was that as sort of a green guerrilla, action seems the most sort of obvious, potent use as you're trying to upend the narratives of how technology needs to be more sort of co-sympathetic with the work that you're doing and the technologies you're doing. I think the word you used was non-exploitative. So I was wondering if you had a plan for disseminating this as an action so that it can be more effective towards the larger goals that you stated, to either bring on awareness of climate change or to create, in fact, a no lawns environment.
I can talk about this, I think. Yeah, so I think that kind of ties in as well to why we chose the plant bomb as an instrument. Right now, our focus is kind of on grass lawns versus native plant lawns and kind of making that shift. But part of our interest in having these plant bombs as our tool is the idea that they could be used in a more sort of like guerrilla gardening kind of a way, in which not only could they be planted for personal use on your own property, but maybe they could be used in a more like active way by throwing them in areas that you think could be repopulated in this way. Just kind of putting the power into the people's hands and letting them question what these decisions are.
I'd also like to add to this, we have several different social media platforms set up and hashtags that we're planning to use in order to get more people involved so that they can use the plant bombs as they see fit, and find a way to connect on social media to make this a larger movement.
Jean, do you want to?
Sure.
Next?
Sure. Great presentation. So why do people like lawns? Why do people have lawns? Why do we like them? And because I think it ties into, for me, I was a little bit on the fence on your presentation. I didn't know if you were doing like a Molotov cocktail or if you were doing sort of like a seed program. So did you talk to anybody about their lawns?
I can talk a bit about this. In our project, we did a lot of research on kind of the history of lawns and how it goes back to having like your own little palace, and how a lawn is kind of a way to like simulate like a castle that was they're like popular in Europe and they're brought over to the US, and it really boomed like during conformity in the 1950s and is really just a big signal of social status. So I think a part of our project is just kind of questioning how social status like impacts the environment around us and why that's important. We didn't talk to like many people about this, but I talked to my family a lot about this, and they really love their lawns. And I think it really just goes back to like how they're perceived by people around them and how culture enforces us to have lawns and habitats around us that aren't exactly healthy.
Okay. I think to throw a ball of seeds onto turf is going to be hard for that ball of seeds to take root. So if you're looking at something to be a little bit more of an action based, I think I would encourage you guys to think about the delivery system a little bit, even though there's something really salient about making a ball with mud and seeds. If you really want to like throw it on the golf course of, you know, our illustrious leadership, or, you know, do a guerrilla act, I think you need to actually get into the works, the physical works of planting seeds and turf and that kind of thing. So that's just a suggestion. Okay, and now I'm gonna mute.
Let's go to Andy, and then we'll have to end with Fred, just in the interest of time. So, Andy, take it away.
Well, I had a couple questions, but I'll just stick to one or two. So just to piggyback off of what Jean was saying, what about yard space that people feel like they want? Like, how do you protect people who want to have picnics in their front yard because it's the only time that they can get outside during this pandemic, or play soccer with people, or just use their yard that we've kind of gotten used to the green grass on? How do you accommodate for people to be able to do those outdoor activities that Americans are really used to on a yard?
I can talk about this. So there are several different types of shrubs and grasses that are native to Michigan, and some of them don't grow very high. And in fact, if you walk on them, they can be shorter and can allow for different types of activity. It just really depends on what you choose. And in fact, we provide on our website a link to all of the different kinds of native plants, and it talks about height and where they can grow and what they can be used for. So there are some that will support different types of activity, not just grass.
Fred, do you want to ask your question?
Yeah, I was just wondering, you show that you made these balls, but I was just wondering if you took them outside and what happened if you did.
Yeah, so I'm the one that made them. I made them with my housemates, and within a few days, they already started sprouting, and I took them outside, and I've been checking on them. Some of them, the squirrels did get to a few of them, but for the most part, they are still there, and we're just waiting on to see what happens. A lot of these plants don't bloom till later in the summer and need more water for that to happen. So we're just waiting and seeing what grows out of it. Yeah.
All right. That is all the time we have. Thank you so much, University of Michigan. Awesome, awesome project. Thanks. We are going to move on to the University of Southern California. You guys can pop off.
As students from Los Angeles, we are intimately acquainted with freeway systems. We are conscious of our current dependence on these systems and their health and environmental impacts. However, we are also optimistic for the possibility of these spaces to be transformed into multi-benefit infrastructures.
The United States has the largest road network in the world. Highways account for nearly 50,000 miles of paved linear space, with huge potential for additional noise barriers. Across the U.S., 8 billion cars use these roads every day, contributing to about a quarter of our greenhouse gas emissions. Highways generate noise levels just below those that cause hearing damage, yet significant enough to have lasting impacts on mental and physical health, including heart disease and childhood asthma. Unfortunately, these impacts disproportionately harm lower socioeconomic populations, who are more likely to be located near high-frequency roads. While these problems are not new, they represent ongoing injustices within the structure of our cities and represent a lack of urgency for integrated abatement. By studying symbiotic species relationships as a means of intervention, our project aims to create an approachable solution to immediately mitigate these injustices, and to begin to generate new visual representations for imagining the future of our built environments. The project began by researching the existing symbiotic relationship between the Elysia chlorotica species of sea slug and the Vaucheria litoria species of algae. Analysis to understand the success of the species relationship included the biological systems, the functional processes, and the conceptual relationship. The research into the species environment identified shallow water ecosystems of coastal regions as the basis for study. This included a variety of salinity ranges from salt marshes to inland freshwater sources. The key factors of all these environments is access to sunlight, carbon dioxide, and additional nutrients for algae photosynthesis. Situated in these environments, the slug can access and extract algae DNA to incorporate into its own DNA structure via horizontal DNA transfer. This allows the slug to also extract and store algae chloroplasts within its diverticula, triggering metamorphosis. The sea slug is a chimera, able to integrate species from both Animalia and Plantae kingdoms for mutual benefit. The animal, the sea slug, benefits by taking on the characteristics of the plant, becoming plant-like both in form and in function, allowing the slug to be photosynthetic to produce its own fuel. The plant, algae, benefits by evolving to survive within the Animalia kingdom. The highway, as a design context, demonstrates interesting parallels to the environment of our symbiont species due to the correlation between the systems. These linear open spaces are abundant with sunlight, carbon dioxide emissions, and potential water runoff. Could the highway system become chimeric and act like the slug by promoting the growth of other species within its bounds? Through collaboration over time, could it create a more ideal environment by serving greater functions, including pollution filtration, noise reduction, fuel generation, and ultimately address socioeconomic inequities? In our search to find a parallel symbiont species with non-aquatic applications suitable for the highway context, our project identified lichens. Where the sea slug become both animal and plant, lichen incorporate algae to become both fungi and plant. Lichens are resistant to desiccation, with water presence activating photosynthesis. Lichen varieties range in terms of resilience to pollution, making them indicators for health and success of the overall system. Based on our research, we began earlier variations for algae-based models, including algae tiles, fognet systems, and storage systems. However, each of these systems were too narrowly focused, required intensive engineering, ignored important socioeconomic conditions, and depended on water sources incompatible with the highway system. Our intervention utilizes the same components as the lichen systems. The layers of environmental protection, photosynthetic growth, and structural support are translated into our proposed project, the acoustic tile. These components become the protection structure, living system, and support structure of our living prototype. The integrated approach provides a nourishing habitat for symbiont species within the context of the freeway infrastructure, resulting in the overall well-being of adjacent communities. The base material of the tile is clay, selected for its natural, sustainable, and accessible qualities. Clay's microporous structure retains water and exhibits pollution filtration characteristics. It can be 3D printed or handcrafted, allowing for the code or model to be open-sourced. The tile form encompasses various elements, while the overall wall meets U.S. transportation requirements to be non-distracting. Primarily, the form mimics the surface structure treatment of the Namib desert beetle, seen in the tile's grooves and spherical shapes acting as hydrophilic and hydrophobic surfaces that strategically move water. These formal attributes create variation for shade and sun exposure, producing a range of conditions for fungal and plant growth. Smaller scale grooving resulting from tooling marks on 3D printed tiles or natural variation on handmade tiles create micropockets which attract water and variegated surfaces to encourage root adherence. The living components colonize the tile in stages, altering the microclimate of the vertical surface, sequestering pollution, and creating acoustic absorption over time. Algae plays an important role in the initial mitigation of pollutants, including sequestering carbon dioxide. This also involves blue-green algae, a nitrogen-fixing cyanobacteria, which additionally provides algae sources for lichen structures. Crustus lichen, the most pollutant-resistant lichen species, would colonize the tile and alter the microecology. Then lichen varieties of Fruticus and Folios could become established, providing additional pollution mitigation and enhancing the tile performance. As biomass accumulates, it would perform as an acoustic barrier for high-frequency sounds. Lower-frequency noise would be absorbed by a small air cavity at the back of the tile, following the Helmholtz resonance principle. The utilization of the low-cost hyper-services sensors embedded within each tile integrates technology into the living system to better monitor the health of both the individual tile as well as the overall system. The data would determine whether the tile remains on the freeway, is to be harvested for its acoustic or biofuel properties, or requires replacement from damage. The project was designed as a low-impact passive system, creating a variety of spaces for water retention and sunlight variation. However, for more difficult living conditions, future modules are being studied, including methods to collect and store water runoff within the cavity of the tile. Acoustic tile asserts the need for biological thinking by welcoming natural systems within our man-made infrastructural networks. It asks whether these systems can coexist and collaborate towards healthier and more equitable futures. On a practical level, the project aims to be a low-cost and easy-to-implement system to provide relief for environmental and noise pollution. Yet it also confronts the socioeconomic inequities constructed within our urban fabric. We are not naive to suggest this project is a solution to these injustices. Rather, it begins the active process of healing by providing a method for interventions to occur at a government or grassroots level. The expanses of living walls alongside the very veins associated with progress bring awareness to a different potential where biological and technological systems are not set in opposition, but can coexist for mutual benefit. Even in a future scenario of improved technologies and reduced dependence on cars, the system creates a lasting mutuality between our infrastructure, our biophysical world, and our most vulnerable communities.
All right. Thank you so much, University of Southern California. Welcome to the Judges Q&A. Judges, if you have questions, you can turn on your videos. Andy.
I hate to pop up too often on here, but one of the questions that came into my mind right away is that there are a lot of walls that do provide protection and sound for neighborhoods against these. What kind of lichens currently already grow and exist on those? Are these something that we could immediately start expanding their population somehow?
That's a great question. We haven't gotten into that exactly yet, but I can say that like in Southern California, to some degree, the habitat on those walls isn't cultivated to be able to support a lot of lichen growth. So the idea is that this is an attachment to those walls potentially, if not creating an entire new wall, and that it can begin to facilitate that growth. But I definitely think that that's something that in other types of regions should be looked into greater, especially more coastal areas.
Thank you. Fred, over to you.
Yeah, okay. I really love the detailed biology that you folks got into. It was very clear that you did your research. And indeed, most lichens are incredibly sensitive to pollution, but you even went to the extent of finding the ones that would stand out as being resistant, which is something that takes a little work, I'm sure. And also your descriptions of the biology were very interesting. I am curious as about, you wanted symbiosis. Well, I think that's a great thing to have, but why a symbiotic situation? Couldn't you do something just as useful without being symbiotic?
That's certainly, I think it was the premise for our class was understanding how symbiosis creates a mutual benefit between two different types of symptoms. And I think that the idea of having a project where that is implemented on such a visible level helps to correct the current mindset and understand that we are very much interconnected with our environment and what that means and how our environment reacts to us and how we can react to it. So I think that on a conceptual level, it covers that. But I think that we were also just interested in understanding how these things can begin to support each other. And instead of just always having a input-output type of system, it's a little bit more of a circular benefit.
Thank you.
Any other judges? Questions for USC? Yes, Nina.
Hey there, great job. Also seeing the prototypes, they were really beautiful. I mean, I would love a piece of that in my home or something like that. They were really stunning. So great job on that. As you know, infrastructure-related projects are not something that our government seems to excel at in the recent times. I'm curious if you had taken any time to think about the implementation of this. What sort of incentives can we create to inspire and motivate the types of partners that would be needed to make something like this come to life?
That's a really good question. I think that was something that we were just beginning to look into. I think that one of the positive things I see in my generation and maybe the generation below it is this is much more actively on our minds, and understanding how we can begin to implement this is important. Also, the government tends to step in and start to do more infrastructural projects when they want to stimulate the economy or when the economy is good. So maybe there's a future for that on the horizon. But that's definitely something we thought about, which is also why we wanted this project to be something that could be approached not just from the government level, but from a grassroots level. You could hand make these tiles if you wanted to, or access to 3D printers is possible for you. You could do that as well. And then that way it begins to generate a conversation at a much more individual level, and then it can grow to the government level if necessary. But definitely thinking about how to begin to initially implement these programs at a government level is something that we'll be looking into in the future, because it's certainly important to have a system for that.
All right. Thank you so much, Tessa and Kingru. I think that's all we have for today. We are going to have to move along onto our next team, but thank you so much for joining us.
Thank you.
So we are going to hear from New York University IDM next.
Divergent labor risk in a post-COVID society, and a reassessment of one of our most fundamental and intimate senses, our biodesign team has explored the connection between inhalation, the olfactory system, and our well-being. Welcome to ModMask, a biodesign research project that aims to take the latest innovations in bioelectronic olfactory sensors, filtration material, and aerosolized healthcare technology to provide people a more integrated experience with their environment in the form of a mask series. As the normalization of masks as an everyday accessory takes off, our goal was to create a long-term use, recyclable system of modular components to enhance the mask-wearing experience, from an overly clinical and impersonal ordeal to a comfortable and biotechnology-equipped piece of your everyday. The AirPods for your sense of smell. The ModMask wearable technology provides nuanced control over your air, mediating your relationship through 4 main methods of intervention: filter, enhance, treat, and sense. Filtration is, of course, the default mask function we're all familiar with. Designed with 6 layers of protection, including a memory foam nasal support clip, a non-woven comfort layer, high-efficiency electrostatic filter cloth, an activated carbon filter, a filter for particulate matter up to 2.5 microns, and an exterior customizable decorative layer, ModMask has the foundational element more than covered. But what if masks went beyond that? With the Sense functionality, biosensor technology interprets the particulates in your environment, alerting you to any dangerous contaminants that could be harmful to your health. The Enhance component, meanwhile, works by mimicking the human olfactory system through the digitalization of smell. It takes the latest in bioelectronic olfactory microchips to not only interpret your environment, but allow you custom control over scents to eliminate or augment through the regulation of microparticles associated with scent, working in concert with the nasal microbiome. The Treat modular component utilizes the most recent advances in aerosolization motors, down to the size of a small matchbook, to provide treatments in the vein of a nebulizer or the flu mist vaccine. Whether with prescription bronchodilators or corticosteroids, or a focus on wellness via essential oils, this component allows for a wide variety of applications for inhalation treatments. We have built a realistic commercial enterprise with an online store to purchase the mask foundation in various fabric styles, the modular mediation components, and essential oil pods. We've used 3D modeling software to construct more speculative designs incorporating these components with an attention to detail that aims to demonstrate the feasibility of more fashion-forward olfactory manipulation in addition to the standard mask series. This includes a Smell the Roses design meant to focus on the biotechnology of enhancing and creating pleasant smells, as well as a Date nasal clip that you can sync up with another user to provide a mutual sensory experience. By including these trendier, speculative designs into our project, we were able to expand our mask series beyond the traditional style and into a fashionable world of scent manipulation being as commonplace of an experience as listening to a playlist. We've provided customers an engaging try-on experience by applying this 3D modeling through accessible apps on their personal phones. We also provided a little background about us as a company and the science that informs the design of ModMask. Our design process started with various mask concepts that incorporated the biotechnology we were aiming to integrate into daily life. An assessment of the media bombardment of the personal politics of masks, imagining a likely future world in which masks were as common as sunglasses and viewed as a basic system of protection and enhancement, and therefore the likely scenarios in which the mask functionality we were envisioning would be put to use. We continued iterating on our mask designs while formalizing the branding of this hypothetical company, constructing wireframes and user flows for the commercial website. Although our team initially wanted to focus on an exploration of scent and the nasal microbiome, the COVID-19 pandemic put the importance of masks into sharp relief. By marrying the newfound relevance and necessity of masks with the exploration of scent in personal experience, autobiographical memory, and emotions, this Biodesign challenge took on new meaning for all of us. The Biodesign challenge themes were vital in our decision-making process throughout design iterations. Our research highlights the feasibility of this project by incorporating existing filtration, bioelectronic nose, and aerosolization technology. Everything we have digitally modeled is an existing biotechnology, but the combination and careful construction of a functioning prototype would take time. The human impact of creating a mask series was paramount during this time, including the reality of accessibility of a mask series in a capitalistic enterprise that would inherently limit the audience. Our design aims to alleviate this concern via the modular design that allows for customization for those who want it, but focusing on protection for everyone. With recyclable components and a long-term use system, sustainability was at the forefront of our design, and the risk involved in such a product is akin to those for other modular, inhalable technologies, such as manipulation of vape pens. Controlled points of sale can limit this risk, and ultimately, we feel the design serves a positive purpose throughout these 4 main biodesign themes. With the normalization of masks in everyday life, the ModMask design aims to integrate existing and on-the-cusp technology into a sustainable, long-term accessory for the well-being of all people. Take care of your air with ModMask.
Amazing. Thank you so much, NYU IDM. I'm loving your theme, Andrew. All right. Welcome, students. Judges, if you have questions, you can turn on your videos. Anybody? Forming questions? Jean.
I have a question. Hi. How did you come by the 6 layers for your mask? Why did you determine so many layers?
We did a lot of research on the filtration aspect of the mask, obviously that being kind of like the default mask function. We wanted to make sure that we were covering it for the tiniest particles, because we wanted it to be good for filtering out, viruses and things up to really small micron measurements. So this construction was a combination of different kinds of masks out there from the medical field, as well as commercial masks. And all of these layers were to cover everything that we wanted to filter for in terms of organic particulates, as well as viruses and stuff like that.
So did you, at home, were you able to take 6 layers of fabric and kind of put it up to your face and see if you could pull some air through? My concern is it's really, really thick now. And if you look at these N95 masks I have here, well, it's not in the package, but there's a bladder there to kind of allow an air exchange, which you are now putting electronics in. So my problem or my issue with it is that I don't know how the air is going to pass through this thing. So that's just a concern I have. So yeah, that's, just my question and my concern,
Yeah, completely understandable. A lot of the layers, they're not full fabric layers. There's only a couple, what I would even consider full layers. Like the carbon filter and the external decorative layer is obviously customizable. And the side portions where you can screw in some of these modular components, the default mask system doesn't have electronics in it. So it would allow for that kind of bladder functionality that you're describing. With the extra electronics, no, we were not able to prototype something with those electronics. So we would have to definitely see how that would affect that.
Okay. Leslie, please.
Hi, guys. Oh, thanks, Alex. Thanks for the great presentation. Certainly a topic that's meaningful right now. So my question is the opposite of Jean's, and that is with the speculative designs, which are really neat looking. I guess, is the expectation that you no longer need the filtration because it doesn't seem to cover the mouth at all. So can you just speak to the purpose of that a little bit more?
Sure. Iterating on our initial design of a typical face mask, we envision a world perhaps post-COVID in a sense where if a vaccine were developed, mask use would still be commonplace. And perhaps there are other augmentations that we can add in terms of how we filter or engage with the air around us. And so designing an item that is entitled Smell the Roses would in fact maybe provide that ability to kind of smell the roses in a literal sense and also be marketed as perhaps a luxurious item that perhaps only those that are kind of privileged are able to do so.
Got it. And then just to follow up, I guess, since nobody else has a question, I'll ask another one. In terms of the treatment component or of the idea, so have you guys thought through like the steps you would need to actually roll out a product that would be treating an underlying condition? I mean, it seems like there'd be a few steps between here and there. Can you sort of speak to that in terms of regulation?
Sure. The regulation aspect, no doubt, would be a little bit of a minefield with the FDA and everything. However, the main thing we were trying to incorporate with the treatment functionality was the possibility as opposed to injections. It's just heavily on the brain because of COVID. But beyond that, there are aerosolization motors that are very small nowadays, new within the past, 3 to 5 years. And so while we can't totally speak to how onerous of a process it would be to, incorporate literal prescriptions into the mask system, the technology being possible seems like a solid step one in it being a feasible treatment option.
All right. Thank you so much, team. Great project. Very timely. I think that's it for right now. We're going to move on to the next team. Thank you so much for your time, NYU students. Up next, we will be hearing from the University of British Columbia is next.
There are estimated to be 5,000 to 6,000 viruses ready to jump from animals to humans, and an additional 1.5 million unknown viruses out there. The risk of infectious disease is growing as humans increasingly encroach upon the natural environment. These viruses threaten us with the potential of future pandemics, many of which may be more lethal than any we've seen before. We can prevent this.
This is a virus. Inside its shell is the genetic blueprint for making more viruses. It is 1/1,000,000th of an inch long, so small that 500 million of them could fit on the head of a pin. This tiny entity is devastating the world. This virus uses its spike protein to attach to a protein receptor on human cells like a key to a lock, letting the virus enter and infect the cell.
Once inside, it hijacks the cell's processes to make 1,000 new viruses in 10 hours. These are released to infect other cells. 200 years ago, humans invented vaccines to combat viruses by killing them or preventing their entry into cells. As we speak, there is a new cutting-edge technology being developed, the mRNA vaccine. mRNA is a copy of DNA that takes genetic information to be made into proteins. When mRNA is injected as a vaccine, our cells use it to make the protein. Our immune systems learn to recognize the protein as an enemy part, and when the real virus enters our bodies, our defenses will be primed and ready. Moderna, among other biotech companies, is developing the first mRNA vaccine with mRNA that codes for the SARS-CoV-2 viral spike protein, and it has passed initial clinical trials with promising results. Conventional vaccines usually take 10 to 15 years to produce, but mRNA vaccines could be ready in a fraction of the time.
Science, engineering, and design have always drawn inspiration from biology and the natural world. Every organism on Earth is constantly evolving as they compete for resources in an evolutionary arms race. But what if we were able to look into the future and predict these evolutionary changes? Imagine the lives we could have saved if we had predicted what was coming. If we'd had policies already in place, vaccines ready to be distributed, and no need for a global lockdown at all. Just as we built optics inspired by butterfly wings and neural networks inspired by human brains, we can create software inspired by evolution. This is Viper, a biology-informed software tool designed to simulate the evolution of viruses. How is predicting the future possible with Viper? We use software very similar to it on a daily basis. Viper is like the predictive text on our phones. It uses deep learning, a kind of artificial intelligence that mimics the way a human brain learns. We give Viper learning materials to start with. This is the training data. For predictive text, this is all the sentences you write on your phone. For Viper, it's all the genetic sequences that contain the instructions for making the virus spike protein. With every new sentence or genetic sequence it encounters, the program gets better at recognizing emerging patterns in the data. Eventually, we can give it a small fragment of data, like the first couple words of a sentence, or the first pieces of a new spike protein, and it can fill in all the data surrounding it using the same patterns it saw during its training. In short, this is how Viper predicts new spike protein sequences. In addition, we are implementing a statistical model called a Markov model to examine the probability of nucleotides changing. It will complement the predictions from our deep learning simulation and increase the robustness of our tool. After we have our predicted spike proteins, we will test the key-to-lock interaction between the spike and our cell receptor proteins. The stronger the binding is, the easier it is for the virus to enter the cell, allowing us to choose the most likely and the most potentially harmful predicted strains.
So, what can we do with this information? Recall that mRNA vaccines are based off of the viral spike protein sequence. If we can predict this sequence before the virus ever emerges, then we can begin vaccine development in advance. The wait time for a vaccine can be devastating, and this is what the world is seeing right now. And this could be even worse in a pandemic caused by a more lethal virus. Instead of waiting for a high-caliber, pandemic-level virus to strike, we can begin vaccine research way in advance. mRNA vaccine development is estimated to cost between $100 and $200 million, which is really a drop in the ocean compared to the estimated $4.2 trillion lost so far in the United States alone. This is a wake-up call. We need to begin investing more in preventative biomedical technology. Viper is our team's solution. Even in the beginning stages of development, Viper is showing promising results. Based on early viral data, Viper predicted genetic sequences for SARS-CoV-2 spike proteins. These had over 99% similarity with spike sequences that appeared 4 days later in Luxembourg and 2 weeks later in Japan. Here, We compare a predicted spike sequence with the known spike protein from Luxembourg. Each axis represents the sequence position within a spike protein, the horizontal axis for predicted, and the vertical axis for the Luxembourg one. The more similar the two sequences are, the longer the line fragments, and the better it matches a 45-degree line on the plot. As you can see, 3 long fragments of our prediction can be mapped exactly back to the known sequence, meaning our prediction is very similar to an actual spike protein. We then compare the different functional regions of our prediction with the known sequence. Functional sections specific to SARS-CoV-2 are present on our prediction. These results show that Viper was able to predict sequences that emerged after we trained our software. Imagine what we might be able to do as we improve it.
But could Viper's predictions lead to lost resources through development of unnecessary vaccines? Extensive consideration goes into developing each and every vaccine by virologists and policymakers alike to prevent the creation of vaccines that are not needed. And even if a predicted virus never arises, the cost of continued preemptive vaccine production would still be less than the cost of a single pandemic. And what if predictions of possible viruses are manipulated to spread undue fear? We can mitigate this by educating users on computational biology and the limitations behind our tool. Our team is already actively participating in efforts to inform the public about synthetic biology and its many applications. As part of our outreach efforts, we sought expert advice from a bioethics professor who suggested that biased and insufficient data collection could lead to inaccurate predictions. These errors will be minimized as more viral samples are collected, leading to higher accuracy in our predictions. What about the potential unequal access to preemptive vaccines? Socioeconomic disparity is a global problem yet to be solved. An economics professor specializing in poverty analysis suggested working closely with local vaccination programs to ensure that the produced vaccines can be distributed to rural and vulnerable populations. Since the production of mRNA vaccines is 10 times cheaper than regular vaccines, this could be achieved under special health acts at a reduced cost or for free.
A vaccine to end this pandemic won't solve the root of the problem. The risk of infectious outbreak is increasing as we destabilize Earth's equilibrium. We intrude on ecosystems by cramming wildlife into unsanitary conditions, decimating habitats, and forcing wild species to come into closer contact with us. Additionally, climate change pushes disease carriers to expand their territory. All of these increase the risk of transmission between animals and humans. H1N1, Ebola, SARS-CoV-2, and 70% of all human pathogens originate from animals. Scientists expect new viruses to emerge and for old ones to return with a vengeance. Although Viper was trained on genetic sequences from SARS-CoV-2, its framework can be expanded to predict the evolution of other viruses as well.
Envision a world in which we diligently monitor virus populations in wildlife, livestock, and people. We could predict when, where, and which viruses will cause an outbreak, and have a vaccine ready before it happens. What if this world was within our reach? As we grow aware of the myriad of ways in which our actions impact everyone on Earth, it is time for humanity to unite with a collective goal, to use our innovation to influence the world positively with our actions. A new world is emerging. A world that prioritizes biology.
All right. Well, welcome UBC students. Thank you so much for being with us today. Wow. I feel like the judges are going to have some questions. Fred, start us off. Fred, you're muted.
Thank you. I was impressed by all the biology, but I was just wondering, in terms of your research, did you look into the models that are used to predict flu and how it changes every year?
Yeah, I can answer that. So we actually did look quite a bit into the way they predict the flu shot every single year, and we sort of parsed through like several different models, but one of them particularly was very similar to the model that we ended up going with for our design. So we kind of took a lot of inspiration from that and used it very closely in our model.
Okay. And I was just wondering, how many folks on your team understand what a Markov model is?
Yeah, it took us quite a bit to really comprehend and grasp the idea of a Markov model, and we're still sort of in the process of implementing it right now. We've had some failures. It is certainly a complex idea.
Thank you. Very good.
Any other judges with questions? Andy.
Hi, this is a really cool project. One of my questions might seem, I'm trying to figure out how to phrase this in a very friendly way, because I think it's meaningful, but it shouldn't be a deterrent. There are lots of predictive software out there already for the evolution of different viruses. What makes yours better or special, or why is it the one that people should be leaning towards?
I can answer that one. We definitely ran into lots of similar projects and things like that. I think our idea was more contributing to this pool of knowledge. Yeah.
Yeah, I can add a bit to that. We noticed in the literature that it's quite a novel approach to use deep learning in this way, and we thought it'd be very interesting and even fun to tackle it this way. We by all means are quite aware of the different approaches we could have taken and all the other softwares that are out there, but we thought we could sort of, as Janella said, contribute to the groundwork of this area.
Thanks.
I actually have a question. Were you guys working on this before the pandemic, or working on a version of this, or did you switch gears?
I can answer this. We're actually a team that participates in another competition called iGEM every year. That's why our team is so large. And when this pandemic hit, we saw it as kind of unprofitable to keep participating in iGEM. So thankfully, our PI recommended us to join Biodesign, and we're very happy we did. And our project before joining Biodesign was completely different.
Jean and then Heather.
Hi, I have a question. It's a pretty amazing project, and I'm not a scientist, so I'm not going to comment on that. But with the recent spate of the measles epidemic and people refusing to be immunized, or the movement of not immunizing children, you did some work, you did talk about education. Can you talk to me a little bit more about that? Like how you might get people to adopt a different version or a different type of vaccine, or something that's perhaps predictive. So if you can just speak to that a little bit, that'd be great. Did you hear me?
Students?
Yeah.
Hello, I can speak to that a little bit. So our team has always been really passionate about educational outreach. So we do host workshops on synthetic biology and also bioinformatics every single summer at our university and for high school students. And this summer, we're also looking to develop a workshop specifically for bioinformatics because it fits more with our project. And I think, educating even young children, like high school students, from a very early age can really help in this movement.
Okay.
Yeah, we also kind of foresaw that hopefully after this pandemic is all over, that there will be a more enhanced public mindset towards vaccination and this idea of real life viruses. And hopefully that can play into the public adopting our idea and realizing how important it actually is.
Mm-hmm. What role do you think scientists have in sort of working beyond just the sort of telling people what biotech is or the potentials of it? But do you feel like that's a role for you guys as potential vaccine makers to make the information accessible? it seems like culturally there's a lot of cultural resistance. Some of that might be a lack of education, or some of that might be a sort of tangential education that they're getting that is in conflict with other forms of education. So how do you guys feel about that as a young cohort of sort of vaccine thinkers, I guess?
Thank you for your question. I can answer that. Yeah, we agree that as scientists, it's very important, especially in today's world, to communicate with everyone else in the public and inform them of the science that we're doing and the knowledge that we're producing. And even with that, we're making, like on our website, we're making it accessible to everyone. Our tool, Viper, is an open platform, open source platform that's accessible online to anyone who wishes to have a look at it or use it for educational purposes. And on our website as well, we've tried to explain the science behind our project to anyone who might want to have a look at it.
All right, and lastly, we have Heather, and we're running a little short on time, but I'll let you go. Go for it.
Sure. I have two quick questions. One is, from my understanding in relationship to our current pandemic, one of the major issues around vaccines is the scalability of being able to make large amounts of vaccine after a vaccine has already been created. So I'm wondering if you've been thinking about this in terms of your development of the types of vaccines that would potentially be stored and then would be employed. So who would you use in terms of scalability? And then the other question is, does your project in any way sort of challenge, or I'm wondering whether it sort of fits within a kind of a model of proprietary vaccine research, which is the model that we've been seeing sort of up until this point. But one of the things that's really been interesting about the current pandemic is the ways in which scientific knowledge has really been shared across so many different universities and so many different laboratories, and whether you see your work as part of this much more kind of like open scientific platform, or whether it would fit better within a proprietary model.
I can answer the first question. In terms of the scalability of the preemptive vaccines we would make, our software can predict many different sequences, but we would probably choose a threshold of the couple most probable sequences that would be made into the mRNA vaccine. And this would be based on the Markov model, or like the neural network, and the receptor binding. So in this way, we hope to make the scalability more feasible. And also the fact that they are mRNA vaccines already makes it more feasible, because as we said in our video, they're about 10 times faster to make than other vaccines.
All right, thank you so much, students and judges, for all your thoughtful questions. We're gonna end there. Judges, if you have questions for the students on the teams you just saw, you can email them to Dan and Vina and I, and we'll make sure the students get those. So, all right, thank you so much. We're gonna go into a 15-minute break now, and we will reconvene in 15 minutes. Bye, everybody.
Going to be living in a surveillance state.
Science holds the key to our security and prosperity as a nation.
This is me.
My DNA.
People are being arrested simply to get their samples onto the National DNA Database.
Police can take DNA samples without a warrant
from anyone they arrest.
Certain segments of the population will be subject to genetic surveillance.
Me.
Me.
Me. You don't have to have done anything wrong.
You're all screwed.
Invisible, 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 invisible.
Sometimes I wish I was invisible. Have you ever wanted to be invisible? Now, you're invisible.
Back2You is a service that enables its users to regularly sequence their gut microbiome at home, maintain a database of their own sequencing results, and regain their lost bacteria. Back2You brings your bacteria back to you. This is Mary, 26 years old, a resident of New York City. Today is February 24th, 2023. As always, Mary wakes up to her alarm and goes to the bathroom. Today, when she enters the bathroom, she activates the Back2You sequencer by saying her name, Mary. After she poops and wipes her butt, she takes the backup pad that comes out of the slot and wipes again for sampling. She folds the pad and puts it back into the intake slot of the sequencer. She then flushes the toilet and starts her morning routines while the sequencer starts sequencing her sample. Later, the sequencer will send the result to Mary's personal Back2You cloud. Mary often sequences her stool sample at home to monitor her health conditions and has built her own gut microbiome database. 2 weeks later, Mary gets a bacterial infection and is prescribed with antibiotics. Over the course of her treatment, she gradually loses part of her bacteria community. At the end of the treatment, Mary opens up the Back2You app on her phone, where there is her personal time machine that has all her past reports. She scrolls back a few times and picks the February 24th report based on the app recommendation. Then she places an order of a 7-day bacterial supplement pills, customized to her gut microbiome condition as it was on February 24th. A few days later, Mary's bacteria order is delivered to her door. Mary starts to take her bacteria pills every day for 7 days. Over the course of taking the bacteria pills, Mary gradually regains her bacterial community. This is how Back to You works.
You wouldn't look at that and say, well, that looks like corn. I mean, the more I look at it, the more I think it would probably make a nice three-dimensional object. Then you could really see, the way, and the thick and thin of the fiber is part of its charm, I think.
The overproduction of corn in the U.S. has allowed it to dominate our food chain. We're taking the corn system's waste and turning it into wearable, sustainable denim. Not only are we using wasted corn husks, we take corn husks from plants that were marked as diseased with corn smut.
Scalability,
circularity,
biocentered design, rethinking waste and disease,
updating denim,
contaminated soils, communities saddled with lack of employment. These are the legacies left behind by the decline of the mining industry in South Wales. However, this contaminated soil can now be used for the production of something called a quantum dot, with the help from a very familiar garden friend. So, what exactly is a quantum dot? It is an incredibly small semiconductive particle. It emits different coloured light in different sizes when excited by light or electricity. They have been used in a range of different applications such as biological imaging, improved LED screens and solar cells. The industry is set to be worth over $11 billion by 2026. In 2013, researchers at King's College found that they can be biosynthesized using the earthworm through a process called mutual antagonism. When earthworms eat soils containing metals, they produce enzymes called metallothionein in its gut. The metallothionein grabs hold of the metals and stores it away in special cavities inside the cells. It reacts with another toxic element to make a safer compound. This is the quantum dot. Professor Mark Green of King's College explained,
Extracting a quantum dot made in a worm is a cheaper, more sustainable process than making quantum dots in the laboratory.
Quantum Industries is one company striving to exploit this phenomenon. Working with a disused coal mine, they have biologically engineered a giant hybrid earthworm optimized for high-yield production of quantum dots. These earthworms are hatched in secure wormeries inside the mine. To attract the wormer runs a light electric current into the soil. The worm is then tickled to release the fluid containing the dots. This mine uses its cadmium and selenium contamination to create a cadmium selenide quantum dot. The BBC interviewed Margaret Howell, CEO of Quantum Industries.
Though we intend to sell quantum dots on as a green and cheaper alternative, our real aim is to generate clean energy using the quantum dots in our solar flags.
Quantum Industries are now fabricating innovative solar flags using spray-on solar cell technology. The flags are raised around the mine, powering itself and the nearby community. Quantum Industries hopes this would re-establish mines as a source of energy, bioremediating the coal industry's pollution and revitalising the industry in the area. Olivia Bargeman, BBC News, Glenavon.
Hello, everyone. Welcome back to our final session of BDC Summit 2020 Day 2. Looking forward to getting back into our student projects and hearing from our judges. First, we're going to see the video from a California school called Nest Makerspace. Okay.
Behind your clothing, there is a huge human and environmental cost to fast fashion.
Fast fashion is built on a system of abusive labor practices. Fast fashion is poorly made and designed to be disposable. It quickly gets thrown into the trash.
The fashion industry is the second largest industrial polluter, second only to oil.
Additionally, right now, much of the PPE we are wearing in the COVID-19 pandemic is designed to be disposable, but it's made of plastic that ends up in our environment.
Imagine a world where that doesn't happen. By using kombucha leather and materials grown from local waste streams, we created biodegradable streetwear, Kokobucha.
It uses locally sourced abundant materials and waste stream materials within a 50-mile radius to grow materials for apparel.
Hi, we are Kokobucha. I'm Addie, and I go to Yerba Buena.
I'm Juan Flores, and I'm a freshman at Mount Pleasant.
I'm Penny, and I am an 8th grader at Castillero Middle School.
Hi, my name is Cuauhtemoc Martinez Marquez. I am in 9th grade and I attend Evergreen Valley High School.
So this is my kombucha leather hat. It's made with cactus fruit kombucha. I decided on a bucket hat because like I wear them and they're like really comfortable and it blocks the sun out of your face and they're just like really stylish, and I knew that people would want to look good if you're gonna wear something, so I'm like, why not make a bucket hat? But also like being in this time with the pandemic, we're worried about like, well, a lot of people are wearing the face masks, so we put a shield inside of the hat to block out any other germs or pollutants getting in our face. Corinne drove the kombucha leather for me thick enough so that when she delivered it to me, I was able to sew it. I was hand sew it together. I made it within 3 days. And embedded in the top of the hat are like the cactus fruit seeds, so that if you need, like if it's destroyed, you can repurpose it and grow another plant out of it.
Hi, this is my jewelry line. I decided to make an assorted group of items because with jewelry there are lots of different kinds, and there are lots of different kinds of plastic jewelry too, which is a big problem because they can't go away. They just stay on the earth, and it's actually a big problem. So I figured, why not try and make these jewelry pieces out of biodegradable materials? That way people can still look good, but they know that their jewelry isn't harming the earth after they're done wearing it. So I made a bracelet out of kombucha leather, and I sewed a little design on it with just like regular thread. But it's like a bracelet. I'm also creating some rings out of mycelium right now. They're growing, but they're in molds, and I just have to bake them. But those will also be biodegradable. And I'm also planning on doing mask designs and charms, because masks are very relevant right now, and, you know, why not spice up your mask a little bit? Because we're going to be using them a lot anyways. I made a line called Animasks, and they're animal masks, basically. It's pretty self-explanatory. There's little animal faces on the front, and you can pick between different animals, so you can feel like you have your own special mask and go out looking cute in pandemic times.
Hi, I'm Juan, and what I have designed are work gloves. And the reason I wanted to do work gloves was because of COVID-19, and also because of I am a gardener, and I work with my dad a lot. So gloves are very important to me, so like I don't injure my hands like when I'm carrying stuff or when I'm like working with like plants that have like thorns. So I experimented making stronger biodegradable materials, like the one on the right, which is kombucha leather. There's a huge human and environmental cost to fast fashion.
Fast fashion is built on a system of abusive labor practices. Fast fashion is poorly made and designed to be disposable. It quickly gets thrown into the trash.
The fashion industry is the second largest industrial polluter, second only to oil.
Additionally, right now, much of the PPE we are wearing in the COVID-19 pandemic is designed to be disposable, but it's made of plastic that ends up in our environment.
Imagine a world where that doesn't happen. By using kombucha leather and materials grown from local waste streams, we created biodegradable streetwear, Kokombucha.
It uses locally sourced abundant materials and waste stream materials within a 50-mile radius to grow materials for apparel.
Hi, we are Kokombucha. I'm Addie, and I go to Yerba Buena.
I'm Juan Flores, and I'm a freshman at Mount Pleasant.
I'm Penny, and I am an 8th grader at Castillero Middle School.
Hi, my name is Cuauhtemoc Martinez Marquez. I am in 9th grade, and I attend Evergreen Valley High School.
So this is my kombucha leather hat. It's made with
I think we might have just had a couple of technical difficulties right there. Don't want to do a disservice to the team, so we are going to play that one more time. So everyone bear with us, and we get to see that one again.
Behind your clothing, there is a huge human and environmental cost to fast fashion.
Fast fashion is built on a system of abusive labor practices. Fast fashion is poorly made and designed to be disposable. It quickly gets thrown into the trash.
The fashion industry is the second largest industrial polluter, second only to oil.
Additionally, right now, much of the PPE we are wearing in the COVID-19 pandemic is designed to be disposable, but is made of plastic that ends up in our environment.
Imagine a world where that doesn't happen. By using kombucha leather and materials grown from local waste streams, we created biodegradable streetwear, Kokombucha.
It uses locally sourced abundant materials and waste stream materials within a 50-mile radius to grow materials for apparel.
Hi, we are Kokombucha. I'm Addie, and I go to Yerba Buena.
I'm Juan Flores, and I'm a freshman at Mount Pleasant.
I'm Penny, and I am an 8th grader at Castillero Middle School.
Hi, my name is Cuauhtemoc Martinez Marquez. I am in 9th grade and I attend Evergreen Valley High School.
So this is my kombucha leather hat. It's made with cactus fruit kombucha. I decided on a bucket hat because I wear them and they're really comfortable and it blocks the sun out of your face and they're just really stylish, and I knew that people would want to look good if you're gonna wear something, so I'm why not make a bucket hat? But also being in this time with the pandemic, we're worried about a lot of people are wearing the face masks, so we put a shield inside of the hat to block out any other germs or pollutants getting in our face. Corinne drove the kombucha leather for me thick enough so that when she delivered it to me, I was able to sew it. I was hand sew it together. I made it within 3 days. And embedded in the top of the hat are the cactus fruit seeds, so that if you need, if it's destroyed, you can repurpose it and grow another plant out of it.
Hi, this is my jewelry line. I decided to make an assorted group of items because with jewelry there are lots of different kinds, and there are lots of different kinds of plastic jewelry too, which is a big problem because they can't go away. They just stay on the earth, and it's actually a big problem. So I figured, why not try and make these jewelry pieces out of biodegradable materials? That way people can still look good, but they know that their jewelry isn't harming the earth after they're done wearing it. So I made a bracelet out of kombucha leather, and I sewed a little design on it with just regular thread. But it's a bracelet. I'm also creating some rings out of mycelium right now. They're growing, but they're in molds, and I just have to bake them. But those will also be biodegradable. And I'm also planning on doing mask designs and charms because masks are very relevant right now, and why not spice up your mask a little bit, because we're going to be using them a lot anyways. I made a line called Animasks, and they're animal masks. Basically, it's pretty self-explanatory. There's little animal faces on the front, and you can pick between different animals, so you can feel like you have your own special mask and go out looking cute in pandemic times.
Hi, I'm Juan, and I have designed our work gloves, and the reason I wanted to do work gloves was because of COVID-19, and also because of I am a gardener, and I work with my dad a lot, so gloves are very important to me, so I don't injure my hands when I'm carrying stuff, or when I'm working with plants that have thorns. So I experimented making stronger biodegradable materials, like the one on the right, which is kombucha leather with cactus fiber, so that the gloves aren't super fragile and can take a beating. And I also designed, I added a kombucha recipe. So if anything happens, let's say it rips or something, you can make some kombucha leather, some extra kombucha leather, and then put it on wherever the rip is, and then just grow it back, and then there you have an automatic fix, so you don't have to throw it away.
My skirt is made out of kombucha and was designed to be a mix of streetwear and elegance. With that idea in mind, it contributes to all ages, which allows all age groups to become aware of what is going on in our world. I added a slit with sensors in the edges, allowing a ribbon to tie it up, making the garment more breathable. The sensors would also serve as air sensors to help users become aware whether the air in the environment is safe or whether the area is high in air pollution and contaminants.
We engaged the community in a couple of activities.
We did a community sensor preference survey and DIY face mask kits.
Tests we conducted. Number 1, we conducted a sweat simulation to see if kombucha leather dissolved in plain water or sweat faster. We did another test where we fed kombucha culture with cooked cactus fruit instead of refined sugar. And then a third test was that we laser cut kombucha. Number 4, we were feeding mycelium a feedstock of cocoa bean husks. Number 5, blending 2 biomaterials. We combined the kombucha leather with cactus bioplastic in between. And then a 6th test that we did was grow bioassemblies, and we grew mycelium into mesh. We spoke to a guest speaker at Stanford, Matthias Kaplan, and he talked to us about biosensors.
Greetings from San Jose, California. My name is Cuauhtemoc Martinez Marquez.
Greetings from San Jose, California. My name is Penelope Medina Sanchez.
All right, hello, I'm Juan Flores.
Hi, I'm Addie.
Awesome. And what's your team name?
Kokobucha.
Kokobucha, yeah.
Our apparel can help save our earth. Help keep our earth clean with Kokobucha apparel.
Welcome to BDC. Our judges Q&A is going to start in just a minute, but I did want to apologize. I'm glad that we were able to play our video and get the right one, and now the judges have seen it. So welcome. Amazing project. I want to point out your shirts. Penny, we can't see, but I'm sure you're wearing one too. Awesome. Cool. So let's open it up for some Q&As. Hey, Jean, start us off.
This is a really great project. I liked your process a lot. And the combining of the two materials was pretty genius. So what drove that? Like, what made you think of taking the mycelium and the kombucha together? You had those kind of floret shapes and you were encapsulating it in the kombucha. So what drove that decision to try that? You guys can answer.
You guys are actually all on mute if you want to keep those off for the Q&A.
Okay, so one thing that allowed us to come to that decision was that from the beginning of the process, we visited a lot of labs. One lab was the Tech Museum in San Jose, California, and we had a little like mycelium and little kombucha section. And in that section, we were able to learn more about those products, and we found a lot of interest towards those two particular items because we from the beginning knew we wanted to do something that was fashion, something that had to do with fashion. And so using mycelium and kombucha, we knew that it was capable and those items are going to be able to be capable of doing so.
Right, because you gained kind of technical qualities from both types of materials. So you kind of doubled down on both of the benefits.
Mm-hmm.
Yeah, it was great. So as you are all in a makerspace now and you're young, what did the effect of like growing material have on how you think about the material world around you and how you might organize yourselves to that as creatives? Do you know what I mean by that?
Yeah.
Yeah.
That's a great question. For me, being able to work and harvest things, it allowed me to change the perspective as to not only how our fashion is right now and how our generation specifically changes our trends in clothing so quickly that it can cause so much harm to our landfills. But it also lets me know more about the idea of biodegradable solutions and how we are able to do so much with the products that have to do with mushrooms and stuff like that. And how there's so many solutions out there that we just have to put our mind to actually being able to solve these problems with solutions that don't affect our earth.
Excellent. Excellent question. And I'm going to leave the floor to my colleagues.
Andy.
Hi, I really liked a lot of the designs that you guys came up with. I was wondering if you've started wearing all of these materials. Have you started wearing the bucket hat and the gloves? Have you started trying to use them? How do they feel?
So yeah, I made the bucket hat, and when I first made it, I tried it on, but since it was just a prototype, and some of the parts of the hat were thinner than others, I didn't want to force the hat onto my head, but I would look in the mirror to see how it would look. And so when I delivered it to Corinne, she took a model of a face and put the hat on that to show what it would kind of look like. And yeah.
It looked really cool. I was really curious about the gloves too, though. Do they have utility right now?
I made the gloves, and yeah, we haven't tested them yet. So
it's exciting next step.
Yeah.
Hey, Leslie.
I got to know the results of the sweat test experiment. Congrats on the project and the set of experiments you did. Do you want to elaborate on one of them? I'm curious about the sweat test. Maybe you guys want to talk about a different one. But the question you asked and the result of that.
So during our sweat test, we took water and salt and put that in a little petri dish and added a piece of kombucha leather, and we let that sit for a week and more because we haven't been at our Makerspace in 3 months, so we don't know if it has dissolved yet. But after the first week that we have seen, it did not dissolve. So we know that with our kombucha leather, it's going to be durable. And so, yeah.
Nice. Thanks.
Anybody else? I do have a question for your team. Have you talked to any of your friends or family or anything and asked if they, have anything that they particularly would want, made out of your material, or if they would wear what you're making, or kind of done any, out in the field, how people feel about it?
I actually got the idea of the skirt from my sister because she's going to law school very soon and she needs a lot of professional work. But we were looking into that and we were like, this clothes isn't even that nice for what the price is. And I was like, maybe I can do a biodegradable version of that can actually work for its purpose and still have more to it. Along with that, I did talk to my friends about it and they thought it was crazy how we were making skirts and clothes out of kombucha, which is a tea. So they all thought it was crazy. And I liked how challenging the idea was and how much we had to challenge ourselves to be able to complete the project.
Addie, can you hear us now?
Yes, thank you.
I'm not sure if you heard any of the other questions. Just to give you a chance to answer, I had asked if you guys had spoken to friends or family or anyone in your community about their reactions to your material and the products you guys are making.
Yeah, I told my stepmom about it, and she was actually very excited because she is very stylish, and when I told her that my team was making clothing out of things that are biodegradable, she was very interested, especially because fast fashion is such a problem when it comes to pollution. Yeah.
Judges, any final thoughts? Otherwise, happy to send along questions to the team afterwards. Okay. Thank you so much, Nest Makerspace students. Really, really exciting project. I look forward to seeing where you guys take it after this. Good luck. And we are going to move on to the University of California, Berkeley. Students, you can,
We are Thermosilk, an undergraduate team of biologists and engineers from UC Berkeley. We believe that applications of hornet silk can improve the lives of millions of people. We designed a flexible, biodegradable, thermoelectric textile that draws its inspiration directly from hornet nests. We believe that incorporation of this novel textile into the structure of emergency shelters can dramatically improve their quality and safety for inhabitants. There are more than 70 million forcibly displaced people across the planet, of whom nearly 26 million are refugees. The COVID-19 global pandemic and the mounting consequences of climate change have made it undeniably clear that every nation and its inhabitants are vulnerable to displacement.
And there is another field hospital going up, this one in Central Park. Not a moment too soon. We just learned there are 14 more reported deaths in New York City.
This is one of the greatest challenges of our generation, yet current emergency shelters are anything but optimal. We designed Thermosilk as a solution to two of these critical problems. We focus on the issue of connection to a dependable power source and a way to keep the temperature of a shelter stable in extreme heat. Access to electricity is critical in forming a livelihood in a modern world, yet 7 million refugees struggle with access to this basic resource. Lack of access to electricity is linked to detrimental impacts to health, education, and economic livelihoods. Access to electricity is linked to improved quality of life and lower mortality rates. Electrification has been shown to be directly proportional to increases in literacy, increases in study time for students, and improvements in gender equality. Households that were electrified saved 4% of their monthly expenditure by reducing their energy costs. Yet emergency shelter housing too often fails to provide electricity for its occupants. Emergency living situations also fail to protect their inhabitants from unlivable weather. Standard UNHCR tents are rated to be safe up to 40 degrees Celsius, but average summer temperatures are well above that in many Middle Eastern and North African countries. It is in these dangerously warm areas that the majority of refugees live. A recent case study conducted within the Azraq refugee camp in Jordan reported that the internal climate of shelters regularly rose to temperatures deemed life-threatening for inhabitants. These conditions were observed for more than 20% of the year. Put another way, for a fifth of the year, many refugees' only shelter from scorching temperatures was deemed uninhabitable. Over half of the world's refugees are children, a population at especially high risk of heat-related illness. Homelessness is also a rising issue and is prevalent in many major cities, including our base, Berkeley, California. Recently, homelessness has increased in response to high rates of unemployment and other conditions induced by the COVID-19 pandemic. Homeless populations and globally displaced refugees share many of the same hardships in regard to their living conditions. Roughly 1/3 of homeless mortalities in the Phoenix area are due to heat-related illness, a number that has increased dramatically in recent years. As climate change continues to place more and more people from every country at the mercy of worsening weather patterns, a solution becomes more important than ever. Humans are not the only species to build shelters to protect from harsh climates. Social insects have lived in inhospitable environments for millions of years, yet their bodies lack many of the complex homeostasis mechanisms that humans possess. Researchers report that hornets have an especially creative way of managing this. Inside their nests, hornets weave silk structures that store heat as electricity on hot afternoons, only to release this energy again as heat in the chilly night. This silk enables hornet nests to maintain steady conditions while the temperature outside fluctuates wildly, even at higher temperatures. Notice how steady the temperature in the nest remains. At ThermoSilk, we are incredibly excited by this research and see an opportunity to apply the Hornets method of thermoelectrical regulations to human shelters. We believe that we can use the properties of Hornet silk to convert excess heat to electricity during the day to use as a steady source of power. We can produce biodegradable Hornet silk linings for compatible emergency shelters that will generate electricity from ambient heat. By converting heat to electricity, thermal silk linings will also stabilize the temperature inside the emergency structures in which they are deployed. This means our silk can act as both a generator and temperature regulator. According to our research, on a hot day, one of our linings could produce at least 9.66 kiloamp hours of electricity. That's roughly 10 times the discharge of a conventional car battery. If redirected, this electricity could be used to power light bulbs and larger devices including medical equipment and food or vaccine refrigerators. ThermoSilk system promises to make emergency structures safer and to provide a higher quality of life to inhabitants while supplying access to reliable electricity. To synthetically engineer the silk, we begin by identifying key genes used to make the protein. Many of the genes used to make hornet silk proteins have already been identified in previous studies. These genes will be coded into a ring of DNA called an inducible plasmid. This plasmid will then be transformed through the cellular membrane into E. coli. We deemed E. coli to be the best vector to use in the research and development process due to its ease of use and versatility. Once the plasmid is transformed into E. coli and the bacteria are screened, the plasmids will be induced to start protein production. The proteins that are produced in the bacteria will then be extracted and spun to generate a thread. This thread will be woven into fabric using methods common in the textile industry. The resulting fabric will be fitted with electrodes at key points to enable us to tap into its electrical potential. We were lucky enough to consult with leaders in the recombinant silk world on our methods of production, especially bolt threads and their spider silk production techniques. Our research suggests that transitioning from E. coli to yeast will allow us to scale up our production and greatly reduce the cost of our product. Once this transition has been made, the process of the production of silk proteins will be very similar to that of beer mated with conventional textile fabrication, presumably with similar costs. Researchers have been successful in manipulating the genetic sequence used in order to change the molecular structure of their silk to develop features that are not present in nature. We believe that we can also use this process to improve the thermoregulatory or thermoelectric properties of the silk. We will distribute our thermoelectrical linings in conjunction with our own polypropylene and steel shelter structures so that the lining and shelter integrate together perfectly. Drawing further inspiration from the hornet's nest, these structures are hexagonal in order to enable higher efficiency, horizontal stacking, and modularity. Our structure will include an integrated electrical system encompassing wiring and lights. It will be built to easily plug in batteries, solar panels, or other components. In the future, the ThermoSilk project can lead in many exciting directions. Although the ideal system is our current design, we also want to make sure that this technology can reach any who need it. To this end, we would like to expand production to linings compatible with many major emergency shelters already in production. Down the line, we would also like to integrate bathroom facilities and water collection systems into the shelter design. On a global scale, we eventually anticipate these shelters to be compatible with different types of climates and lifestyles. At ThermoSilk, we have designed a novel and biodegradable emergency shelter lining that produces electricity while protecting its inhabitants from dangerous heat. We've done it because we believe technology should reflect the changing global landscape and that the best way to evolve is to follow nature. If we have anything to say about it, the emergency shelter and bioenergy sectors will look very different in coming years.
Welcome back. Welcome, University of California, Berkeley team. Awesome project. Thank you so much. I'm going to open the floor to our judges. Jean.
I will kick it off again. So it's pretty interesting. You were talking about the hornets. Did you investigate the shape or the kind of manufacturing of the nest and its relationship to the ventilation and sort of the temperature management in the nest?
Yeah, absolutely. There are some other thermoregulatory processes going on within the nest. And these were not our focus, but they do contribute to that graph you saw where the nest was staying stable. But we think that part of the effect is in the thermoelectric properties.
So you think that it's beholden in the material itself?
Right.
So if I can take that material out and put it into another situation, then we can accomplish that task. I'm going to shut off my mic. Someone's coming in. So. Okay, so yeah, I'm going to leave the floor.
Okay, thanks, Jean. Andy and then Fred.
Hi, this is a really cool project. I was curious, have there been attempts to try to harness the electrical capacity out of this silica in the past and convert that into a useful electricity? And if so, how do they actually do that? How do you, because you'd said there would be electrodes spaced throughout, but how does that actually change from the potential to something that could be of utility?
Yeah, I'm happy to take that question. Thank you for asking. I know the technicality, sometimes we like to ponder about, how possible is this technology. And through the research that we have read, they've been able to increase the voltage from millivolts, which is produced from a single thread, to thousands of millivolts, so accounting to the number that we need by stacking the layers concentrically. And and that's what allows us to scale up in voltage. By then aligning each of these threads, which are layer by layer, we're then able to increase the amount of current that we're able to pull through these. So while the laboratories have experimented what it looks like with a single thread, then they have theorized that we can scale upwards and through the processes that they have described. That's exactly how we've applied that technology with this project.
All right, Fred, go on.
Yeah, this is a very creative and aggressive sort of project. I think that you can do this. Very interesting. And again, good biology. I am curious though, because people have been very interested in spider silk, right? Because it's so strong, and there've been lots of attempts to, people have made cloth out of spider silk, but the idea is that it takes thousands of spiders or more than that. So can you do that in bacteria? Can you do that in something else? And it's turned out to be really complicated to get the proteins and all that to line up. Have you thought about the challenges that would be involved in moving this from the insect into some kind of culture that would produce it at bigger amounts?
I can take this. Thank you so much for your question. So if you just look at fundamentally spider silk and hornet silk, their purposes are very different, right? Spider silk is for catching bugs and then stuff that they can eat. Hornet silk is designed to produce nests. And so that automatically brings in more stability, more interesting properties that we want to harness. So in terms of challenges, like you mentioned, spider silk requires a lot of spiders to make the actual product. What we plan to do is to take the genes that are responsible for making the hornet silk and put those into the bacteria. And E. coli is a really great starting spot for that because they will just do whatever they're told to do. So that's how we plan to, initially at least, start mass producing the protein that we need from them. some challenges that we anticipate, like if there's any issues with hornet silk, like we mentioned before, there's some properties that we may want to amplify or even delete, like that could be done with synthetic biology techniques. So that's just going to be like some research stuff that we need to get over first. But like I said, because the fundamental purpose of each of these silks is so different, we know that hornet silk is going to be like really well suited for this.
Mm-hmm. Are there any factors actually in the silk itself, not just the silk threads, but in the structure that leads to this property, or is it just the protein itself?
I can take that question. Thanks a lot for your question. Yeah, so the mechanism of the thermoelectric properties within the nest are poorly understood. We know that the properties are there, and there's conjecture about how it works, but it's not well understood. The thought right now, the best theory, is that the silk forms a semiconductor with 2 parts to each thread, and one is a p-type. I don't know if you're familiar with like the idea behind the semiconductor, but that one is a p-type material and one is an n-type material. So you have electrons moving from one to another. And so there is structure within the thread, and this also poses a potential roadblock to us, but we think we can make it work.
Yeah, good going.
Well, unfortunately, because of time, we do have to move on. I'm sorry, Yasmina and Heather. We're already running quite late. But ThermoSilk, thank you so much. Judges, if you have questions, please do email them to Vina, and we'll be sure to get them over to the team as soon as possible, basically right after the session, so the team can answer. Thank you so much, UC Berkeley.
Thank you.
Awesome
Thank you.
Thanks.
Thank you. Thank you for having us.
All right, and up next, we're going to hear from Cal State LA.
Hello, we are the Cal State LA team, and we are ecstatic to show you our innovative product. But first, let's get into some brief history of the Feminine Hygiene Pad.
Menstrual products have come a long way these past 100 years, from humble beginnings to simple rags to complex belts and highly uncomfortable aprons. It wouldn't be until the early 1970s, however, when menstrual products would become more convenient and comfortable for female consumers. By the 1980s, various menstrual product brands would emerge, such as Always, Kotex, and Stay Free. These brands would set the trend for disposable pads and tampons that never biodegrade. And where do these products usually end up after being discarded? Well, in our landfills, of course, but unfortunately, these products will leave an everlasting environmental impact due to their fiber makeup that consists mostly of plastic materials. If only there is a new, innovative menstrual product that could challenge the environmental issues of the modern pad and tampon.
As we know, the world around us is filled with plastic, and menstrual products are no strangers to contributing to plastic pollution. According to the National Geographic, in 2018 alone, people in the US bought 5.8 billion tampons, and over the course of a lifetime, a single menstruator will use somewhere between 5 and 15,000 pads and tampons, the vast majority of which will wind up in landfills as plastic waste. Top leading U.S. brands such as Always pads are packed with plastic layers. Unfortunately, plastic has been our normal standard, and that clearly shows in the whole pad assembly itself. Pads now incorporate leak-proof polypropylene or polyethylene as the base. Plastic wings were incorporated to have a secure attachment to underwear, and designers were also able to weave thin polyester fibers to wick the fluid into the core of the pad. This is just the basics of the pad that incorporates plastic. The packaging, on the other hand, is another contributor to the unsustainability of menstrual products. As you can see, our project is critical to addressing the current environmental problems that the feminine hygiene industry fails to change. Our solution is to erase all plastic materials in a new feminine hygiene product. Therefore, we created a 100% biodegradable pad made from all organic, regionally sourced materials. Let's take a closer look into our pad. The top layer is made from organic, unbleached cotton for comfort, breathability, and to serve as a hypoallergenic and antibacterial barrier. The middle layer is a liquid transfer layer made from seaweed fabric fueled by algae for its absorbency, which is also a great natural plant fertilizer. Lastly, the bottom layer is a leak-proof layer made from corn-based materials, a better alternative to plastics. This layer also comes with wings to ensure the placement of the pad. Now moving on to our packaging. Our boxes are made entirely from recycled cardboard, and our seed envelopes are made from recycled paper and regionally sourced plant seeds here in California. That's what makes our product special. We envision Flora Nouvelle as a start for people to see how great an impact a product like ours will do to better the environment. Even if our product is misused, it will not contribute to plastic waste in landfills.
We envision a product that can be efficient and mindful of the environment. A closer look to the life cycle of Flora Nouveau's menstrual pad starts with the raw materials. We source the cotton, seaweed, corn, and including the poppy seeds from within the state of California. Throughout the process of creating the perfect blend, we have to refine and break down the seaweed and corn in order to draw their unique properties to create an absorbent inner pad and leak-proof outer layer. The lab breaks down the seaweed and allows for us to create absorbent textiles. Once the pads are made and packaged, they are distributed to your local supermarket where they can be available for purchase. While being environmentally mindful, take home one of the Flora Nouveau's poppy flower pads to grow your own garden. After the use of the pad, you can enclose it in one of the paper seed envelopes and plant a new garden. After planting it, the pads will decompose and serve as vital nutrients for the seeds to sprout. If you continue to water it, soon enough you will have a healthy, flourishing garden that will be beautiful and eco-friendly. And don't worry, if gardening is not for you, throwing it away won't harm the environment. It will decompose on its own, and who knows? We might end up with flowers in the landfills. We welcome you to join our journey of protecting our home and be part of the solution.
All right. Thank you so much. Welcome. Judges, opening up the floor. Heather, you're on mute.
Right, I am. I was just saying that thanks for your presentation, and I appreciate the part where you're talking about the planting of these pads, although I'm not totally sure how many people would do that. But besides that, I'm wondering primarily why you chose to have disposable products rather than reusable products, like the range of reusable products that are already available for people who menstruate, including things like Thinks underwear or the Keeper, which is made of natural rubber, or other kinds of products. So why disposable products rather than reusable ones?
I can go ahead and answer this question. So one of the main motives why we wanted to create this product was we wanted consumers that purchase these pads or tampons that are disposable, the outcome of planting this pad into a garden or just planting it in general, or knowing that if you're going to dispose of it, that it's going to emerge some kind of new life. It kind of brings this this emotion of being proud of your menstrual cycle for consumers. In some cases, sometimes women consumers feel like discomfort or some kind of shame when it comes to like their menstrual cycle. So with this product, we would be able to promote a sort of proud feat, and knowing that when you're either planting this pad with the seed paper, or you're either just going to dispose of it and it's going to do its thing and biodegrade, it's going to promote this form of new life through the poppy plant. So we wanted to focus more on that this product is biodegradable, but the aftermath of it would evolve into the birth of a new life form.
Great, thank you.
Thank you. I'm going to go to Anne, just because.
Hi. So thanks for taking on a problem that is, I guess, common in I think everyone's looking for solutions that are non-toxic and healthy and promote the planet. So I sort of laud your efforts there. On the other hand, you also looked into making this a local product in California for a sustainable purpose. I'm curious if in your research you looked into the perception of the biohazard of sort of personal bodily fluids at a time when we're hyper conscious of where we put our sort of biomaterials or biohazard materials, what the barriers to that use might be with such a sort of innovative product, and particularly the idea of then burying it for sort of recycling into a second life.
Thank you for the question. Yes, so we did look a little bit into that. So by putting the seaweed and also adding different types of like bacteria into the packaging, it allows for the breakdown of all the like the blood or the menstrual cycle that would, in a sense, just kind of break down and create the energy that it needs to grow these new plants. And kind of piggybacking on the idea that it was only in California that would be like our start, and we would continue growing forward into other like states, not just poppy seeds, but other flowers as well.
Thank you.
Let's do Andy, and then Annie Lou. If you had a question, you can be the last one. Go on, Andy.
Well, I just had a really quick question about the biodegradability of this. The idea is that you found biodegradable materials, but if this does go to a landfill, does this break down readily if it's in a space without any light or oxygen or any of the things that we usually think of when we're talking about stuff biodegrading in our front yard? And then how is that going to be affected by the addition of high nutrient levels?
I can take the question. So we did some research on the certain layers. They all have like cotton, the seaweed, and the corn, and they all had a different, let's say, time spans of biodegradability, and some were like a few weeks or a month, and then some had some temperature requirements, but that's where we would have to like do some trials and errors that we didn't get to do.
All right. Thank you so much, Cal State. Awesome project. Thanks for being with us. Looking forward to seeing what you guys do next.
Thank you.
Hi guys. We are going to move into our final video for the day, final presentation, which is going to be the University of Cincinnati.
New York City. Home to almost 8.4 million people, this beautiful city is where we wish the 2020 Biodesign Challenge could have been held. Like many large cities, New York City is filled to the brim with energy, excitement, and life. However, it is also plagued with the problems often correlated with urban areas: inequitable food access, severe lack of local food production, and high food insecurity rates. With 1.2 million individuals in New York City being food insecure, that is 12% higher than the national rate and 21% higher than New York State. This is Sam. He's decided to try his hand at a little food production. He lives in an apartment in Queens and wanted to grow some produce on his balcony. He figured, how hard can it be? Little water, dirt, sun, and soon enough plants will be there, right? Wrong. Growing produce is complicated, labor-intensive, and difficult. Creating a climate-appropriate growth matrix and balancing the various components of the soil often requires expertise beyond the average person. We want to change that. This is Cece, and she's holding a life brick. Like Sam, she also wants to start growing some urban produce, but she is utilizing a much simpler tactic. That is, she is using life brick. The layers of the brick all provide essential components of soil development. So, all Cece has to do is follow the provided instructions, and soon she'll be flexing her green thumb with ease. The importance of accessible urban agriculture cannot be overstated. It can play vital roles in supporting communities through social, environmental, public health, and economic facets. Adequate access to nutritious food can also directly affect the mental, social, and behavioral development of children and cannot be taken lightly when food should be a human right.
Thanks for watching. Let me introduce you to our team. I'm Cecilia Favorito, a freshman in design originally from Buffalo, New York. I love cycling, doing watercolors, making my own earrings, and going on hikes. I am passionate about conscious and human-centered design, and biodesign allows me the innovative space for these types of developments. LifeBrick grew from the roots of our team's interest in solving important, interconnected environmental problems.
Hi, my name is Manish Chidambaram, and I have just graduated with a degree in medical sciences and a minor in psychology. I'm a singer, adventure seeker, and seek to do my part in helping people. LifeBrick resonates with me because it is our chance to change the way food affects those with limited access to it. It lets us grow where, how, and what we want, and can be a game changer in more easily providing food.
Hi, I'm Sam, a recent graduate with a focus in medicine and nutrition. I play soccer and love all things outdoors. Through my experience working with nutrition deficiencies in urban areas, I've seen the direct impact that inequitable food access can have. By easing the process of urban agriculture, I believe LifeBrick can play a direct role in making our communities healthier and more sustainable. Our interdisciplinary team has been working diligently to create this solution, and we are so excited to introduce you to LifeBrick.
Our process in designing LifeBrick sought to follow a design thinking approach. We started by getting inspired. We also did an initial mind mapping exercise to start laying out some of our initial ideas for a solution to an environmental problem, or even just to understand the network of those environmental problems.
DC really wanted to focus on soil science, agriculture, potential fertilizer upgrades, and maybe even like working with compost, and then maybe even an urban environment. Hopes to capture and recycle nutrients and
We've really narrowed down the idea we want to move forward with.
Our collaborative brainstorms, individual research, and discussions with experts helped us narrow down our path of investigation.
It will reach for those nutrients, which is super important because that
a much more viable and larger topsoil to where now you can grow.
In order to get further, we dug in and made a couple of phone calls.
Hi, Professor Arnold?
Yes. Hi, Sam. This is David Cable. How are you?
Hi, Professor Kupferly. Thank you so much for taking the time to talk with us today.
We learned a bit about their backgrounds and how they could help us with their expertise.
Tasked with setting the vision, enabling our scientists to address agricultural problems using the best science there is.
After explaining our concept, we got some feedback on how to improve from where we were. They asked questions that pointed us in new directions, expanding our perspectives on the project.
Is the matrix you're selecting suitable for your crop growth? And that's another question we need to address is what crop do you see growing or what products? And is it going to be economically?
They even suggested some new ideas that we ended up incorporating into our product.
The opportunities for, I don't know if you've read about these, but cover crops.
The experts also gave us direction on literature to further our studies of the soil science. We did lots of reading to solidify our direction.
It's actually empty spaces, the soil pores that are...
We all got together and watched Symphony of the Soils, which really gave us a whole new perspective of what it takes to create soil, how long it takes to create soil, and just the relative abundance of different essentially things that are part of the soil.
To define our goals, set timelines, and discuss our findings, we met bi-weekly. Before COVID-19, we were able to solidify our idea and even started planning for prototyping and model making.
This can't. This is totally chemically. I mean, that's so feasible, especially when you're talking about the
Working remotely forced us to adapt to a new form of collaboration. We began to synthesize our ideas, validate our concepts through research, and brainstorm ways to best communicate our project. Through persevering, we were able to collaborate remotely and present you with LifeBrick.
Thanks, Cece. Now that we've talked a little bit about our process, I want to talk to you about our concept and what exactly LifeBrick is. We wanted to create a soil source that retained all of the vital components of naturally occurring soil in a user-friendly way. To do so, we created a brick composed of 5 layers: mineral, carbon, profile, biota, and cover. Let's begin with the mineral layer. Naturally occurring soil often derives nutrients from its underlying geological material, referred to as parent material. We synthesize this with the granite and limestone matrix. This supplements the soil with nutrients such as calcium and phosphorus, and also acts as a buffer to maintain a neutral pH for optimum soil use. Like all of our layers, our mineral layer is comprised of recycled materials, primarily from restaurant and construction waste. Next up is the carbon layer, primarily composed as wood chips. As this layer breaks down over time, it will continue to provide long-form structure for the brick. In addition, it will also become a fertilizer source, decomposing into usable plant nutrition. Next is our profile layer, comprised of sand, silt, and clay. We use an equal parts ratio to form a lush loam layer. In addition, sediment diameter and surface area can be varied to determine porosity, water retention, and water drainage. This allows us to customize the optimal brick for varied climates. The soil is then inoculated with a mycorrhiza biota layer. These fungi form a symbiotic relationship with the crops. In exchange for simple sugars, mycorrhizae present water and mineral nutrients in a form plants can readily consume. Lastly is the cover layer. By utilizing the short life cycles of carbon sequestering legumes and wheatgrass, we work to strengthen the integrity of the soil, and the resulting decomposition works to catalyze the next batch of crop growth, upon which the customizable seed sheets, comprised of the desired produce starters, can be easily integrated into the soil system, and the consumer is well on their way to a flourishing urban garden.
As the current LifeBrick is geared towards urban farming in New York City, this product is expanding to include terroir, restaurants, education, and communities. The concept of terroir includes a set of farming practices and soil composition similar to that of the original soil where the crop is originally grown. This practice actively changes the phenotype of the crop and produces a more authentic taste. As restaurants would benefit from this and seek to provide the most fresh foods, and as we can benefit from certain restaurant wastes to be used towards specific layers of the brick, this mutual relationship will aid in their culinary gardens and our LifeBrick functionality. As child obesity and nutrition is of alarming concern, LifeBrick is an educational tool. It has also been found that garden environments have a positive impact on students who learn to increase fruit and vegetable intake and physical activity. LifeBrick is an opportunity for communities to come together through efforts for community gardens. These efforts bring community ties while increasing sustainability and food security in the poorest of neighborhoods. We are so excited to see LifeBrick continue to grow. If you have further questions or are interested in learning more, please check out our LifeBrick website.
All right. Thank you so much. Welcome, University of Cincinnati. I'm going to open up to the judges and get right into it. Fred.
A couple of questions. Just a quick one. You mentioned that you're going to get the mineral layer from restaurants and construction sites. It seemed like an interesting pairing. What are you going to get from each of those?
Yeah. Oh, sorry. I'll go ahead and answer this one. Our composition is actually going to vary based on the climate that we are aiming to create this life brick for. So the life brick specifically designed for New York City was going to work specifically with the zebra mussel invasive species that is infested the Great Lakes there, which have a great amount of limestone within their shells. So we plan to compost that and create that into a bit of a mineral layer. In addition, using restaurants in the surrounding New York City area would just be an example of using something like mussel shells or something that creates that kind of mineral layer that we would be looking for, has those components in grinding those up and creating that bottom kind of layer.
Okay, and then the second question is more about, you mentioned how difficult it is to garden. This is this an easier solution than people who get hay bales and grow everything in a single hay bale, or that move away from soil completely in the city?
Well, what makes LifeBrick so essential for people who are moving away or who have limited access to resources is that LifeBrick has all the soil layers in it and has the seed implanted in it. So when we're trying to combat food insecurity and trying to help people who have the least of resources, the only resource LifeBrick really critically requires is water. And so that's what makes it most effective for people who are moving away from soil or for people who have little to no resources or space for gardening.
Very good. I didn't get that you were actually going to put seeds in that. Very interesting. Okay.
All right. Heather, do you want to ask next?
Sure. Yeah, I have a couple of questions. One, you sort of mentioned this originally just now, but I wonder if you could expand a little bit on precisely what kind of area you need in order to implement this. Like, is this something people can put on their stoops? Do you need access to raised beds? Like, where do you see this actually functioning? Because a lot of people in New York City don't have access to any kind of outdoor space at all. I'm also wondering if you had any kind of conversations with the very large and pre-existing networks of community gardens that exist in New York City, and asking them sort of what they need in terms of resources or other ways to think about expanding those networks.
Great, thank you for the question. So I think one of the main things we were thinking about in some of the earliest stages of our project development were how to kind of combat barren landscapes and kind of recreate a barren landscape into a viable space for growth. So the one thing that we have kind of always brought along with us on this development is like, how could we turn a parking lot into a garden? And so I think one of the main goals of LifeBrick is to challenge what spaces could be used for that. And by creating this kind of modular structure, we're kind of aiming for it to be used in different spaces. It doesn't have to require a raised bed. It doesn't have to require a kind of containing structure, as long as it has the space for it to break down. So, and on the question, sorry, excuse me. So on the question about community discussions in New York City, we actually were not able to get into contact
in that way, but that would be a amazing direction to go in and to speak with people that require some of those needs.
Great, thank you.
Jean.
You refer to this as like a brick. So, and I think the last speaker just talked about not even needing a pot. So my question is, you have a lot of loose materials and you're putting into a strata. So are you going to put all these loose materials in, and then are you going to saturate it with water and then desiccate it, kind of like as you would with a brick model? Or how do you imagine taking all these kind of loose materials and kind of constituting it into a unit? Is my first question. And then as they are a unit, I am a New York gardener. So I take any little spot I have, which is like a very minuscule. The soil is really poor. So we've had to do a lot of composting and a lot of building up sort of the biota of the soil. And so I'm wondering, if the seeds are at the top of the strata and I need a really large amount of your bricks, can I put them on top of each other? Or do you see this as being kind of like customizable as well? And then I guess I have three questions. And then in the city, there are a lot of people from all over the world who also garden, which make it a really exciting gardening space. So even in Brooklyn, you'll have people growing things that are indigenous to their home countries and stuff. So again, as you are thinking about this as a commodity, is there room for people to kind of independently configure these bricks with different kinds of seeds or no seeds and all that kind of stuff? So those are my questions.
I can answer the third question. So your third question closely aligns with terroir that we were mentioning at the end of our video, where you can grow. Say you wanted to grow a vegetable that was native to China. We're trying to create a brick that has the components of the soil in China so that this Chinese vegetable can grow effectively in the life brick. So life brick is evolved, or we're trying to evolve so that we can contain the soil layers necessary for different agricultural yields.
Okay.
Yeah, and I can talk a little bit more about those first couple questions. In terms of keeping the brick together as one unit, we imagine in the transportation process, it would be a dehydrated unit, kind of like you talked about, and then the user would set it up and hydrate it. And this is one of the conversations we had with Dr. Knable, who's the one of the national soil program leaders at the USDA. And this is where we began to institute cover crops, which are these quick crops will grow in anywhere from 2 to 4 weeks. And really provide the soil with significant amount of integrity that it didn't have prior to this crop growing, building roots down into the soil, and then also adding additional organic material to the soil itself. So we hope that, and we aim for that cover crop to provide the soil with that integrity, so you don't just buy a life brick and it blows away on your roof. So that's the aim for that main aspect of the soil. And yes, sorry to answer the very last question, I believe. We do imagine LifeBrick to be a very customizable, user-friendly product. So if your needs were to need, like you said, a LifeBrick that stacks on top of each other, that's something we definitely aim to accommodate for.
Okay, great, thank you.
All right, Andy.
It's a really quick question, I promise. There's a lot of stuff that you guys are layering onto this, and so I was just wondering what the hypothetical, or what you think the weight would be for these, just because how do you distribute these, and is it going to be more effort to get these bricks around or than just the materials themselves?
Yeah, I'll go ahead and answer this one too. So in about a cubic foot of soil that you might buy at a Home Depot or something like that, that can weigh anywhere from 40 to 60 pounds. And about a third of that's going to be water, and about the other, maybe I think like around 50% of it is going to be from the mineral substance in the soil. So because we kind of concentrate that mineral substance in one layer that can diffuse easily into the soil, and in the transportation process, our soil bricks are dehydrated, we're aiming for anywhere from around 12 to 15 pounds for our one soil brick, which is going to be a cubic foot. So by no means is it light, I'd say, but it is user friendly when compared to the current alternatives out there now.
Thanks.
I think it's also worth mentioning that prior to COVID-19 and us being split apart, we were going to create a brick on our own just to actually be able to test it out. And we did have a local farm, Turner Organic Farms, that was going to test out our product so that we would actually get quantitative data on how effective this brick really is.
All right. Thank you so much, team, University of Cincinnati. And thank you, Andy, for starting us off and leaving us on a high note. All right, that concludes the day. Thank you so much for your students, for your wonderful presentation and project. Thank you, all of the judges, for being with us in this session and those previous, and those tomorrow. I'm very excited for our future if it looks like what we've seen over the past 2 days. If you couldn't join in, we will be posting the live streams on our YouTube channel as well as on our summit page where some of you might be watching on our BDC website. Hopefully you can join us tomorrow as well. We'll be starting at 8:25 a.m. I know it's an early day in Eastern time. And then no programming on Thursday. And then please join us on Friday again when we will be announcing the top 6 winners. We'll have 2 really phenomenal speakers, David Benjamin and Annalie Newitz, in conversation with Troy Bosch. You can read their bios on our BDC page as well. Hopefully you will be voting in our Community Choice Prize. Just a reminder to vote before Thursday at noon. And we will see everyone tomorrow morning. Thank you so much. That's it.